there. We're saying that reproductive isolation characterized by animal displaying specific action to attract the mating pattern. Remember, even I gave you an example that except the humans, they like to copy, and they say that. Okay, we call it species specific courtship behavior, and I said that the way how you do your dating is not the same way how frogs, how dogs, how cats, and I said that except but humans, they like to copy. Yes, all right, so this one becomes the most correct answer. So we go to the next one. Then you're saying that which one of the following genotype is homozygous dominant? Homozygous. So I need to look for letters which are the same, but they say the dominant. So this one is dominant, this one is recessive. So it is C. So C becomes the most correct answer. They're saying that how many alleles control blood groups have been singing this song? We say there are 3: allele for A, allele for B, and allele for I, allele for O. So we said it. 2 marks there. Guys, the paper was flowing. Okay, which one of the following is an example of discontinuous variation? We say that anything which you are born with and it's exactly the same way it is. We say the blood group. We say the height is different, the skin color. You are born yellow bones, now you are dark skinned like me. Color, the hair color keep on changing. You understand? So the answer here is B only. If you are blood group A, you are born blood group A, you will die blood group A. So that becomes the answer. So 2 marks there. Don't get disappointed if you see that your answer they are not like the same. Yeah, still at least if we get maybe 80 out of 8 out of 100, maybe 80%, that's a distinction. Yes, so to fall in a distinction doesn't mean that you have to get 100%. No, it's 80%. All right, we continue. Which one of the following is a correct description of stem cells? Stem cells, remember that we say that these are cells which are unspecialized. They have ability to divide and form specialized cells, meaning that they have specific function. So we have to look for that. So we have to look for cells, specialized cell that divide by meiosis, by mitosis, divide by mitosis to produce cells that become specialized for specific function. So B becomes the most correct answer. We go the next question. We are saying that a list of different lines of evidence to support the evolution given by we say that the 3 evidences, we say that fossil evidence, genetic evidence, cultural evidence. I don't need even to read the rest. Those are the 3 we talked about. So we look for it. 2, 3, 4. 2, 3, 4. 2, 3, 4 is C only. So we go to the next question. Say that the police arrested 2 suspects accused of housebreaking. Their DNA profile was analyzed and compared to the profile DNA. Okay, let's collect it from the crime scene. So they're saying that what conclusion can be made from the DNA analysis conducted by the police? So we need to look at, are they corresponding? No. They are not corresponding. No, they are not corresponding. No, they are not corresponding. No, they are not corresponding. It looks, but still this one is a little bit up. No, they are not corresponding. No, they are not corresponding. What corresponds here? Nothing. Only this one corresponds, but this one no. This one corresponds, but this one no. The answer is no, meaning that there is no suspect. The suspect here, no one who did that. So the answer here we have to look for neither of the suspect are positively linked to the crime. So D becomes the most correct answer, because there's nothing. If they were matching, they're not matching. Therefore, the answer becomes none there. So those 2 marks. So we got the next question, the terminologies. I hope guys you got 18 out of 18. If you got 18 out of 18 or 14 out of 18, congratulations. Clap your hands. All right, we got the next question. Alternative form of a gene, we say this allele. Allele, DNA containing organelles found only in plants, chloroplast. Plast. Yes, they are saying that the diagram representation showing the possible evolutionary relationship among the different species. We call it what? Call it phylogenetic tree. Yeah, we call it phylogenetic tree. Allele that is masked in a phenotype, masked meaning that if even we say that if the vice president is there, if the president is there, the vice president cannot talk, meaning that there is that allele which is being masked. So we call it a recessive allele. So we say recessive allele. They're saying that the permanent disappearance of species, we call it extinction. The deliberate breeding of organisms for desirable characteristic, we call it artificial selection or selective breeding. Artificial selection. They're saying that genetic disorder characterized by absence of blood clotting factor. We said it when we did a lot of examples here. Hemophilia. Someone is gonna say, does spelling count? It counts. A group of organisms of the same species, that habit. A group of organisms of the same species in a specific habitat at a particular time. How do you call that? We say that it is called a population. We say that, hey guys, give me happiness. I told you that when I come back here, I need happiness. Yes. Chromosomes pair in 1 to 2. We call it autosomes. There's nothing we didn't do, guys. There is nothing we didn't do. A representation of number, shape, and the arrangement of all chromosomes in the nucleus. The moment you see the nucleus, that a nucleus we said is called the karyo. So it's a karyotype. We talked about it. All right. They're saying DNA containing material in the nucleus of a non-living DNA. How do we call that? We say that we call it chromatin network. Before the DNA divides, before the cell divides, it is chromatin network. Yes. Because that's why they said a non-dividing cell. So it's B only. They're saying that location of DNA. Where do we find DNA? We find in a nucleus, in the chloroplast, and the mitochondrion. So the answer is both A and B. We continue. They're saying that the first alhamdulillah. People saying 3 out of 10. No, don't. Others saying 10 out of 10. Don't cry, man. Who's there? Zanele. And then who is that? Guys, Kahiso. Alright, guys. Tabom. Alright. We continue. They're saying that by the way, alhamdulillah means praise to God. Yes. They're saying that the phase between two consecutive cells, there is nothing here. Remember, it's called a growth phase. Inter in between the two. You understand? That's why I said use IPMET. Interphase means in between the two phases, the two growth, the first growth and the second growth. That's it, interphase. So the answer here is none. 2 marks. We go. We kill the paper. We said we're gonna smash the paper. The diagram below represents two biological processes. So if you look at this, we see this is transcription. Why? Because you are forming a messenger. Check U here and then check T here. So that is transcription. And then if you look at this is T, this is T, T meaning this is DNA replication. So okay, there are some questions I ask you to do. Diagram 1, transcription, we said. Diagram 2 is DNA replication. Identify X. What is X? If you look at X, where is X? X is a messenger RNA. They are looking for this molecule. It's messenger RNA. This is messenger RNA. So correct. Someone saying that we'll try next year. For us, we will be here always to help you. We will be here, God willing, helping you so that you get what you want. Yes. So keep yourself on Thundereduc and other channels, Facebook, TikTok. Yes, we are always there. Yes. But there are some people who are having different channels of Thundereduc on Facebook. Those people, there are some people who have opened. So guys, please watch out. They are asking money from people. Please make sure that you don't become a victim of sending money to people who are requesting you. They pretend to be Thundereduc. Yes. So we continue. If you are going to send the money to Thundereduc, for example, you are buying a book, it must be an account which is linked to Thundereduc, no e-wallet. Yes, all right, thank you. We continue. We are saying that identify the molecule X. So we done that with that. Molecule X, what is molecule X? Is messenger RNA. We say this is the messenger RNA. Natural shape of Y, we talked about it, and I said only one answer. Natural shape of this DNA is double helix. Double helix, nothing else. They're saying that the phase of cell cycle during which the process in diagram 2 occurs. Diagram 2, that is, these are the questions I asked you. We said it's interphase. When does it occur? It occurs during interphase. Full name of the nitrogenous base that is found on molecule X, not Y. Molecule X, meaning that it is found on messenger RNA, but is not found in the so the answer here is uracil. They're saying name the sugar found on X. Sugar found on X is the RNA, therefore it is ribose sugar. It's ribose sugar. Someone is telling me that, hey sir, write nicely. Sometimes also when we are marking you is the same thing, you don't write nice. All right, bond Z. Obviously when they ask you bond Z is this one, is this one. So it is weak hydrogen bonds. Weak, I told you in the word weak must be there. Hydrogen bond. Yes, please make sure that is there. They're saying state the number of codons. We say that codons are three bases on X. So we're gonna count them. How many codons are there? This is 1, this is 2, and that is 3. So I see. Did I say on X? No, man. That is not X. Let's check properly. On X, we are seeing it here. It is 1, it is 2, it is 3, it is 4. So it's 4 bonds. So the answer here is 4. They're saying that not 4 bonds, 4 codons. Then we continue to the next question. 1.5. We go to 1.5. In a plant species, gene coding for plant height has 2 alleles. The allele for tall is capital T, the dominant over allele for dwarf, which is small t. The gene for flower color has 2 alleles, where allele for red, okay, and then allele for is dominant over allele for white, which is small r. Dwarf and white plant is crossed with dwarf and white, okay, crossed with homozygous tall, meaning is like that, crossed with the heterozygous red, meaning that red, remember, is heterozygous. Okay, state the characteristics that is being investigated in this. So we have height. First of all, we have that, and then someone is gonna say height. Okay, cool. And then we have height, and then we have, they said flower color. Flower color. Cool. They're saying that give the genotype of the plant that is homozygous tall and heterozygous red. Give the genotype homozygous tall. They said homozygous tall and heterozygous red. So it's like that. So meaning that you get a mark there, you get a mark there. Below is incomplete Punnett square. Okay. We say that what is the genotype of X? I will just say, remember this goes with that. So I'm gonna say capital T, small t, capital R, small r. get that 2 marks. A is 1 mark. Then they are saying that the phenotype for Y, meaning that if the phenotype for Y, I will need to know what is Y. So it's gonna be capital T, small t, small r, small r. Capital T is tall and white, because small r is white and white. So it's gonna be tall and white. This is the answer. I was just using this to identify what is it. What is this Y? So it is tall and white. And then this one, you get that mark. That is dihybrid cross. I told you it won't be a big problem. This one, the way we set it, the way we teach it is different from the way we set it. It's simple. That question is done. We go to the next question. Don't tell me that you didn't do this, because we repeated it several times. Diagram 1 and 2 show the cells of the same organisms in different phases. They are not on scale. I told you, when you see it, these spindles must be in these sides, facing that side. And I told you that when this diagram comes, what should you do? Just rotate your paper, ne? Just rotate your paper to look like this, so that now these things, these spindle fibers and the centrioles, they are here. So don't you see that there is a proper space in between, which shows that this is the anaphase. And then even this one, you see that there is a proper space. This is also anaphase. So if you are confusing it, now at least you know what you're supposed to do. For those people who are using this paper as a revision. Okay. Now, we continue. We are saying that state the phase of the state 1 place in human body where meiosis occurs. Meiosis occurs, we said the testis. We said in humans, we said it is testis. This is singular, this is plural. Or ovary. They said in human. They didn't say a male or female. So we go. Structure P is spindle fibers or spindle threads. Structure Q, we say it is centrioles. Yes, a centriole. Identify the phase of cell division that is represented. I showed you, meaning that they are being represented by what? Diagram 2. Diagram 2, remember there is a space here. I told you to rotate it. So meaning that it is, since these are chromosomes, they are still together. Chromosomes, we talk about chromosomes. Therefore, it is, they are still 2. Meaning that they are separating these homologous chromosomes. They are separating. It means that separating apart. So it means that is anaphase. Since they are still 2, meaning that it is anaphase 1. Anaphase 1. Remember we said if they are still double, then we say 1. If they are single, then it is 2. You understand? We do the opposite, ne? Like this one, ne? They are chromatids. The chromosome is the one which separates. So it's single. Therefore it is anaphase 2 in diagram 2. In diagram 1. All right. So in diagram 2 is anaphase anaphase 2, anaphase 1, because their chromosomes are still double. And they're saying that how many chromosomes will be found in a somatic cell, of this organism. They didn't say that this is a human cell. That's what I told you guys. Don't talk about human cell 46 chromosome when they say that. This diagram. So how many are they? 1, 2, 3, 4. In meiosis 1, those cells, those chromosomes are going to be 4. Why should you so talk about 46? Did they say that this is a human cell? No, I told you that. I hope you guys didn't disappoint me. So you deserve a trophy. Thank you so much, Mona Lisa. All right. Oh, I like that name, ne? Mona Lisa. Yeah, you know that Mona Lisa there. Yeah. All right. State 1 difference between the process occurring in diagram 1 and diagram 2. State 1 difference between the process occurring in diagram 1. Diagram 1, meaning that you just gonna talk about here the chromosomes separate, they move to opposite poles. You understand? While here the chromatids move to opposite poles. That's it. Simple. This year, May, June, they did how do I say it? They need a trophy for you guys. You need to be getting good marks. At least you continue with your life. Most especially people who are writing May, June exam, these final exams. There are those people who are, maybe they have gone back, or they are upgrading. Maybe something did come up right, or they missed up the exam. They are not fresh. Those are people, it could be their first time, but their age is a little bit higher than those ones who are writing November, October, November. So you see, so thank you so much, the person who said, however much, yes, there are some questions which are not good for those who didn't prepare. But if you prepared with us, it means that you got it. All right, chromosomes in diagram are genetically different due to they are genetically different. Chromosome in diagram are genetically different due to crossing over. All right, I told you, don't go to the paper without knowing. Without knowing the process of crossing over, and we discussed it. Describe the process of crossing over. Talk about when does it occur. In prophase 1, what happens? Homologous chromosomes, they come together or they align. They exchange their chromatids. Yes, homologous chromatids at a point called chiasma. Yes, and then we say that after that, what happens? They, after exchange their genetic material, they separate in such a way that new genetic material from both is being shared. So basically this process, even I brought a diagram to explain to you. So guys, I hope you got this process right. You see such process, and then you get your mark. So yes, those people who are doing the revision, you can take a screenshot. You pause it, take a screenshot. Now you know what you're supposed to write when they bring such. They're saying describe the non-disjunction. I explained this. How does it lead to Down syndrome? And I said, hey, when these are some of things which you might not miss out. Yes. So I said it here. Where are you? Yeah, after this. Yes, I explained it here. First of all, you need to explain to us non-disjunction. When does it occur? What happens? So basically, we say that you explain that it occurs in anaphase. Yes, in anaphase. It can be anaphase 1 or anaphase 2, meaning that the failure of the chromosomes to separate. It tells us that the chromosomes fail to separate. Yes, meaning that one result in one cell having an extra chromosome, meaning that it's going to have an extra chromosome. It's going to have 3 instead of what? Instead of 2. Or another one is going to have 1 instead of 2, meaning that one cell is going to have an extra chromosome, and then another one is going to have less of chromosome, meaning that it's going to have 23. Another one is going to have 24, because it's supposed to be 23. Another one is going to have 22. So if this 24 fertilizes a normal ovum or a normal gamete, yes, because we don't know what is this, a sperm or ovum, a normal gamete of which is 23, is going to result in a cell. Is going to result in a cell having, or an individual having, 47 instead of 46. And if this non-disjunction occurred on chromosome number 21, therefore it becomes trisomy 21, which we call Down syndrome. Yes, so that's what you're supposed to explain. I explained it. I can't repeat it now because take a screenshot. Because we're just discussing what you are supposed to be linked. Yes, I hope you guys, you got 10 out of 10. There we go. The next question 2. Question 2.3. The diagram below represents the stage of protein synthesis. Name the stage of protein synthesis there in diagram 1. In the diagram, we say this is the moment you see these boxes. Remember, I said that amino acids, they either circles or they are boxes. They brought them as boxes. So cool. State the process. We say this is the translation. Name the stage of protein synthesis. So we say this translation. Then they are saying that name the process. Name the stage of protein. Okay. State the site where the process named above occurs. It occurs on the ribosome in the cytoplasm. In most cases, they want to see the word ribosome. Sometimes they want to see cytoplasm. So that's why I told you that I prefer you to say that, for you to say that ribosome in the cytoplasm. Definitely they will give you. If they want this one, they will give you. We have to be there. You said diagram one. Someone was okay. I was just reading one of the comment. The diagram one opposite chromatid put opposite pole. Diagram two chromosomes are put opposite pole. Yeah, that's it. That's what I said. Yeah, that's what I said. Tabo, Mona Lisa. That's what I said. Cool. It's cool. All right. We go to the next question. Which question is that? Describe the role of X in protein synthesis. Role of X. What is the function of this in protein synthesis? In actual sense, they're asking you, what is the function of transferrin A in protein synthesis? It brings the required amino acid or the specific amino acid to the ribosome. Yes. So if you want to add, yes, when the anticodon of it matches with the codon of the messenger. When the codon of the messenger matches with anticodon of molecule X, yes, molecule X will bring the required amino acid to the ribosome. If they're asking you to describe that part, just to remove that part of what? Of translation. Yes, that's what they are looking for. So basically, this is what I'm talking about from our distinction material book. This is what I'm talking about. I told you that this book, I don't want to show you everything, but if I want to show you each step by step, you would have seen. So you see, this is what I'm saying. Transferrin A carries specific amino acid when the anticodon matches the codon of the messenger. Therefore, that molecule X will bring the required amino acid to the ribosome. So this is what I was trying to say. You see how many marks are there? There are a lot. All right, we continue. This identify the complementary base from left to right, representing anticodon 2. Let me first clear it up, and then we see anticodon 2, anticodon 2, anticodon 2. So anticodon 2 is anticodon 2. We are seeing that CAU. They're saying identify the complementary base. Identify the complementary base from left to right, representing anticodon 2. So it's gonna be C goes with the G, A goes with the U, and U goes with A. So you get the mark there. They're saying on DNA, codon 1. So I go to codon 1 is, if I pick this one, it's gonna be codon 1 AUA, AUA, AUA. So I need to convert it into DNA. A goes with T. Remember, this is DNA, and then U goes with A, and A goes with the T. Third. Table below represents the anticodon for different amino acid. The mutation, I told you this one is coming. Make sure that you do. Then they're saying that Maria, we have not finished the paper. Why do you say that you didn't? You failed it. I see you. All right. They're saying that a mutation occurred during replication. The mutation, I know that this one is gonna kill some people, and that's why I emphasized it. Make sure if you don't do it, you are gone, man. All right. They're saying that use the table to explain how this mutation will affect the protein. So we want to know whether the protein remains the same or is gonna change. But remember, you need to show us where did it change, which codon number or which anticodon, which code, all those things. Yes, but now they have given you, this time they have given you what we are supposed to get. So what they say is the mutation, what happened to the mutation? The mutation changed this to this. All right, let's find out prorine to arginine. So this one, what does it code? I'm just doing this so that you guys are able to get it right, ne? And then what is adenine is CGU. What is this one doing? This one is gonna go back to CCU. So C goes with the G, and then C goes with the G, U goes with A, and then so this is the anticodon, ne? So this is codon. Because they say that use this explain how the mutation will affect the protein. The mutation occurred during replication. So replication, it means that it occurred on DNA. That's why you want to find out which code from DNA where the problem came from. Then this one, C goes with the G, G goes with the C, U goes with the A. So this is codon, meaning this is messenger. And then now let's go to DNA so that we know exactly what happened. They're saying that C, A goes with T because it is DNA. So it's gonna be G, C, it's gonna be C, G, T. You understand? So what happened if you look at it? The second base C was replaced with G on DNA during replication. So that's the mark. You have identified what happened. You understand? But they say 7, so it means that you have to show which is transcribed as GGA and GGA respectively on what? On messenger, and then CCU and G and CGU respectively on the transferrin A. That's anticodon. Hence proline and adenine, which are 2 different amino acids resulting in different protein. You see? So they wanted you to identify what happened here. They will give you a mark there. You translate it to messenger. They will give you a mark there. They will give you a mark there. And then, yes, that codes for that. They will give you a mark there. And then they will give you a mark there. So they will keep giving you marks there. So you explain what happened. So that mutation, you always, if you see that the amino acids are different, it results in the same protein. Sorry, in different protein. If the amino acids are the same at the end, result in different protein. All right, I hope you guys got it, né? Yeah. We got the next question. That is question 2.4. They're saying that in some chicken, feather colors are controlled by alleles of alleles, feather, W and black. This is white, and then this is black. But look at the letters are both the same, meaning that they are dominant. So it might be related to incomplete dominance or codominance. If the heterozygous white chicken is crossed with the homozygous black chicken, offspring have the feather that is both black and white. And then they are saying that known as so when we are using the example, we use that one of flowers. Yes, we use that one of flowers, and then we explain it. Explain why this inheritance is an example of codominance, because it results in offspring having both colors, that is white and black and white, meaning that it's a mixture of the 2 colors. So I told you that is a mixture. It's a mixture of 2 colors, that is black and white. Then we got the next question. They're saying that explain why this the inheritance of blood group in human shows the same type of dominance. Explain. Yes, because when A is crossed, homozygous is crossed with B, homozygous, they will result in AB, which is the codominance. That's it, meaning that A is dominant, B is dominant, and then you form AB, which is, it looks the same like this one, because white and black, then you produce a mixture of the 2. You see that this looks like that. White chicken is crossed with the eminent chicken. using the genetic cross, meaning the eminent is this, ne? Is this, yeah, cool. Use the genetic cross to show the phenotypes. Yeah, this one is simple, guys. Remember I told you that when you have this, you have 3 marks free. So P1, F1. So you got, I think you got that mark before you go to the paper. You went to the paper, they said phenotype, we said genotype, we said meiosis, we said gametes, we said fertilization. So we say that you got this mark also. So those are 2 marks. And I say that for phenotype, you go to the question, they say that white chicken. So I said white chicken crossed with eminent chicken. That's another mark. You see, 3 marks. That's what I said. So you got it. So now they're saying that genotype, we say that white chicken, they said white is W, isn't it? Is W, so it's gonna be capital W, and eminent is capital W, capital B, because it's a mixture of the 2. Then they say that meiosis, we say that capital W, capital B. Then we say that capital W, capital B. Then these are gametes. Then we say this cross with this, capital W, capital W. You can use the Punnett square. Capital W, capital B, correct. Capital W, capital W, and then this with this, capital W, capital B. These are offsprings. What do they want? They say the phenotypes. So if they were looking for the phenotype, so I will come back here and I will say that if they're looking for phenotype, I have 1, 2 white chicken, and then 2 eminent chicken. Yes, 2 eminent chicken. So basically that's it. You got it. So they start to mark. But you went to that paper when you know they wanted to see this. This could be a compulsory mark, that if you didn't get it, instead of getting because there is a lot of marks here, there is a mark there, there is a mark there, there is a mark there, so there is a lot of marks there. All right. Then we got the next question, which is question 2.5. Question 2.5. They are saying that hypophosphatemia is a sex linked. They said it's a sex linked. You see, I said whenever they say sex linked, you use X and Y, inherited condition that is caused by dominant allele capital X and on X chromosome. They said dominant. You see, dominant. Yes, it is as a result of abnormal 2 level of phosphate. It's as a result of abnormal low levels of phosphate in the blood, which can cause rickets. They're saying that the recessive allele small h code for normal, meaning that if you have to be with both like this to be normal, or you have to be with this to be normal if you a male. All right, they're saying that male without. Okay, they give the type of diagram shown. How do you call this? We call it a pedigree diagram. They're saying gonosome found in male, gonosome found in male is Y. Yes, it's gonosome, meaning that the chromosome which is found in male. Yes, yeah, male only. They don't want you like this. They said male only, meaning they are looking for that chromosome which is Y. State 2 effect of this, they said it here. It will cause rickets and also causes abnormal low level of phosphate in the blood. So they say that how many individuals in F1 generation have this condition. We say that the one which is shaded, remember this is shaded, the one which is shaded is suffering from the disease. They said in first F1 generation. This is F1 generation, but this got married, so meaning that it is this one, and this one, meaning there are 3 individuals in there. So 3 individuals. They're saying that all possible genotypes of individual 3. All possible genotypes of individual 3. Where is individual 3? Let me clear it up. we say that individual 3 is normal. And normal, they said that you're supposed to have the recessive allele, and this person is normal, so this is a male, so it's gonna be like that because he's a male. And then individual 4 is having the condition, meaning that is gonna be X capital H and then X small h. Why not X capital H, X capital H? Because they produced a person who is a female and is normal, meaning that this one is like that. So meaning that one came from the father, one came from the mother. So the mother can't be like this. That's why this is the only option which is there. So you get 1 mark there and you get 1 mark there. Explain why all the daughters in one will have the what? Will have this condition, and they say the 6 marks. So do you know what they want? They want you to cross, do a genetic cross daughter. Yeah. So we are going to show you how are you going to explain. We say that the male, who is the male? The male is here. We say the male is what? Is sick, meaning that if the male is sick, it's gonna be capital H, meaning that the female is like this, isn't it? And then the female is like that. So when I cross, okay, after that you're gonna explain. When I cross, yes, okay, I have that and I have that, and then the female is gonna be small h, yes, you understand? So now we want to find out if all the daughters are going to be sick. Then from there we're gonna explain why. Then this gonna go like that, capital H, small h, okay, that is a daughter, is sick because it has a capital H. Then this gonna go with that, so it's gonna be capital H, yes, and then small h, okay, this is also a daughter. Then now this with that is gonna be capital H, small h, and then y, this is normal. Then this one with that also gonna be capital H, small h, and then y. So you see the boys are normal while the girls are sick. Not sick, they have that condition, you understand? Now how are you going to put this in wordings? You understand? So when we come back here, we see that the boys are normal, you see, and then the females, all of them are sick. So what does it mean? It means that the males have a dominant allele, you understand? And then the females, they have 2 recessive allele, you understand? So the male will only transfer the Y chromosome to the males, you see? They will only transfer the Y chromosome, these Y chromosomes, to the males. Are we together? Meaning that all the X chromosome which are dominant, causing the individual to be sick, are going to be transferred to the females, you see? The ladies, the female, the mother, will only transfer the recessive allele, which will be masked by this dominant allele, causing all the females to be what? To be sick with that condition. Which condition is that? They said hypophosphatemia. Yes, I think now you get it what I'm trying to say. Yes, I wrote 15 line, yo. Yes, it's fine, because you were just supposed to explain to us that the male have the dominant allele in this case, and then the female has 2 recessive alleles. The male, the Y chromosome is going to only be taken to the males, meaning that the dominant allele on the X is going to be transferred, or is going to be inherited to the female offsprings. You understand? The daughters. You understand? Yes. The recessive allele from the mother would be masked by the dominant allele, which was transferred, which was inherited from the father, making all the females to be having that condition of hypophosphatemia. Hey. All right. So someone saying, yeah, I get it. All right. Thank you. Since you got it, thank you. We go to the next question. Is just it was a dominant allele, and then I said that when they bring dominant allele, people go off. All right, we continue, guys. Thank you so much if you got it. And thank you for if you get it, if you have a challenge with the question, you tell me. All right, they're saying that describe the describe. They are saying describe the process of cloning. So what do you say when it comes to cloning? We talked about the process of cloning, and let me just use the distinction material book and then just to show you what you are supposed to talk about when it comes to the process of what? The process of what? Cloning. So we said that we use a sheep which we call the dolly sheep, but it's still the same process. Use the same what? You see the same process of cloning. It is. So it means that you have to first identify which cell are you going to use. You understand? Yes, you have to first identify the somatic cell. Get a somatic cell. We said get a somatic cell from an individual whose characters are needed. Yes, step number 2. Then remove the nucleus and then keep the nucleus. Then step number 3, get another cell, a mature ovum from another sheep. Yes, and then after that, basically remove the nucleus and keep the cytoplasm. Yes, or the casing. And then after that, combine the nucleus of this and this and the cytoplasm of the cell from the other sheep. Then with mild electric shock, then after that it will form a zygote like cell, which is allowed to undergo mitosis to form a ball of cell. So those ball of cell is gonna be split into small embryos, which are going to be inserted into the what we call the surrogate sheep, so that now the surrogate sheep can develop that embryo into a mature or a baby of that sheep. So basically that's what you're supposed to explain, link to this, and I explained, said this is what we want, nothing else. Yes, and H, there's someone who said that H, you talked about it, but I neglected it because I knew that it's gonna come in objective. Hey guys, I'm sorry. We go to the next question. Name 3 benefits of cloning. Basically, the benefits of cloning, we can use it to get medicine. Yes, like this. Where is that? Yeah, to get medicine like this. Basically, they are looking for this. To get medicine. Where is it? Yeah, medicine. Yes, medication. Basically this is what I wanted to say. This cloning of insulin. Yes. We can also be used for producing identical organisms, which can be used for research purposes. Yes, process of cloning. Yes. And then we can also use basically what else can we use for cloning? We can use it for research. We can use to do the medication. Yes, and then we can use it to produce. There is this scenario also which occurs, like preserving the endangered species. Like if the species is about to get extinct or is about to get extinct, we can bring that genes and then we clone them, and then we come back, we bring them back to life, like so that we multiply them in large numbers instead of getting extinct, like one organism remaining or 2 organisms. So we can do the cloning so that we multiply them to be many. Yes. All right, someone said doesn't know the process of cloning. I did a full video about cloning. So just go to YouTube, type cloning process, and with the word Thundereduc, it will come. Yes, those people who are using the distinction material book, this book, then it is being explained based on this distinction material book, meaning that at least, yes, you'll be able to use the same information. Let's go to question number 3. What are they saying? Question number 3, read the extract. Sickle backfish are small fish found in the ocean of the freshwater lake. The one in the ocean have spike fin for protection against predators until about 100 years ago. Most of the stickback fish live in the ocean, but the last ice age ended. The melting ice created new lakes and some population swam into the lake. The connection between the ocean and the lake eventually died up, trapping the stickleback population in the small lakes for thousands of years. Over time, the stickleback fish population in the lake lost the spike fin. Okay, how will Lamarck explain this? Remember, this is absence of this. How did they become with absence? Remember, Lamarck uses law of use and disuse and the inheritance of acquired characteristic. So what are they saying? You need to explain based on Lamarck. So you're gonna explain to us that there was no variation among these fish. All of them had what? Had the stickback or spike fin. All of them they had the spiky fins. Okay, that's step 1, meaning that there was no variation. All the fish they had the spike fins. So number 2, these fish, they stopped using their fins because there was no need for them to use. Remember, they were using them for what? They were using them spike fins for protection. They are using them for what? For protection. But where they went, there were no predators which were against them. So they stopped using their fins because there were no predators acting on them. So eventually the spike fins started to decrease and then eventually disappear. You understand? Yes. Then they passed this character of no fin to the next generation, and then all of a sudden, all fish in the fresh water they had no spike fins. Yes. So you explain based on what is being asked in that regard. Yes. So you link. You don't use giraffe. You can use the same concept, but you in this case you talk about the spike. And I said that last time, I said that they might bring it in the disuse. You have to use one. Either use it and then they develop, or you don't use it and disappears. Here you don't use it, then it disappeared. Why? Because there was no need for them to use the spike fins to protect themselves from predators, because where they were no what? There were no predators. Therefore they stopped using them, and then all of a sudden the fins started to decrease in size, and then they disappeared completely. They passed this character to the next generation. The next generations, they were born without the spike fins. That's Lamarck's theory. The diagram below represents the skull of pelvis of human and chimpanzee. No particular order. Cool. They're saying the name 3 structure differently. We did this several times. Okay, we say that draw a table, a complete table. Some of you did it just a T, 0. They're saying that is A and that is B. So they're saying A, you tell us. We use the same example. You people. We say that they have large canines, small canines, rectangular palates, U-shaped palates. Then you see the foramen magnum is in backward position. The foramen magnum is in forward position. What else do you want? You are shocked? Someone saying that I'm shocked I didn't see this question. Sorry. They're saying state Y pelvis C is regarded to humans. Remember we said that the one for human is short and wider. Short and wider. That's all. Guys, this is the things. I don't want to go there. This is the thing we said and emphasized. Say that, hey, when I don't make mistake to go there. Yes, I gave you that actual yes. Okay, we use this, but still I use the same thing. No, this is not what I want. This, yes, it is short and wider. It's the human. Remember, I say, see, it's a human. This is the bipedalism for human. Biped, because we walk on two limbs. You see, it's a human. So it is the answer here they wanted is short and wide. That's all. Nothing else. Short and wide. Question 3.4. They are saying that malaria parasite, I talked about this also. Malaria parasite is transmitted to human blood by mosquitoes. People living in malaria affected areas take anti-malarial drugs to prevent the infection. If the malaria drugs are effective, there should be no parasite in their blood. The presence of malaria even after treatment indicates they have evolved to be drug resistant. Explain. The research wanted to determine if the use of anti-malarial parasite could causes the resistance in malaria. Okay, they gave you blood sample was collected from people of this affected area. Okay, the collection of blood sample was done in several villages. Okay, and the same blood were kept in the laboratory and tested. Then the number of sample having the malaria parasite was counted. They're saying investigation was repeated 3 times. That is good. They're saying that in total, okay, that is good. They're saying that was collected, okay. They're saying how did malaria parasite transmitted? They said through blood by the mosquitoes. So that is the answer. They're saying what is the independent variable? The independent variable is, they said it here. They say that the researcher wanted to determine the use of anti-malarial drug caused resistance, meaning that the independent is anti-malarial drug. So that is the answer there. And then what depends is the drug resistance. Drug resistance will depend on which type of anti-malarial drug they are using, or you can also say presence or absence of malaria. It can be the presence or absence of malaria. They can also credit you, but you saying that resistance is a key point. Calculate the average drug resistance in the blood sample taken. Show your working around of your answer to. Okay, here you are saying that the percentage of drug resistance. They're saying that the sample were collected. 511 had malaria parasite, meaning that they were able to resist. This one they didn't have the parasite, meaning that I'm gonna say that 551 divided by 1485. 1485 times 100, which gives me 37.1. They said 1.1%. Name 2 ways in which you can increase or researcher ensured the following. Validity was done. Basically they use the same blood testing blood sample. They tested blood sample from the same individuals in the village. Yes, they said it here. The same area. Yes, meaning that the same village. Okay, cool. Then they also use this the same testing method. They didn't change the methods they used. They said they tested it, so they used the same method. And then how did they ensure, because it is past tense, how did they ensure the reliability? They used the large sample size. Don't just say large sample size. They used 1,485 individuals or samples. 1,485 samples. Yes. Then number 2, that's the large sample size. They repeated the experiment. They repeated it three times in the same 7 in this same village. That was reliability, that was validity. Yes, they repeated the investigation 3 times. Yes, that is also reliability. So you tell us what they have done. Yes. Then they are saying that describe how malaria parasite developed drug resistance. In this case, during our example, we used what you call DDT. Yes, we use this. With this DDT. How? So the first step, you need to talk about variation. The second step, you need to talk about selective pressure. The third step, you need to talk about the interference. The fourth step, you need to talk inheritance. The fourth step, you need to talk about the adaptation. Meaning that in this case, you tell us that there was variation among the parasite to the there was variation among the malaria parasite, those which are resistant to the drug, and then those which are not resistant to the drug. Yes. When the first application is done, yes, what happened? Those which are not resistant, they died in large numbers, while those which were resistant, they survived, but those were few numbers. You understand? Yes. Then those which survived, they pass their genes to the next generation. You understand? After many generation, yes, the resistant one, they increased in the population. You understand? Resulting in the resistant parasite. Therefore, the drug will become ineffective, hence resistance. So basically, you explain exactly this. Instead of talking about DDT, you talk about that malaria parasite. Instead of talking about DDT, you talk about the malaria parasite. That's it. That's what we call the application. The question, when the question comes, you just replace it. So that's what they wanted about these mosquitoes. Yes. So yes, then the last question, this one we did it. I don't know how many times we did it, but we did it. And I told you that don't make a mistake to go to the paper when you don't have this. This is what I told you. Don't go to the paper without knowing these questions. And I went through them, and you'll find out that all the questions they said, they are there. And they're saying that number one, how many genera are there? Yes, how many genera are there? How many genera are shown? You see, that's the question. And then they're saying how many genera are there. So you look at Adipithecus and Homo. So there are 2. So basically 2. Because this is Homo erectus, Homo neanderthalensis, this is Homo sapiens. All of them are Homo. When did the Australopithecus africanus became extinct? The question is there. When did the Homo habilis became extinct? You see, they're saying that. So Homo habilis, they say now Homo Australopithecus africanus. So it's the same thing. So it stopped here, meaning it's 2.0 million years ago. Then they are saying that name 2 Australopithecus africanus species in South Africa. Name 2 Australopithecus africanus species in South Africa. So this is what I told you, that we always ask you that when they come to the fossil, they're going to ask you name 2 Australopithecus africanus species, you know. So you can talk about Taumchild, Mrs. Place, or Littlefoot. They said africanus, Australopithecus africanus. Yes. So when you come back to that, yes, is this thing Australopithecus africanus species. That's when I was explaining it, I said this. They are saying that they use the only the species in the diagram describe the fossil evidence, meaning the species only. Remember we said that Australopithecus africanus, you give example, yes, and Homo habilis. Australopithecus africanus species only were discovered only in South Africa. And then here Australopithecus africanus species only in South Africa, and then Homo erectus, even this one, Homo neanderthalensis and the Homo sapiens, the oldest was discovered in Africa. This is what I'm trying to say. Yes, because based on the question, they only want, you see, this Ardipithecus, they didn't talk about it. They started from here. Australopithecus africanus, yes, and Homo habilis only found in Africa. Yes. And then the oldest fossil of Homo erectus and Homo sapien were found in Africa. I said that this one, you must use the word oldest. If you don't use the word oldest, you are gone. Yes, so, and I repeated it, guys, and I said make sure you write the word only, because if you don't say the word only, then you are gone. They're gonna cross it. So, and this word oldest, it must be there. Don't make a mistake to do that. That's what I emphasized you, and I begged you. I said, guys, this question, when it comes, make sure that you answer it properly. So guys, those who are revising with this paper, make sure that you use it now the next time they bring it. But still, in the next paper, we'll also predict for you, and then we think what is gonna be coming in that regard. Explain how the influence of brain volume on the complexity of the tools made by Homo habilis and Homo sapiens. How is the brain size? This question is linked to the question, which I asked you, that how is the brain size? This one, the last one, how is the brain size? Describe how the brain size evolution trend. How does the brain size show the evolutionary trend? This is the question which I said that make sure you answer those questions, guys. Yes. Explain how is the brain volume, meaning that the bigger the brain volume, the bigger the brain, yes, the higher the number of brain cells, meaning that the higher the intelligence. You understand? They're saying that the complexity of the tool, you understand? Basically, it shows that, as actually I showed you this diagram when I was explaining it. I showed you this diagram. Where is it? Yes, this diagram. And I say that as the brain size increases, yes, the number of brain cells also increases, meaning that the intelligence increases, meaning that as also the tools also became more advanced. So the brain size, as the brain size increases, the advancement of tools also increases. So as the brain size increases, okay, the advancement of the tools also increases. Then why? Because these tools require thought to be developed. You understand? Yes, that's what we said. And then, yeah. And after that, I told you guys, I wish you the best. And then it's the same conclusion. See that, yeah, it was 99% success prediction. Yes, and we will always predict for you, and we want you to pass. Stay with us, study from the beginning until the end. I promise you, you will never regret you studying with us. There are some people who try to study with us, they put some pressure or say, no, we know what we are doing. Yeah, we know what we are doing. Yes, and we know that step by step. You don't need to rush for something before you grasp and understand the concept. Thank you so much, guys. Thank you. Thank you all the people. Sabero, I see you. No cross night for all of it. Saying that geography paper 2 was released already. I didn't do a lot of useless mistakes. I'm sorry. So thank you, guys. Thank you. I see you. And I wish you the best in your life, and if things don't go right the way you wanted them, you can register again. You can register again by, I think, by what? By what? By August, yes. And then you never know, you might make it again. Thank you so much. See you again in the next class. Line for our September exam. Yes. However, we will try to highlight some of the areas where you can focus more related regarding to these upcoming papers. Also, especially for those provinces which have not written, and yeah, we can use it for final exams. and then you can also use it any exam as long as it has paper too. Anyway, let's start without wasting time. His Saidi as usual. Thank you so much for subscribing, liking, sharing, taking care of this channel. This channel wouldn't have been there without you and without your input for following us. Yes, let's go to question number 1, and then we see what is exactly they are talking about. Question number 1. Which organelle represents the location of extra nuclear DNA in animal cell? When you talk about extra nuclear DNA, remember that we have three kinds of DNA. The first one is the nuclear DNA. The second one is the mitochondrial DNA. The third one is the chloroplastic DNA. So now, if you have the nuclear DNA code of life, basically here we are looking at the nuclear DNA. But we have other areas where you can find DNA in the cell. So if it is a plant cell, we can find it in the chloroplast. So we call it chloroplastic DNA. If it is animal cell, then we're going to find it in the mitochondrion. But because the mitochondrion can also be found in plant cell, then it means that in the plant cell, we can have the nuclear DNA, chloroplastic DNA, and the mitochondrial DNA. But animal cells only have the nuclear DNA and then the mitochondrial DNA. So what does it mean? What does it mean? It means that they said animal cell. If you look at it here, we see that they're talking only about the animal cell. So our focus is going to be on the cytoplasm out, nucleus is out, the mitochondrion, it can be found in plant cell and animal cell. Chloroplast is out. So because this is plant, this is this one they said extra nuclear. So the nucleus, they have talked about it. So the only answer which is much more correct is what? Is C. So C becomes the most correct answer. So you get those 2 marks. We go to question 1.2. The main purpose of the genetic cross in the genetic crosstalk diagram is to measure the DNA sequence, not really determine the genetic mutation, not really predict the possibility of the offspring's characteristics, and then solve the solve the crime. So if you look at it here, we see that one of the function, not sequence, we see that when you do the genetic cross, you find what we call the offspring. So the offspring's characteristics can be obtained. So when you do a genetic cross or a genetic diagram, you're able to identify the possibilities of the offspring's characteristics. Yes, so the answer becomes C, so C becomes the most correct answer. Which one of the following is the significance of DNA replication? We see that chromatin network condense, that's the description which happens during interphase, so this is out. Complementary pairing of the nitrogenous bases, yes, it happens, but that's not the function. This will lead to what? So this one goes out. Weak hydrogen bonds break, yes, they break, but it's a description of what happens in the DNA replication or in the DNA in the protein synthesis. But in this case, they're talking about DNA replication, so that's not an answer. So this one goes out. The most correct answer is the only option which is remaining, and they're saying that the genetic material is doubled. You double the genetic material so that when the cell divides, when the cell divides, each cell must have equal amount of the genetic material. So that's the function of the DNA replication, so that it can bring about the multiplication of this DNA, so that during the division of the cell, the genetic material can be equal in each cell. So the answer becomes D. So D becomes the most correct answer, and then you get those 2 marks. Thank you so much for liking and subscribing. All right, we continue to the next point. The biological evolution refers, remember when you talk about evolution, it means that the increase in the complexity of an organism. So when you increase the complexity of an organism, so we call it you're evolving. Yes, But this, because it is about life, that's why you're linking it to an organism. So evolution can happen, evolution of a fawn, evolution of the sun, evolution of the moon, evolution of the what, but because it is about the organisms which have life, then it's going to become biological, because biology means, bio means life. So biological evolution must be referred to the increase in the complexity of an organism which has life. Let's go to the objectives and then we find out. Formation of new species, that's speciation. Gradual change in the characteristics of species, that is also not gradual change in the characteristics of a species. Over time, gradual change. You see, you are changing the characteristics. So this looks to be that correct. Present day distribution of species in the world, that is distribution of organisms, that is by geography. Structures that have the same basic plan but adapt for different functions, though that is homologous structures. So this is also out. This is also out. So the most correct answer looks to be B. So B becomes the most correct answer and you get your 2 marks. So we go to the next point. Peacock, you know them, use bright colors to attract the mating. Mating, you know the mating. No, we don't need to explain about mating. Everyone knows about mating. Don't forget to stay virgin. All right. The reproductive isolation mechanism is called breeding, hybrid infertility. No, this is not about the colors. They're saying about reproduction at different times of the year. They're saying use bright colors, attract. There is nothing related to time there. So time of the year, no, out. Species specific courtship behavior. The way how they dance is not the same way you dance when you're looking for the mate. It's not the same way. So basically here, this one becomes the species specific courtship behavior. Each species has a specific way in which it attracts the different sex, the different gender. You understand? During the time of mating. So each species, when the frog is doing it's nice for them. It's ugly for you. But for the frogs, it's nice. So that's how they do their behavior. You understand? Yes. So this is species courtship behavior. Cooperation, you know that the insertion of the penis inside the vagina, that's out. It's not about that. So this is the most correct answer, which is species specific courtship behavior. Each courtship behavior is specific, a specific species. That's what it means. However, humans, some of them are ugly. You want to try everything. I didn't say that. So the answer is C, and then becomes the most correct answer. You get 2 marks. Thank you so much, guys. We continue. Life science is nice, guys. By the way. All right. Mendel, the scientist who played a role in the discovery of DNA. They are not talking about discover the DNA. They're asking the person who played a role. Whatever part you did take in the discovery of DNA, that's what they are looking for in this regard. Let's continue and find out. Mendel, and Crick. Yes, Crick, yes, but Mendel out. That is evolution. Bloom, this evolution. Watson is yes, is correct, but Darwin, that is evolution, is out. Franklin, yes, correct, and Wilkins, yes, correct. So D becomes the most correct answer. James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. Those are the 4 individuals who were participating in the discovery of DNA structure based on our molecular. Yes, so that's it. We continue. Difference between genotype and phenotype is that the genotype is a genetic makeup, while phenotype is external. Genetic makeup, genotype, the gene type. Which kind of gene type do you have? Which kind of genetic composition do you have? That's the gene type. Is it a recessive? Is it dominant? Which type of gene you have? So that's what it means. And then you have the phenotype, the physical expression of an organism. How do I look? I'm dark-skinned, man. You see me. So that is physical expression of an organism. You see? So that is a phenotype. So this looks to be correct. Saying that the genotype is an allele inherited from maternal origin, while the phenotype is an allele from non-man. They're saying that the phenotype is external appearance, non-man phenotype, gene type. So it's not about the external appearance. Genotype is the inheritance, while phenotype is not. Man, the characteristics you have, you obtain them from your father and your mother. So since it is like that, then it means that we inherit even the phenotype, because the genotype will determine the kind of the phenotype you have. And this genotype you obtain it from the mother and the father. You understand? So, so this one also out. So the most correct answer becomes A, and A, you get 2 marks in that regard. You see, by the way, life science is so nice and enjoyable. When you try to do this life science, you'll find out that life science is nice. It's nice. By the way, every day you practice life science. How can you fail life science? Let's continue. 1.1.8. The graph below shows the average height of giraffe. Giraffes in different time period used by Lamarck in the evolution theory. All right, it's a story. The average height of giraffes in different time periods. Okay, sharp. Which one of the following is shows the correct percentage increase in the height of giraffe different times of the year or periods? Remember, percentage increase, we say that it is final minus initial over initial and times 100. So I need to find out that if I get the final, is saying that from 20 to 15, yeah, 20 to 15, yeah, 20 to 15, now means okay, from here to here. So if you put this minus this, divide by this, times 100, do you get this out? B, 15 to 10, meaning that final, which is 3.3 minus 2.2 divided by 2.2 times 100, do you get this? If you don't get it, then it's wrong. 10 to 5. 10 is 3.4, 3.3, and 5 it is 4.5 minus 3.3 divided by 3.3 times 100. Do you get this? If no, this is wrong. Then 5 minus 1 or until present, 5 from here until present is 5.5 minus 4.1. That is 1 divided by 4.5. Then times 100. If you get this, then that becomes if it's wrong, then it's wrong. If you get it, then it means that that's the correct answer. So our most correct answer here is D. So D becomes the most correct answer. You understand? Yes. We go to the next question. 1.19. All right. The ultimate purpose of artificial selection is that increase the genetic variation. Artificial selection increase the genetic variation. I don't think so. Allows the organism to survive in natural environment. Artificial? No, man. No, man. Reduce the size population of species by removing the organisms with the undesirable characteristics. Why should we reduce the size of the population? For the organisms. Yet we want more of them. I don't think so. Let me see it. Increase the organism with the specific desirable trait for human benefit. Discard acid. For example, milk. Different characteristics we are looking in organism. Meat, disease resistance, meat. Talk about meat because I like meat. Hey, man. Nyama. All right, we continue. 1.1.10. Which organic catalyst is used to cut the plasmid? Plasmid in genetic engineering. Remember, plasmid is that bacteria which, did I say bacteria? Yeah, is found in the bacteria. This plasmid is the one where the enzymes are used We call them restriction enzymes, basically. We call them restriction enzymes. Restriction enzymes, these are enzymes which cut at a specific area, you understand, of the genetic material. So they are used to cut that plasmid's DNA so that the one which you want to multiply is inserted inside there. So basically, when they say the organic catalyst, definitely we are talking about the enzymes. They are specific, the enzymes, these ones, which we call the restriction enzymes. So they're saying enzymes, okay, looks to be correct. Stem cells, nope. Recombinant DNA technology, nope. Bacteria, nope. The plasmid is found in the bacteria, so it can't be. So the answer is A, which becomes the most correct answer. And that it means that you get 20 out of 20. So you see that, guys, it is so nice chilling, and getting 20 is very easy. Guys, make sure that in these objectives, you get 20 out of 20. Yes, if you see that you have challenge, check out what you think it's not right. There are some answers which you see that this is wrong, can't be. So the remaining one could be the correct answer. So you can use what you call the elimination method. Yes. All right, we got to question terminologies. I like them. I like them. Yes. Pattern of DNA bars, a pattern of bars, black bars, bands on X-ray films where sample is put through special biotech process. So we call these ones we call them DNA profile. These are DNA profile. Yes. Then they are saying that a fluid that has free RNA nucleotides complementary to DNA strand. A fluid that has DNA. When you say that fluid, remember, when you talk about free DNA, RNA nucleotides complementary to DNA strand, meaning that this, they are talking about, this is DNA. So where is DNA found? DNA is found in the nucleus. The what? In the nucleus. So if it is found in the nucleus, how do we call the fluid inside the nucleus? What is this fluid called? Since it's the fluid, the one which is outside here, we call it cytoplasm, the fluid of the cell. The one which is found inside, we call it nucleoplasm, the fluid of the nucleus. So the most correct answer here is going to become, we're going to say it is a nucleoplasm. The type of dominance that produce an intermediate phenotype. Whenever we talk about intermediate phenotypes, we're talking about incomplete dominancy. For example, none of the two is completely dominant over the other. It means that each contributes 50%. For example, I always give an example of presidents. The president of South Africa and the president of which country can I say? Zimbabwe, Malawi, Botswana, Eswatini. So all of those, if they are there, yes, they have the same power in their own countries. So when they sit on the table, they address them as presidents, meaning that if they are discussing about something, each vote will be equivalent to the same power. If this vote is 1, even the other one is going to be 1, meaning that they have equal power. None of them is completely dominant over the other if they are sitting on the same table. None of them, because all of them are presidents. So if you have red crossed with the white, you produce a pink. Red is not completely dominant over white, and white is not completely dominant over red. And then now you produce what we call an intermediate color. Yes, or an intermediate characteristic. So this is called incomplete dominancy. Then we say that the type of barrier in speciation that split the single population into subpopulations. We call it the geographical barrier. Yes, we call it geographical barrier. Yes, remember we said that there was one population of. Remember, you have to apply when it comes to explanation of the speciation. There was one population of rats. You understand? The population is divided into two by a rock. So a rock becomes a geographical barrier into two subpopulations. So it divides this population of rats into two subpopulations. So how do you call structure which split this population into two subpopulations? We call it what? We call it geographical barrier. Correct. A reproductive isolation mechanism that occurs in flowers only. Flowers only. Obviously, flowers, they do carry out what you call pollination. So it's can call it, you are adapted to different pollinating agents. Yes, that is the word I wanted to modify. Pollinating, you are having a different pollinating agent. For example, the insects, you are specific to a specific insect with a specific shape of the beak. So we call it pollinating at different seasons of the year. So that is it. Then we go to the next question. They are saying that the genetic disorder resulting from a mutation caused by absence of blood clotting factor. A genetic disorder resulting from absence of a genetic disorder resulting from a mutation caused by absence of blood clotting factor. If you don't have blood clotting factor, it means that when you have a hole, maybe you have got something on your skin, the blood will ooze. So when the blood oozes, then it means that we call it hemophilia. Hemophilia. Yes. This is very common in ladies. Did I say in ladies? Many, they are more affected because they don't have remember, hemophilia is caused by a recessive allele. So male are more affected by hemophilia, yes, than female. So we mostly find it in the males than in the females. Why? Because males, they only have one X chromosome. So you can, we will talk about it. If you find a question, they can ask you that, why are males affected by this sex-linked disorder than females? You understand? As a result, you can say that males have two X chromosomes if a recessive allele appears on one X. Did I say males have two X chromosomes? Yeah. Females have two X chromosome. If a recessive allele appears on one X, it can be masked by a dominant allele on the other X. You understand? And then the person is still normal, but is a carrier. But if the males, once the recessive allele appear on one X, is carried on one X, automatically the person is what? Is the hemophilic. Then they're saying that the source of variation where individuals in the population has equal chance of mating with other. The source of variation where any individual in a population has equal chances of mating with other individuals. So how do you call that source of variation? So we say that we call it random mating. We call it random mating. Then evolutionary theory proposed by, remember that you must always say A only, B only, or A and B. Rapid periods of change, meaning that they are alternating with one another. So it is the punctuated equilibrium. So you guys, yeah, we got the next question, which is 1.1.3. They are saying that a component of nucleotide found in DNA, component of nucleotide found in DNA and RNA. They're saying DNA and ribose sugar. No, this one is found. This one is found only in RNA, but they said DNA and RNA, so the answer becomes A only. A only. Yes, they're saying that segment of DNA that code for a particular trait. This one is the segment of DNA that control a particular trait. So we call it a gene. It's A only. Advantage of bipedalism: wider view of the surrounding, climbing trees. A only. Okay. Remember that you must always say A only, B only, or A and B, and then both A and B or none. So please make sure that you have this word only, 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 only. Yes. So we go to the second question, which is 1.4. The diagram below. So once they bring such diagram, they can ask you describe crossing over, draw the structure of a chromosome after and before. They can ask you a lot of questions here. They can talk about the karyotype. They can talk about Darwin's. They can talk about trisomy, trisomes. They can talk about the trisomes. That is Down syndrome. Yeah. So what are they asking? So you guys, I always tell you that once you find this question, try to rotate it so that it looks like this, so that this and this, they're on the sides. Okay? So once in the side, you can see that this is the middle. So if you look at them, then it means that this is metaphase. And then once they are like this, because these are the spindle fibers, they are radiating like that. They are supposed to be in sides. So once they are like that, then it is easy for you to make it simple. So since it is one chromosome per, if there was another one here, then it would have been meiosis 1. But since it is only one chromosome, then it means that is meiosis 2. So now we know that that is yeah, so now we know that that is which diagram 1 or 2 represents meiosis. Diagram 1 2 represents meiosis and then represents mitosis. So this one looks meiosis, diagram 1, and then diagram 2. Why did they bring this question? Anyway, I will say that it's diagram 2. Do you know why? Because here there are no variations. Here you see that the chromosomes, they are varying due to the crossing over. But here I don't see any variation. So it could be mitosis. So I'll conclude it like that. Name the process that occur in the cell division, diagram 1, that resulted in the variation. You see, it is crossing over. So I will say that crossing over. Crossing over. In which phase does the process mentioned above occur? It occurs in the prophase 1. It occurs in prophase 1. Name the type of cell in which the cell division occurs. Name the type of cells in which the cell division, diagram 2. Oh, diagram 2. You remember I said this is mito. It looks to be like mitosis. And mitosis occurs, where does it occur? In the mitosis occurs in the somatic. Somatic. Use this color. Somatic cells. It occurs in the somatic cells. Then they're saying that state the diploid number of chromosomes in the diagram. Diagram 1, diploid number of chromosomes. Diploid number of chromosomes in diagram 1. Did they say it's a human cell? They didn't say anything about human cell. So it's going to be diploid number. It's going to be 4. And also this one is going to be 4. So, state the number for the cell. No, in diagram 2, remember, if this is mitosis, let's analyze it like this. If this is mitosis doesn't change the number of chromosomes. So if it is 2, they're gonna be 2 in meiosis in from the beginning until the end. So they're gonna be 2. So if they're saying diploid number, and here I'm seeing 2, so it means that this is not 4. This is gonna be 2. While this one, since this one it looks to be metaphase 2, then it means that in metaphase 1 there were 4. So there are 4 here. Do it like this. There are 4 here, while here there are 2. Why? Because in mitosis, the number of chromosomes don't change, while in meiosis, the number of chromosomes change. And if you look at it here, this is meiosis 2. That means that in meiosis 1, the number of chromosomes divided. So since these are 2, so in meiosis 1, then it means that there were 4. And give the number of chromatids in diagram 1. What I see. So it's 1, 2, 3, 4. So remember, each chromosome is made up of 2 chromatids. It's 1. Check. It's this. Then it's that. So it's this 1. So if I have 2 chromosomes, so it's going to be 1, 2, 3, 4. So the answer here becomes 4. In this bird species, dark feather color, capital D, is dominant over light feather color. Okay. And then long tail is masks the expression of the short tail. Okay. They are saying that the square below shows the possible results when the gametes of the parents were crossed. Okay. So if they are crossed, they are saying that name the type of cross shown in the diagram above. So this is a dihybrid because we are considering 2 characteristics. Yes. All right. Here you are seeing 4 of them, but those are not. If you are saying that size, you have big or small. So that is one character, which is size. Your body size. So that is one character. So you have to understand that concept so that you don't get it wrong. So they're saying that they name the type of cross represented in the Punnett square above. So we say that is dihybrid cross. They're saying that write the phenotype of the offspring. Right, the phenotype of the offspring. Just a moment. Write the phenotype of the. Just a moment. Okay, the phenotype of offspring X. It is easy. I go with this and this because that's where the cross is gonna be. Capital D, capital L, small l. Yes. Then they are saying that the genotype, right? State the genotype of two parent 2. Oh. This is parent 2. So how do we get the genotype? So what I do, did I say genotype? They're saying genotype. So let me first identify the phenotype so that I'm able to identify the genotype. So the phenotype, I'm gonna say capital D, the first letter. Do I have any small d? Yes, I have a small d. You see? So small d. And then capital small l, and then even this is small l, so it means that it's capital D, small l, small l. Yes. So this is the genotype. Capital D means what? We come back and then find out. Capital D is dark feather. And then small l is small l, small l, small l, small l, small l. Long tail masks the expression of the short tail. So this is small l. So it means that it's going to be dark feather with short tail. Yes, so that is going to be the most correct answer. Yes. Then they are saying that the dominant, the type of dominancy, the type of dominance in tail length. With tail length. One is completely dominant over the other. So it means that is complete dominancy. So there I'll go with the complete dominance. Remember, if complete dominance, if one allele is completely dominant over the other, for example, light-skinned crossed with the dark-skinned, then you produce a kid of dark-skinned, then it means that dark-skinned is much more dominant over the light-skinned. Tall crossed with a short, and then you produce a short, then it means that tall is much more dominant over the short. Then you can look at any character and then you see what you get as a result could be that the other one, which is not expressed, was masked by the dominant allele. Yes, so which parent, one or two, has the heterozygous genotype for these characteristics? Which parent, one or two, has the heterozygous genotype for both characteristics? They're saying heterozygous. I see it is the parent one. You know why? Let's find out. Let's see here. Because here I have capital D, I write the capital D. Do I have capital D again? Yes. So I cancel it out. Do I have a small d? Yes. I have small d, so I write small d. So I don't write the D again. So I have capital L, capital L. Do you have another capital L? Yes. But do I have a small l? Yes. So I would go with a small l. So that is heterozygous for both characteristics. But if you look at parent one, you have capital D, capital small d, yes, small d, small l. Do I have a capital L? No. So I have to go with the second L. So you see that this is homozygous, recessive, or this is heterozygous. Well, this one is heterozygous, and also this one is heterozygous, because this is a capital L and then this is a small l due to what? Then they are saying that name two Mendelian principle that describe the separation of allele during the gametes. So here we can talk about Mendel's principle of segregation, and then you can also talk about Mendel's principle of independent assortment. We talk about the segregation, and then you also talk about the Mendel's principle of independent assortment. And then that makes us to get 50 out of 50. Who got 50 out of 50, or who is willing to get 50 out of 50? And if you got them, thank you so much. You are a star. So question number 2 is saying that, okay, question number 2 is saying that the diagram below shows different processes involving DNA molecule. So you have process A, you have process A. So it's question 2. You have process A and then process B. They're saying identify process A. If you look at here, we see that process A, you're forming DNA. So this is DNA replication. While DNA replication. While process B, we see this is the protein synthesis. Yeah, basically we see that this is transcription. So which part of protein synthesis is transcription? So process B is transcription, while process A is DNA replication. We can ask you a question: what's the difference between transcription and translation? What are the similarities between transcription and DNA replication? They can ask you the similarities between DNA replication and transcription. So you look at the steps that DNA replication occurs in the nucleus. Also transcription occurs in the nucleus. DNA replication, double helix DNA unwinds. It's the same story here. It also double helix DNA unwinds. If you look at the differences, you can talk about two strands they form the act as a template, while the other side you can talk about one strand act as a template in transcription. You can talk about two new DNA molecules are formed. This side you can talk about messenger RNA is being formed. So you look at the differences and the similarities. They can ask you those questions. They can talk about the differences between nucleotide of DNA and nucleotide of RNA. So they can ask you a lot of questions concerning about that. Name the organelle where process A and B occurs. Process A, so that is the nucleus. And then they're saying that basically we only have two organelles we usually talk about. We usually talk about the nucleus, then also talk about the ribosome. They state the phase in the cell cycle in which the event takes place, state the phase in the cell cycle in which event A takes place. We say that it occurs during interphase. So done with that, describe the event that occurred during protein synthesis after R molecule attaches to the ribosome. So if you are talking about R is a messenger RNA, so they're asking you to describe the process of translation. So you describe it. You must have the distinction material book. You'll be able to answer all these questions simply. Maybe you can say that according to the RNA anticodon, the transfer RNA with the complement. According to the RNA codon, the transfer RNA with the complementary anticodons will bring the required amino acid to the ribosome. The amino acids are joined together by the peptide bond to form a required protein. So basically, that you can explain it, or specific amino acid is going to be brought to the ribosome when the anticodon of the transfer RNA matches with the codon of the messenger. You understand? The anticodon, the amino acids are joined together by the peptide bond. The bond is peptide. Don't make a mistake and say polypeptide bond. 0 will give you a 0, and that 0 will have ears and eyes. Please don't do that. Yes. So the bond between the amino acid is called the peptide bond. Yes. Then they're saying that to form a required protein. So you explain as long as you talk about all the marking points which we are looking for. Describe the event that occurs during. Okay, we are done with that. Explain one functional relationship between DNA and molecule R. What is the functional relationship between DNA and the molecule R? Basically, molecule R contains the coded message from DNA so that the proteins can be manufactured. That's what they are looking for. It has coded information for DNA so that that thing can be processed, can be formed. Yes. The proteins. Then they are saying that explain the function. The table below shows the amino acid between DNA and the molecule R, which is a messenger RNA. They are saying that use the table below to determine the codons. When you talk about the codons, remember we have the codes which we find on DNA. We have the codons which we find on the messenger RNA. We have the anticodons which we find on the tRNA. Yes, so I've explained this before. If you check on our channel on Thundereduc, then you'll be able to get all these questions and explanations about it. Yes, make sure that you get a distinction material book so that you're able to obtain these distinctions. Yeah, nobody, if you are using the distinction material book, you only don't get a distinction because you don't want. If you want it, is the distinction material book. The distinction is in your hands. All right. Then they're saying that write the triplet bases. Okay, triplet 1, 2, 3. So if you look at 1, 2, 3. So they are saying write the codons. So if this is DNA, then it means that I'm going to have to change it into a messenger RNA. So T goes as TGC, and then you have TAC, and then you have AGG. So it's gonna be T is gonna go with A, and then C, then G, and then it's gonna be A, it's gonna be U, it's gonna be G, then it's gonna be U, it's gonna be U, it's gonna be C, it's gonna be C. So basically this is what we are looking for, and that is what you are supposed to write. Yes. So you get your 2 marks there. How do they mark? I don't know, but it's supposed to be like that. Determine the amino acid for DNA based triplet that is complementary to this. Guys, we always ask you these questions, and the mutation. Did they ask you? They didn't ask you about the mutation, but we always ask you about this. Determine the amino acids for DNA triplet that is complementary to TGC on strand 2. Determine the amino acid for DNA-based triplet that is coded to TGC. Strand 2 is DNA, and what is here is also DNA. So since they are giving you DNA and they are looking for DNA, then definitely we are not changing anything. So it's going to be TCG, Let me clear it up. TGC. So it's gonna be cysteine. So that's what is gonna be the answer. Let me check properly. Yes. Cysteine. Determine the amino acid for. Let me first clear it up so that I don't want to make a small mistake and then. Yes, they're saying that determine the amino acid for DNA the DNA base, the amino acid for DNA base triplet. Okay, that code that is. Oh, you see, that is complementary. You see, because this thing was covered. So you see. Let me just do like this. Yeah, guys, make sure that you're always keen. They're saying complementary. So this is complementary. This is C. So what is the complement of this? T goes with what? Goes with A. G goes with what? G goes with C. C goes with what? With G. You understand? With the C, né? This is C, goes with the G. So it means that this is still DNA. It's still DNA. It's not messenger, because what is given here is DNA. So it means that what I'm going to say that this is just they are looking for the complementary. You understand? So since this they are looking for this, so I'm gonna look for this. So it's gonna be ACG, which is threonine. So that is the correct answer. So you guys, you see these questions, né? You see, even me, I was gone because I couldn't see this because of the words which are there. So make sure that you take note of these questions. Né? Don't just give the answers and then you just go like that. Please make sure that you read these questions, because a small mistake like this makes you to get out of the what? Don't finish the paper and then you sleep. Doesn't make sense. It is like you are making the examiner to be a fool. Yeah, how can he set a paper for 2.5 hours when you finish it in 1 hour? You are making the examiner a fool. Yeah, when you don't know what you're setting. So guys, don't make us fools. Yeah, don't make examiners fools. Okay, we are saying that name the type of mutation that causes Jacobo's syndrome. What is Jacobo's syndrome? Karyotype below. Remember that karyotype is the type of the nucleus. Karyo means nucleus, and type means the type. Which type of nucleus we have? Of the male suffering from Jacobo's syndrome. What's Jacobo's syndrome? We'll find out. Where is it? Name the mutation that occurred that caused Jacobo's syndrome in this image. I see here on chromosome number 23. Is this a male or a female? It's a male. Why? Because I have X and 2 Y. But anyway, I'm not going to talk about that too much. I'm just answering a question which they said, and then so in most cases we don't bring these questions which we bring the one which has the trisomy is on chromosome number 21. The one which were like this, they had been removed from the examination guidelines. So if they are brought them, let's answer them the way they are. Yes, but it's not supposed to be. Let me just say it like that. It's not supposed to be in this format. Anyway, following the examination guideline, that is my opinion. Yes. However, they also had their opinions. Why did they say it? Maybe they want to say it about something. All right, we continue. However, you can use the same principles used here to answer these questions. The good thing, they're not asking you what is it. They're just application question which you can apply and then explain. So use the concept of Down syndrome, and then you are able to obtain or transfer that knowledge to this question. Maybe that's the instance for them setting such question. They're saying that for this individual, determine the number of autosomes. Remember, autosomes are the chromosomes which are not they are not sex. So I'm going to have to count out is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Guys, always count them because sometimes there is one where there is 3 of them. So autosomes, there are 22 pairs. Or I can say they are 44. Yes. Then gonosomes, we see them, there are three chromosomes. Yes, I can't say 3 pairs because they are just 3. 1, 2, 3. So these are 44 chromosomes. Not pairs. Yeah, 22 pairs of chromosomes or 44 chromosomes. Use letter X and Y. Write the gonosome of this individual with Jacob's syndrome. So we see that this is X, the one which is long is X. Let me clear it up so that you're able to view. This is X, this is X, this is X, and then this is Y. So basically that is it. Yes, they're saying describe the way in which the karyotype of the person with the Down syndrome will differ from the one mentioned above. Remember, Down syndrome, the trisomy is basically happening on chromosome number 21. Like the 3 chromosomes will appear on chromosome number 21, while this one it appeared on chromosome number 23. You understand? Yes. So basically the trisomy 20 Down syndrome, it occurs in the autosomes, the autosomes which are non-sex chromosomes, while this one occurs in the sexy chromosomes, which we call the gonosomes. So I think that's it. Explain how the error during the gamete formation in meiosis 2 could result in the Jacob syndrome. Then it means that if you look at this, what happened is this could be for a female or it could be for a male. But what happens? It means that during anaphase, yes, the chromosomes for the chromosome pair, like 23, won't pair. The 23rd chromosome is supposed to, for example, this one is supposed to split like this. But now, it is in metaphase. So during anaphase, they are supposed to split so that they look to be like this. So they are moving in opposite poles. Yeah. But now what happens on this gonosome? What will happen? This chromosome, which is the gonosome, for example, instead of this one, instead of them, instead of splitting like this, both of them are going to go to one side, meaning that leading to this other side having nothing. So we're gonna result in this cell having 2 of the gonosome. You understand? And then this side there is nothing because nothing went there. It's only have these autosomes which are there. So it means that if this was a male, it was a male, yes, it's going to have X. You understand? 2 of them, X. So if it was a male, it's going to have Y. And then this one is going to have an X. 0. Nothing is there. So there is no gender there. So if this cell fertilizes a female ovum, the ovum, yes. So what is going to happen? It means that the ovum is going to bring X, so that now this individual is going to have 1 X and 2 Y chromosome. So that is what is supposed to be. So basically here, non-disjunction occurs as the failure of the chromatids fails to separate at position 23. So that's what we talked about. And then he said that during in anaphase 2, because this is, they are saying in meiosis 2, and non-disjunction occurs in anaphase 2. During anaphase 2, the sperm cell will have an extra chromosome, you see, due to the failure of them to separate. When it fertilizes a normal ovum, yes, the individual is going to have one X and two Y chromosome. So, which is what we call the Jacob's syndrome. But I talked about it that in most cases, we don't usually set this. I think they were just setting it to link it to Down syndrome. We like it to bring it to Down syndrome. And then if you see how I was explaining it related to Down syndrome. But instead of writing 21, chromosome number 21, I used the chromosome number 23. Yes, so that is it. And then we are saying that 2.3, they're saying that state the phenotype of anger. Okay, here we are having this and this. So to know the phenotype, you need to know what is the kids. How many generations? There are two. 1, 2, 3. So there are 3 generations. How many kids are for James and Anga? Is 1, is 2, is 3. Okay. Someone is going to ask, how is it 3? Because this shows that it gave rise to that and that and also that. So this they just got married to those kids. So they're saying that the genotype of this, so if this is blood group A, they produce blood group AB. Definitely this is blood group B. You understand? So that this person, since this blood group B, blood group, yes, they produce this. They produce this. Why not blood group AB? AB. Because if there was blood group AB, then it means that the genotype is going to be like this. There is no way how they're going to produce candle. So it means that this one is blood group A, heterozygous, and this is blood group B, heterozygous. So that this and this, they combine and they produce candle. Yes, so it means that Anga was blood group B. Then the genotype of individual that plays codominance, which is codominance, that is blood group this AB. So the genotype, so definitely it's going to be A, B, because they're looking for the genotype. They're saying that give the number of individuals with homozygous recessive allele, that is blood group O. That is blood, but they're saying give the number. How many are they? So it's 1, it's 2. So it's going to be, in this answer, this question is going to be 2 of them. Explain why the future children of JD and Lisa could display different blood groups compared to their current children. Lisa, JD and Lisa. I have to look for JD and Lisa. So since JD and Lisa, this is A, this is B, this is A. And then since these ones were like this, then automatically JD was a small letter. And then to produce a different blood group, yes, to produce a different blood group, then automatically even A must be heterozygous. Oh, this is B, ne? JD is B. And then so that their children could be, when this smaller and smaller, when they combine, they're able to produce blood group 4, meaning that the JD will pass a recessive allele to the offspring, the same way Lisa is going to pass the recessive allele to offspring to produce blood group O. But I need to find out what is the genotype of Lisa, which is blood group B, heterozygous, and also JD is blood group B, heterozygous, and also Lisa is blood group A, heterozygous. So you see that to obtain any answer related to this, make sure that you look at the babies they have done what they have produced. Then you are able to obtain those blood groups. Yes. So we go to the next question. Use the genetic cross to show the percentage possibility of beta and Oneka having children with blood group O. So I have to go back and look for beta. Where is beta? And then Onek, what? Bela and Onek. The same. Genetic cross. So you have, since they are on the second generation, I will say that P2, F2. And then they are saying that the phenotype, phenotype. I told you that this 1 mark is obtained free, mahala. So genotype, I come the question. They're saying that Baila, blood group what? Blood group of blood group what? Baila is blood group AB of blood group AB crossed with Onika of blood group what? Of blood group of blood group O. Blood group O. So when you look at the genotype, blood group AB looks like this, while crossed with Onika, which is blood group O, small I, small I. Then they form meiosis. You have to write all these points, and then you say that A is gonna be B, is gonna be small I, small I. So these are gametes. So we call them gametes. Gametes. I say that don't leave them naked because they're not at the beach. So they are supposed to be dressed up. So after that, you can use the Punnett square, or you can just do the crosses. So let me just use the crosses so that I'm able to be a little bit faster. That with that is going to be A, small I, and then now this with that is going to be A, small I, then this B, this with I, that's going to be B, I, then that with that is going to be B, I, small I. So these are offsprings. So we see that these two blood group A, and then two blood group B. So we don't have any chance for someone to produce blood group which is O. So it means that 0 out of 4 times 100, so it gives me 0%. So the probability to produce this kid who is like this is gonna be 0%. Then we go to the next question. This is genetic engineering. We can ask you genetic engineering questions. We can also ask you about stem cells. Yes. So the type of cell produced by meiosis that is extracted from the donor cell. So we look at this. This is a somatic cell. This is a somatic cell, and this is the ovum. That's a gamete. That's the gamete. So somatic cells are always diploid, while this one are always haploid. So that now, because we're looking for the animal which looks like this, so I'll keep the nucleus here. Check, this looks like this. So it means that we use the genetics of this, which is the somatic cell. So it means that I'll keep the nucleus here. I throw away the shell. Here, I throw away the shell. The nucleus, I keep the shell. That's why you see the nucleus here looks like the nucleus which came from here. You see? And then if you look at the shell, looks like that of thick and thick. So now when you have this, we use what called a mild electric shock to combine the nucleus from here and then the shell from here. So we combine them together using the mild electric shock. Then now these embryos are being developed, meaning that they have the genetics of this. But because this embryo, this cell looks like a zygote, because remember zygote, it has a sperm, and the sperm only the nucleus fuses, and then the ovum, so this nucleus of the sperm goes into the nucleus of the ovum. So outside is the ovum and the inside is diploid. So since this is a diploid, yes, and the nucleus outside here, this is the membrane we used, looks like this. So this cell looks like a zygote. So it's not a gamete. Why? Because this is 2N, it's a diploid. So now it undergoes mitosis to form a ball of cells which develops into embryos. So these are being implanted into surrogate mothers, and then each surrogate mother develop, remember these are just stem cells, the function of stem cells, then they develop to form mature babies, and these babies are being released. So that is it. So name the type, the type of cell produced by meiosis that is extracted from the donor cell. The type of cell produced by meiosis that is extracted from the donor cell. So we're going to call it a gamete. Yes, basically it is ovum because it is a female. They're saying the organ where implantation takes place, it takes place in the uterus. Uterus. They're saying the cattle counted to be 63% of 395 different cloned animals in the particular country. Calculate the total number of cattle that were cloned this country. So this one is simple: 63,3 divided by 100 times 3 0.95. So the answer you get, that is the answer. I don't have a calculator here, so you can calculate and then you find what is the answer there. Then they are saying that explain how the process of cloning will affect the variation of the color. Obviously, cloning result in the identical organism. So it means that the variation is going to be reduced. So it's going to be reduced variation. Suggest the different characteristics desired by human that influences the cloning of the 3 characteristics desired, like meat. You can talk about meat. You can talk about resistance of diseases. You can talk about meat, resistance of diseases. You can talk about milk. So all those can be used to clone, be reasons to clone these organisms. So this question is also 50. So if you got 50 out of 50, congratulations, you are a star. Let's go to question number 3. Go to question number 3. They are saying color blindness is a sex-linked genetic disorder that is characterized by the genetic defect of on X chromosome. The moment you say X chromosome, then it means that you want to use X and Y. They're saying that the people with color blindness cannot differentiate between certain colors. The table shows, okay, according to the table, which region has the highest or has the lowest percentage of male with the color blindness. So if you look at it here, we shall see that this is the answer. So it becomes Sub-Sahara Africa. This is just a question which is an application. It can be changed. It's easy to answer. Yes, suggest one reason why the results in the region identified in the question might not be correct. Why should we? Why should do you think? Maybe there are no proper facilities which are used to test. Yes, maybe the facilities are not there. Maybe there is lack of reporting of some cases might not be reported, and then so the information could be wrong. So things which can make the results not to be right, or not everything which has been collected could not be right, or some information might be missing. Yes. So the prevalence color of color blindness in the female is 0.5 globally. Explain why female have a lower chance of being colorblind. So this one is talking about, this is what I explained, that females they have less to be affected by hemophilia. This is a sex-linked character. You understand? Yes. Why? Basically, females have two X chromosomes, so they will require 2 recessive allele for them to be color blind. So if one recessive allele is appear on one X, it can be masked by a dominant allele on the other X, and the female is still normal. However much is the carrier, but it's still what? Normal. So females will require 2 recessive allele on the X chromosome for them to be color blind. You understand? Therefore, it means that even if one recessive allele appear on X, then it can be masked by a dominant allele on the other X, and still the individual is still normal. Then you draw a bar graph. You guys, this is question you must be knowing. Make sure that the bars are the same size. Make sure what you have here, this is X, this is Y, so this one goes to X, so it comes here, and then these numbers, they go here on Y. You can't have words here, never on Y axis. No, they must always be on the X axis. Then you put the caption. Make sure that the caption, you put them properly. Make sure that what you put here, you put units if the units are there. In this case, the unit is percentage, so the percentage must be there. Countries, region, is basically, these are countries, so you can put in brackets a region. So it can be something like a unit, and then it must be indicated there. And then make sure that these bars are the same, the spaces between the bars are the same. Then you'll be able to obtain all the marks, the 6 marks they have given you. Then we go to question 3.2. 3.2 is a little bit easier. They're saying the diagram below shows the scar of hominid ancestor. Yes, the hominid ancestor, and they used the tools as their evolution. So they are looking at the evolution, the tools they used as their evolution. Okay, they're saying state the line of evidence for human in evolution in the diagram. State 2 lines of evidence for human evolution in the diagram. So in this case, we see that we have the tools, and then we have the fossils. So remember, when you're explaining, you don't say tools. Remember, the evidence is cultural evidence. Example of cultural evidence is use of tools. So the answer here is cultural evidence, and then here number 2 is fossil evidence. Then they're saying that name 2 Australopithecus africanus species found in South Africa. We talked about them: Taumchad, Littlefoot, and Mrs. Place. So this one are easy to identify. Identify the type of tool used by species with the largest cranial capacity. So if you look at the tools they use with the largest cranial capacity, so we see these are the tools they used. Then they're saying that how many genera are shown in the diagram. So if you look at the genera, we look at the first, like the surname. So we see the Homo is 1, is still Homo, Homo 2, and then this is 3. So there are 3 genera which they shown in that diagram. They're saying that based on this diagram, the Handyman species was in existence 1.8 million years ago. Write the scientific name for Handyman. That is Homo habilis. So Handyman, remember, is the first man to use tools, so we call them Homo habilis. Provide a reason why species named above is regarded as handyman. It means that it's the one which first used the tools. So I've given the answer. Explain why the evolution of large cranium in human is associated with the development of tools. Why is this that the more the brain is developing is linked to the advancement of tools. Advancement of tools, these tools, they require thought. So it requires the brain which is thinking, the brain which is much more how do I call it, much more developed to think and develop these tools. So we can say the increase in the brain will lead to the increased intelligence. Actually, when you have a large brain, you're going to have more brain cells, which leads to the increased intelligence and then leading to the development of the complexity tools. So increased brain means that you have much more brain, yes, which increases the intelligence, leading to the increased intelligence, and then this means that you're going to have to develop more complex tools. 3.3, what are they asking about? 3.3. Here is an extract you're supposed to read. And then we are saying that lactose intolerance refers to the ability to digest lactose sugar into glucose, and lactose tolerance individuals do not experience comfort after drinking milk, eating dairy products. Lactose. However, some individuals struggle to digest lactose due to the production of lactase enzymes, and then classified lactose intolerance. Such cannot be. So scientists investigated the influence of lactose intolerance gene mutation on the lactose tolerance in African population. Okay, this is very important. They're saying that they collected DNA of this. They're saying they were found in Tanzania, Kenya, and Sudan. And then they're saying that divided the participant into 2 groups. They're saying that I'm seeing the groups gave the participant lactose sugar solution to consume daily. They ensure that participants ate the same diary during investigation, measure the blood glucose before and after. What is the independent variable? So what determines this concept? So basically, the independent variable is the lactose intolerance gene. This, because I say investigate the influence of this on this. So it means that this is the independent, while this is the dependent. So lactose tolerance is the dependent. So this is done. Name the type of variation existed in the ability to digest lactose. So it is discontinuous. Either you digest or you can't digest. You can't try. No, you either do it or not. So it is two, you get your mark there. It is discontinuous variation. Then they're saying that state way in which the scientists ensure the reliability of the investigation. Remember reliability by increasing the sample size. So they said they ensured, meaning that they used 470 people. Yes. the individuals were from different ethnic groups. You understand? We see them there. We see that all different area of the what? Sorry, guys. Alhamdulillah. So we can talk about all of that. State the factor that should be kept constant when the lactose solution was consumed. Which factors? The amount of lactose which you consume must be kept constant. The concentration of lactose must be also kept constant. Why are scientists measure the blood glucose level before and after? Basically, they want to compare these results by developing what called a baseline. Yes. Okay, before it was like this, after it was like this, what caused what? That change is caused by what? So they want to find out the effect. So by developing what called a baseline. Then they're saying that describe how scientists could establish whether the gene mutation has occurred on this, whether a genetic mutation had occurred on lactose this gene. So basically the sequence of gene of DNA or of the participant on the chromosome, yes, should be determined. And then they have to determine it using the DNA sequence, you understand? And then after that, they have to identify the mutation of this gene, and then they compare it to the one which is lactose intolerance. If they see that there is a difference, then automatically it means that there was a mutation. So they have to first identify the one which is tolerant and the one which is intolerant. And then after that, they must compare. And then they see that, oh, the one which is intolerant, maybe the mutation occurred. If there is a difference, then automatically it shows that there was a change in the sequence of the gene. So which means that it was a mutation. Then we go to the next question. Large scale overfishing. Okay, so remember much of the rate of display humans in target fishing and port next. So this 30% reduction of body size and faster maturation. Okay. According to the paragraph, which human activity has resulted in the... So when we bring such question, guys, make sure that you read the passage and then you read it at least 3 times so that you're able to obtain the information which is there. According to the passage, which human activity has resulted in the population of Atlantic codfish? Basically, if we see that there is large scale overfishing, then they are saying that explain the effect of GHR gene mutation on the survival of the chance of Atlantic cod fish. So basically, what is explain the effect? What is the effect of this? What is the effect of this? Yes, mutation on the survival. So we see that this mutation was useful. How was it useful? Yes, because it's basically reduced the size. So if the size is reduced, it means that the fish could be a little bit faster and then increase the chances of their survival, meaning that they are able to move very fast. They are swiftly moving, and then they are hiding. They're able to hide from the predators or from the hunters so that their population can increase. Used down in theory of natural selection, explain the evolution of small body fish, small body size, and the faster maturation rate in the Atlantic codfish. So what do we explain here? Remember, this is natural selection, so you can have to tell us that there is variation among these codfish. You have to tell us that. Then you tell us the variation. Which variation is that? Some had experienced mutation of on GHR genes that resulted in the small body and then making them a little bit faster, and then than others. And the others did not have that, meaning that the one which had the mutation, yes, they were able to be faster, you understand? And then others, they didn't have that ability. So that is the variation which we are talking about. So basically, when overfishing occurred, yes, so that is now the pressure which is being exerted to these fish. The codfish without the mutation, what happened to them? Meaning that they could move less, so they were being hunted, they died in large numbers. All those ones with the mutation, they could have moved faster, so they survived in large numbers. You understand? So they reproduced also in large numbers. They passed the allele for this mutation to the next generation, and then after many generations, it means that the next generation will have a higher proportion of this codfish with this mutation, and then with a less proportion of those ones without that mutation. So basically that's what you're supposed to explain. Please don't confuse this natural selection with speciation, because in most cases students confuse this and then they end up losing the mix. They are saying discovery, there's so many passages. Discovery of fossil in remains of 87 million years of okay, scientists Europe understand the okay this call found in this transition has a transitional future similar to that of Africa. Okay, name the hypothesis of human origin that is being challenged in this passage. So we call it the out of Africa hypothesis. We don't have any other hypothesis. State the hypothesis mentioned above. Modern humans, the word modern must be there. If you don't put it, remember we have different humans, homos. All homos are humans, but the modern is the Homo sapiens. So you have to be specific and say that modern humans originated from Africa and migrated the rest of the world. Yes. Then they're saying that describe 3 characteristics of the skull that were similar to that of these African apes. So foramen magnum at a more backward position. Yes, it is in a more backward position. And then you can talk about large brow ridges. You can talk about they were prognanthus, meaning that they were prognanthus, meaning that more protruding jaws. Yes, and then you can talk about the rectangular palates. So the way how these apes were. Yes, they're saying that what is the name of the birthplace where most fossils of mankind were found? So this is cradle of humanity. So basically that is the paper, and since the paper was not that difficult, I see that you guys you will make it, and then we see that um so guys, yes, we wish you the best in your exams, and we pray that you guys you pass you very well. I've not talked about the phylogeny. The phylogeny was there. Make sure that you do all those questions which are found in the phylogenetic tree at the back of the distinction material book, so that you'll manage the paper. Don't stress about it. You'll manage it. You'll pass it. It has been Saidi. I love you so much, guys. Thank you for loving Thundereduc. And just know that we are here to see that you guys, you become responsible citizens who are not just citizens, but those citizens who are rich. Welcome back to our class. Today we're going to be talking about the paper 2. This paper 2 was being written. At least you don't get lost. The first question is saying that the position of a gene on a chromosome. How do we call the position of a gene on a chromosome? It is called it's called a locus. It's called a what? A locus. That's the position of a gene on a chromosome. Then we go to the next question. They're saying that the diagram below represent the process occurring in the cell. The process, yes. So how do you call this process? If you look at this process, you see that you have a DNA structure, and both strands are acting as a template for formation of the new strand. Therefore, this is DNA replication. I hope you got that answer. So these are 2 marks. Those are 2 marks. Then we go to the next question. They are saying that Jason and Robert are identical twins, meaning that they are coming from the same egg, same ovum. If they're identical twin, which one of the following uses of DNA profile is not possible when considering these identical twins? Number one, identification of Jason and Robert's father. It looks to be correct. Then they are saying that determining if the woman is Jackson or Robert's aunt. Which one looks to be correct? Also, they're saying it's not. So we are looking for the one which is not used. It could be it could be the function, but it's not used in terms of the identical twins in this case. Distinguishing between Jason and Robert when determining who committed the crime, because they have some differences in the DNA, but most of their DNA is almost the same because they're identical twins. Therefore, this might not work that much, because here you are looking at the differences among them. So this one not really. And then determining Jason or Robert could be, which one could be, or can they be a kidney donor? Yes, this is also correct. So the answer, which looks to be not that much, is this. So gets the 2 marks. Then you go to the next question, is about the graph or the table. They are saying that part of the gene produces a protein that has this triplet TTC, meaning that if you see TTC, it means that it's coming from DNA. Yes. And then you have is being replaced. They're saying that a mutation occurred and then the base triplet became this, meaning C is replaced by T. So it's also still DNA. And then they're saying that the table below gives messenger RNA codon with their corresponding amino acids. So in this case, they're saying the anticodon, they're saying anticodon. Now this is transfer RNA, meaning that I have to convert from DNA to messenger RNA to transfer RNA so that I can get the answer here. The anticodon will be ensure that will ensure the correct amino acid is placed in the normal protein, in the normal this one. So it will be, it's gonna be exactly it's gonna be exactly the same, but because amino acid is on the, it's not there, it means that it's gonna be UUU. So it's gonna be, basically it's gonna be this. So you're gonna convert it TC, convert it to, it's gonna be AG, and then convert it from messenger RNA to transfer RNA. So it's gonna be UUC. So that becomes the correct answer. So D becomes the most correct answer. Then we go to the next question, that which amino acid will be found in the protein as a result of the mutation. Yes, which one is gonna be correct as a result of the mutation? Here we are seeing that found as a result of the mutation. Since we are looking at the mutation, so we have to go back and look at which one is it. So we have to consider that. So they are saying that which amino acid will be found in a protein as a result of mutation. Amino acid will sequence will not change or unaffected as okay. Let's find out. When you convert this, because this is messenger RNA, so this is gonna be AAG. So I find out what is AAG. AAG is leucine. No. Yes, it's not leucine, is lysine. And then what about this TTT? When you convert it, it becomes AAA. So what is AAA? Is the same thing. So the answer here therefore becomes the amino acid will not be changed or affected. So that is the answer. Which of the following describe the event that occur in meiosis but not in mitosis? So in this case, we are looking at what occurs in meiosis but not mitosis. Chromatin network becomes, yes, both they occur. Nuclear membrane disappear, correct. Cytoplasm separate, yes. And then halving. Remember, mitosis keeps the chromosome number. Therefore, here the answer becomes D. So we get that as D. We go to question number 7. Question number 7. What are they saying about question number 7? They're saying that Lindewe suffer from a disorder called hyperhidrosis. This condition is inherited disorder, meaning that you obtained from the parent. Yes, which means that you cannot change it unless you deal with the genes. That result in the person sweating too much. So the scientists have discovered that the dominant allele causes the disorder is homozygous dominant for this disorder. The doctor successfully treats Lindewe's condition by injecting her with the drug to reduce the sweating. Which one of the following statement about Lindewe's treatment with the drug is correct? Treatment with the drug will ensure that Lindewe's offspring do not inherit. You did not change the DNA, so this one is wrong. Affect Lindewe's phenotype. Yes, it could be correct because the sweating is not that much, so you're no longer seeing that sweating. Don't forget that phenotype is what you see. And then they're saying that affect Lindewe's genotype. No, because you're not dealing with a gene. Affect neither Lindewe's genotype. No. So the answer becomes B. So B becomes the most correct answer. Here they're saying that the scientists raise the hypothesis that female peahens will choose to mate with a male peacock that has longer tail, more than the more eye spots over the male with the shorter and fewer eye spots. So here, what are you looking at? Which factor is considered to be dependent? If you look at this, you see that they're saying that mate with male peacock that has a longer tail, more and more eye spots, meaning that if you have longer and more eye spot, you are likely to get more mates. You understand? So the number of mates you get will depend on how many of this do you have and how long your tail is. So the length and the number of the eye spots is independent, and the number of mates depends on how long do you have it and how many are they. Therefore, here the answer becomes yes, the number of spots. No, the number of mating attempts. How many attempts do you gonna get will depend on how long do you have. So this one is dependent. It depends. The number of mating attempts will depend on yes, will depend on how many females will come, and when they come, they will be having how many attempts you're gonna be having. Actually, the female can come, but because it is interested in you. So if you look at this and this, it looks to be confusing, but how many attempts you're gonna have, you understand? Because here they're saying that to choose the mate with the male peacock. So this female will come depending on how long do you have, but how many attempts do you have when they come, it's gonna be interested, and then you'll be having more successful or more mates to solve that that issue. So the answer becomes B. it looks to be the most correct. This is a karyotype. So in the karyotype, what are they talking about? Read the following statement about this karyotype. Ne? Then they're saying that something unique on this karyotype. 44 chromosomes. Are they 44 chromosomes? No. If you count them, it's 1, 2, they are not 44. So this is ruled out. Ne? It's ruled out. I'll show you why. This is 45 orosomes. This karyotype has 45 orosomes. This, we're going to put a question mark because there is something we need to look at again. Gonosomes are different. Are they different? Someone can say yes, but here there is nothing. They're saying that karyotype is from a gamete. Yes, to look to be from the gamete because it is only having one chromosome. Okay, lastly, karyotype has Down syndrome. No. If you look at chromosome number 21, the two gonosomes are identical. The two chromosomes are identical. Are they identical? No. Yes, because check this. If it is a female, then they're gonna have chromosomes which are what? Identical. So in this case, the answer, because they're saying 45 orosomes, this individual, yes, this individual is going to have, look, is going to have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Check here. So here there are 2. You see? And then if you count them, you'll see that there are 45 orosomes. So you remove this. There are 45 what? Orosomes. So this marks to be most correct answer. So it means that number 2 is correct and the number 4 is also correct. So to become D. Then I'm saying that which example? Why? Because there is no Y here. There is no Y. Someone is going to ask you why. There is no Y there. The Y is not what? Indicated. They're saying that which example is not source of variation? Here they're saying mutation is correct. Crossing over is also correct. But when does it occur? They are saying prophase 2, wrong. Random arrangement of chromosome, correct. Random fertilization, correct. So this one becomes wrong. So the most correct answer here, because they're saying not. You see? So the question is coming in negatives. So the answer becomes B. So you get the 2 marks there. They say, let's go to the terminologies. And then we find out how is these terminologies. The type of nucleus that contain one set of chromosome, haploid. Then they're saying differentiation. Differentiated cells that can be able to specialize into specialized tissue. I think those people who tried to attend my class yesterday, and yes, I think you got the marks. I think you got the what? The marks. And I also want to talk about one student who just sent me a message, and that message was so good. Yes, the student sent me a message just trying to thank me for what, unfortunately, the student did not send the name. He said that, I just want to thank you for all the resources you have shared and continue to share. They really mean a lot, sir. More especially for us who cannot afford extra classes. I know that outside there are some people who cannot afford extra classes. So That's why this channel was created, so that we can help you out. I really hope that one day I can touch your life. Thank you. The very same way you have touched mine. I didn't know that outside there I touch some people's life. But thank you very much. I pray that we have responsible citizens, because that is my agenda for this channel. And at this point, I don't see a reason for not getting a distinction in the life science after all your tutors. Thank you very much. Also, please use the distinction material. I really appreciate that. Some people, they send me comments, they are very happy for what we are giving them. And if really there is a challenge you find, let me know, or let us know, so that we can rectify those challenges, or we can try to modify them so that at least they become better for you. Yes. But there is only one challenge. I don't know how it's gonna deal with it. Is my handwriting. My handwriting is not good. I don't know. That's why I like to talk too much than writing. Okay, this is stem cells. Ne? Then they're saying that a characteristics where large protruding jaws. This is called prognanthus. Nanthus. Yes. They're saying the organism that contain genes inserted from an organisms of a different species. Organisms. How do we call these organisms? We call these ones, it will depend on the marking line, but scientifically it is called a transgenic organism. Yes, transgenic organism. Yes, so it will depend, transgenic, genic, yeah, because of you have transferred one gene to the next gene. So maybe there will be another option for those people who have not written. Yes, the type of inheritance that result in the intermediate phenotype in heterozygous intermediate. When you talk about intermediate, we are talking about incomplete dominancy. Incomplete. If you use the distinction material, yes, I think you are getting it right. Ne? Yes, people are using distinction material. You are getting it right. We call it incomplete dominance. African ape share the most recent common ancestor. It's a chimp. It's a chimpanzee. Ne? Is there any other African ape? Don't say hominid. Those are not apes. They are still humans, but not the modern humans. Yes. So the answer here is supposed to be chimpanzee. Okay, they're saying that the inheritance disorder result in the absence of pigment in the photoreceptor. This is eye. So the moment you talk about eye, you are talking about color blindness. Yes, we call it color blindness. Then they are saying that all genes in an organism of a species, we call this one a genome. I think people who were discussing with, I was discussing with yesterday, I think you are getting it right. Ne? They're saying decrease in genetic variation. What decreases? Decreases genetic variation. This one increases, but cloning decreases. So it is A only. The word only must be there. Site for meiosis in plants. I asked a question yesterday, one of the students, that give me where is meiosis taking place? Then someone saying that ovary. Say, he said testis. Then I said that I didn't say that. Give me where meiosis occurs in animals. I was asking where is meiosis, because I know they like this. Anyway, here they are saying site for meiosis in plants. Here it's none. It's not ovule, because ovule are being produced as a result of meiosis in plants. And the stigma, no. So the answer is none. And they are saying that the variation in blood, the type of variation in blood type, how do you call that? You're either A or B. You can't be both. You're A, B, or O. You can't be O and at the same time A, B. No way. So it's discontinuous. So the answer becomes discontinuous variation. And I gave you a clue that if you find out that the variation or the characteristic you are born with are not changing, you still have them, then we call that type of variation discontinuous variation, because it's not changing. But the size, look at the size. The way you are born is not the same size the way you are. No. So it becomes continuous variation. Let's go to the next question. They're saying that give the letter and the name of the part. Letter and name of part. That they want two of them, this and this. Forward position is in forward position, and the head and the body are vertical. Yes, it is D. So its answer is D. Is it foramen magnum? Yes, that's what we call. It's foramen magnum. So that's what you're supposed to say. And then you're saying that the functions to support the internal organs. Basically, it's a skeleton. You can say a skeletal system. This, it could be this. Yes, skeletal system. Or maybe, in this case, because they're saying support the internal organs. So basically, is this vertebra. Yes, this tries to support lumbar. You call it lumbar, or lumbar, if it is this, because basically here what is being pointed on is lumbar. Yes, so you can call it lumbar, or basically is a skeletal system, or it is the specific name there based on this is the lumbar. Yes. They're saying that state the shape of the spine in gorilla. Gorilla is the C shape, and then humans is S shape. I think the people who are dealing with the distinction material, you are understanding what I'm trying to say. Yes, I think you are getting it right straight away. Ne? Because why? Because we have been talking about this over and over and over again. Okay. Okay, it was opening the distinction material. Sorry. Yes, let me show you what they are trying to show, to ask you here. Let me make it smaller. Yeah, this is what they're asking you. This, the shape, you see, is C shape, while this one is S shape. Then they're saying that name the model of location or locomotion the spine has. Check. You see that here we say this is quadrupedalism. Yes, for this, and then this is bipedalism. So those are the answers for that. So it's quadrupedalism here, quadrupedalism, and then this is bipedalism. So yeah, I repeat before you go to your final exams, please use the distinction material. You'll never regret. You will never regret. Yes. And then they're saying that cats inherit many genes that affect the appearance of their coat. One gene control whether the coat is striped or solid color. And this animal, or this cat, the allele, this is A, is for stripe, and then for solid is this, meaning that this is dominant over this. The second gene is meaning that now the moment you talk about the second gene, then automatically this is the hybrid cross. You have, yes, long hair, short hair. Long hair is capital R, short hair is small r. So they're saying that the male who is heterozygous for both this hair mate with short hair, okay, state the type of genetic cross represented above. We say it is dihybrid. Dihybrid cross. And then they're saying that the genotype of the female cat. They're saying that the genotype of the, here they said that the mate with a male mate with the short and solid. So in this case, we see that striped is dominant over solid, and then long is dominant over short. Therefore, it's gonna be short. So if it is short, we say the small l, and the solid small a. But because they are starting with the, okay, you say it is small a, small l, small l. So it's supposed to be small l. So that is the genotype of that. And they're saying possible gamete produced by the male cat. Don't forget that this is heterozygous for both, because they said heterozygous for both characteristics. Therefore, I'm going to have, let me write it here, both characteristics, meaning that is going to be capital L. What is it starting? Let me see. Long, okay, capital A is the same thing. Capital A, capital, no, heterozygous. They're saying heterozygous. They say the heterozygous, capital A, small a, capital L, small l. This is the genotype of that, but they're looking for the gamete. So this is not the answer. They're looking for the gametes, of which this is going to go with this. I'm going to have capital A, capital L. This is going to go with that. It's capital A, small l. And then this with this, small a, capital L. And then this is this. It's going to be small a, small l. So basically, these are the gametes which are going to be formed. So I think when they're marking, it's going to be 1 mark there and then 1 mark there. Or any 2, you get 2 marks there. So that is this answer. This is the answer for this. They are saying name the type of inheritance shown in the this above. How do we call this? This is basically it's complete, but it is inheritance of multiple genes. Yes, multi-gene inheritance. Yes, it's multiple genes. If you are looking at it there, yes. Then they are saying that what percentage of offspring, yes, this same phenotype as the mother. Now here you have to cross and then find out. It's a long question, eh? Okay, so what are you supposed to do here? You're supposed to do the cross and then find out what is the percentage. But we will find out that, remember, if you put it in the, remember the mother, they are saying as the mother. Remember the mother is small a, small a, small l, small l. So if you put it in the Punnett square, you'll find out that we say that it is we say that is small a, small a, small a, small l, small a, small l, small a, small l, small a, small l. So you can complete it. And then here we say it is capital A, capital L, capital A, small l, and then small a, capital L, small a, small l. So when you look at it, we are looking for something like this. So the moment there is capital L, this, all these are out, because all this, it will have all of it. So it is only this 4. This, and this. So it means that it's going to be 4 out of 16, because the whole thing is 16 times 100. So this is not what they want. They want, which is 25%. Maybe they will just say 25 and a percent. You saying 25 and a percent, they will give you a mark in that regard. So the answer is supposed to be 25%. We go to section B. So 50 marks are gone. Section B looked to be a little bit easier. Yeah. Anyway, let's look at it. The diagram below shows the DNA and RNA that is involved in transcription. So if it is involved in transcription, let's look at the questions. What is 1? Where is 1? 1 is the strand. This check. This strand is DNA, you see. So definitely this is going to be adenine. And there are 2. Where is 2? This one, don't say ribose sugar. Don't say sugar. No, it's deoxyribose sugar. You see? Why? Because it is a DNA. Yes, because of the thymine. And then they're saying that number 3 is phosphate group. Phosphate group. Identify the strand that represents messenger RNA. So it's this strand X. Strand X. Give one visible reason, because it has uracil. You see? It has uracil. And then they're saying that describe the process of transcription. This one you have to write. Everyone knows the process of transcription. Even if I don't repeat it, It's the same thing. DNA double helix unwind, weak hydrogen bond break. Then unzip is the same thing, weak hydrogen bond break, to form two templates, one acting as, to form two strands, two separate strands, one acting as a template for formation of messenger RNA using free-floating nucleotides, using free-floating messenger RNA nucleotides from the nucleoplasm. And then whereby adenine goes with thymine, adenine goes with the uracil, and then guanine goes with cytosine. Messenger RNA is formed with the coded information which moves out of the nucleus via the nuclear pore to the cytoplasm on the ribosome. Basically, that's what you need to describe. All those points, at least you'll be able to get 5 marks. As long as you arrange them in order, and then those points are there. There are more than 5. That's why I don't usually talk about, I'm not going to write that much. Explain one reason why transcription is important. It's basically important because transcription results in the formation of messenger RNA, which is used to form the proteins. Basically, the messenger RNA has the coded information for the protein. That's what you are supposed to write. Formation of messenger RNA, which contains the coded information for formation of the protein. Yes. So, then we go to the next question. Next question is Mr. Mega Buck. Hey, I like the name. The wealthiest man in the world. Yeah. Hey. Recently died, and according to this will, Mr. According to his will, he said what? What did he say? The children will inherit. His children will inherit, yes, his wealth or fortune. Since his death, 3 ladies claimed to, yeah, obviously, if there is money, even those people you have never seen, their mothers, the mothers might come and say, no way, this is my son's father. Okay, before this died, his lawyer insisted to do the DNA testing. And then what happened? The DNA test was done, and was done between him or among the wife, the children, and him. All right, so this is his DNA. So the first thing you need to do, when there is 3 people which are involved, the DNA of the mother, and the DNA of the husband, and the DNA of the kids. Yes, if the kid is the son of this parent of one of these people, basically all the bars, yes, their profile must be matching with this. If here I have 25%, the remaining 75% must match, meaning that there must be no bar which is left out to confirm that that is the parent. Therefore, let's go. Give a scientific reason why lawyer insisted on DNA profile from the woman and their children to determine the parent. Why? Because DNA is unique. Yes, there is no way how you're gonna manipulate it. Is unique, so that you identify the actual parent of these children, or to identify the actual children of this gentleman. You understand? Yes. They're saying that which child will inherit this is wealth. So you have to identify. Okay, now let's go. Start with this. This doesn't match. That is just as doesn't match with the mother and the father, then that is out. Okay, we go to this, out. This matches with the mother. So this matches with the mother. Nothing matches the mother. Yes. So this one is out. This is completely out. What about this? This doesn't match with the mother, but matches with the father. So this is first. This doesn't match with the father, but matches with the mother. Okay, this matches with the mother, but not the father. Correct? This matches with the father. You see? Does it? Yes, it matches with the father. Correct? This matches with the mother. Correct? This matches. Are you sure? Yes, with the father. Yes, correct. This matches with the mother. Correct. This matches with the mother. Correct. This matches with what? With the father. You see that all of them have been utilized. If there is one which is out, that's not your father at all. Then this one, we come here. This matches the mother, matches the mother. Nothing. It's out. The moment it's out, forget about the rest. You see? So basically, this child, wise child, is going to inherit. Actually, it's the son. Yes. Explain your answer using the evidence above in the diagram above. Why is this one a child? Is because all the bands of the child match. All the bands the bands of the child. Yes. Some match with the mother, the woman, and the remaining one match with the, you see, those which match with the mother is fine. The remaining one must match with the father, or some match with the father and the remaining one will match with the mother, meaning that this is the mother and this is the father. Yes. Oh, if you calculate, if you find it's 50% matching with the mother, correct, and 50% they must match with the father to make it 100. Name one simple source that could have been used to get DNA. It could be blood, it could be hair, it could be saliva, it could be skin, it could be anything which is easy without affecting the mother or the father. Just in case electricity goes, maybe we can be stagnant for some few minutes so that we switch to the system. Anyway, let's continue. They're saying that what is A? This is the chromosome. I told you that when you get this, you have to first, and then this is the spindle fiber. Yes. Then they're saying name the process that is represented. Look, this is single chromosome, meaning that is must be meiosis 2. So because it is in the middle, therefore is metaphase 2. Metaphase 2. They say name the chromosome that were represented. How number of chromosomes presented in the parent cell, because there are 2 here, so there were 4. You see, one daughter cell at the end, there are 2. And they are saying give the name of the structure. A, we said is chromosome. And then give the function of structure B, to hold the chromosome in what? In a position. Yeah? Yes, you see. And also they are used to pull the chromosomes to the opposite pole. Yes, they contract and then they pull the chromosome to opposite poles. Yes. Describe the process of crossing over. I think people okay, anyway, let me not talk about it. Crossing over, basically you look at chromosomes of homologous chromosomes. The homologous chromosomes, they align together. Chromatids of homologous chromosome, they cross over at a point called chiasma. You see, and even they didn't talk about the phase. So you mentioning the phase is also another mark. The genetic material is exchanged, and then after that they split, and then they will have genetically different genes. Yes. So chromosomes of which are homologous, they align together. Chromatids of homologous chromosome, they cross over at a point called chiasma, and then after that, what happens? They exchange their genetic material, and then they separate or they split to form 2 genetically different chromosomes. This process occurs in prophase 1. Yes, you can also talk about that, so that maybe if the mark is there. All right, they're saying that draw the phase, the diagram of the phase that occurs after the phase. So here they are looking at anaphase. But when you're drawing, you make sure that, let me show you. Make sure that when you're drawing, is actually supposed to be like this. Let me show you. It's supposed to be like this, ne? I don't know whether you can see. It's supposed to be like that. You see, it's gonna split. When it splits, so make sure that these variations are correct. Yeah. So make sure that the variations are correct. So this one is gonna move this side, and then this one is gonna move. So meaning this and this is gonna be black. When you're shading it, you must shade it black. You see? And then this one and this is gonna be it's gonna have a black thing there, and then this is gonna have, you see, you're drawing what will happen. It's a black and a white, black and a white, and then this side is gonna be what? A white, based on what it's shown there. And then spindles, and then you write the phase there, the title of the what? Of the diagram. So meaning that you're right, and then you say that because the question they're saying that the next phase, the diagram of a cell and what did they say is the question? Give a function, okay, describe the process we done, draw a phase, okay, in the, yes, diagram of a cell showing anaphase 2. Yes. So basically that's what you're supposed to say, caption, and then you draw these chromosomes I've shown you with, you draw them nicely, ne? The way I've tried to show you, and the variation, because they might be marking the variation, but what I want to show you is that they are supposed to be 2 chromatids on each side and then separated, and then the caption, so that when they mark you, they give you correct answers, they give you ticks. Let's go to question 2.4. They're saying that familial this syndrome, whatever, is inherited condition caused by the dominant allele. People with this condition have a high level of cholesterol, fats too much in their blood, and then this increases the risk of their blockage, meaning that you are likely to suffer from stroke. The diagram below shows inheritance of the parents of several. Something is important here. Generation, okay, this is caused by a dominant allele. Look, the person with the, because here they are saying that caused by a dominant allele. So that's a point. As I was explaining yesterday, that this dominant allele, you won't miss it, because they know that these days you fail it. So in that case, you come and then you explain. Let's read and find out. Let's read this question. Something is not right here. Is it me who is colorblind? I don't know. Okay, we shall find out. How many generation? You know why I'm saying is because I'm seeing a key here. They're almost the same thing. But if I'm not forgotten, I've not forgotten, this was shaded, this was also shaded, it's supposed to be shaded, one shaded, but these ones, some of them are supposed to be shaded, some of them are not supposed to be shaded. But I remember this was shaded. I think even one of it was shaded here. But it's okay. You'll check on your paper and then find out. Ne? How many generation in this? We count the generation. 1, 2, 3, 4. So 4 generation. Someone is gonna say, no, not this one you don't count. Why not counting the parents? Are they in the same generation with you? Are you in the same generation with your parent? They'll say that our generation need to be like this. Okay. Determine the number of males in the diagram above who are homozygous recessive. So in this case, I'm not going to give the answer for this for now. Not because I don't know. Yeah, it's because they shaded. I would have been with the original copy, ne? And I don't have the original copy, ne? Just use the photocopied one. Okay, I would so count the box you're supposed to count. Let me just give you a clue. You're supposed to count the box. Yes, the box who are shaded. No. Just a moment. Let me just go get just 2 minutes, 1 minute. Get the original paper. Yeah, I'm back. I've got the paper, the original paper. Let me just find out. Let me just shade it for you so that we are on the same. Sorry, I'm not having a memorandum here. Just creating the memorandum as I'm discussing here with you. Yes. Yeah. So it's like I'm also writing the paper. I'm also writing the paper. All right. Okay, yeah, now I've got it. Yeah, this one is shaded. This one is shaded. Yes. This one is shaded. Yes. This one is shaded. Yes. Cool. J is shaded. Yes. Shaded. N is shaded. O is shaded. P is shaded. Yeah, if I don't shade all of them, then you'll find out. Yeah, but let me just show you. All right, they're saying that the shaded ones are the one who are with the problem, and then the unshaded are the one without the problem, meaning that the shaded ones, they are supposed to be either capital T, or capital T, small t. Those are the shaded ones. The unshaded ones are supposed to be, don't forget that this is caused by a dominant allele. If it was caused by a recessive allele, then this would have been the shaded and this is unshaded. But because it's caused by a dominant allele, the shaded ones are the one which have the what? Yes. So now let's go through this. The question is saying that determine the number of males. They are looking for males. Oh yeah, it's the males. So the males are the one with the box. They're saying the males with the problem. So it's 1, it's 2, it's 3, it's 4, it's 5. Based on what I've shaded, yeah, it's 5. 1, 2, 3, 4, 5. Yes, the answer is supposed to be 5. Determine the number of males in the diagram above who are homozygous. Okay, we are done with AA. The answer, this answer is 4. Just down here, it's 5. Here I said how many generations? There are 4. Saying, hey, G and are planning to have the 4th child. What is the cross to show? Use the genetic cross to show the percentage probability of child without, meaning that it's supposed to be homozygous recessive. So you guys remember they're saying that they are planning to have another what? Another child. Yes. So okay, I'm gonna say P2 F2 phenotype. So you're saying that G is a male who is shaded, who is having the disease. Yes, you put it there, cross with a female which is H without a disease. You also put it there, and then here you say gamete. Sorry, genotype. So if it's a genotype, because this one is having a disease, but this person came from top here. Where is the G? Oh, G is here. It came from top here, of which this one was also sick, meaning that this one is heterozygous. You understand? Yes, it's heterozygous for the what? For the condition. Yes. So why? Because this one is capital R, small r. Sorry, capital T, small t. This is small t, small t. Definitely this one to be having a disease must have a capital T. There is no way how it's going to be capital T, because this one was the already what? Normal. Therefore, now the genotype is going to be capital T, small t, and then H is normal. Remember, it's a recessive. Yes, I think. Now, gametes. Gametes will form as a result of meiosis. So meiosis is going to take place. You form the gametes. It's going to be capital T, small t, small t, small t. Then now fertilization is going to take place. This with that, capital T, small t. This is that, capital T, small t. This with this, small t, and then this with that, small t, small t. So since small t, these are offsprings. You see, F2, it must be in the same line. We come here, we give you a mark. Meiosis and fertilization, we give you a mark. The genotype and phenotype from the question still, we give you a mark. Then we give you a mark here. But there must be a compulsory mark here. A compulsory mark here, the question is saying that use the probability. So in this case, the probability is going to be a compulsory mark. So it's going to be 2 of them, they are normal, and 2 of them, they have the condition. So it's going to be out of 4 times 100, because they are looking for the probability. Yes, the probability, or the percentage probability, which is equal to 25%. So that is the answer for that case. So yeah, let me put it back. I think I'm done with that question. Let's go to question 2.5. These papers were too much reading. Anyway, lactose is the sugar in milk. Lactose is found in milk. It's digested by the enzyme called lactase. Then some people cannot produce lactose, and therefore they become lactose intolerant. And then the condition is caused by recessive allele. This means that the people with the homozygous recessive, they become lactose intolerant. People from different countries, okay, were tested to determine if the lactose intolerant, they were lactose intolerant. They are saying that the table below shows the number of people tested in each country and the lactose intolerance percentages. Okay, let's find out. The population. Here you're having the percentage, and then tested people. The population size of country B. Where is country B? Okay, is 190 million. They're saying that calculate the number of people in this country likely to be lactose intolerant. Here they're saying the percentage of people who are lactose intolerant. So here these are the number of people they tested, and this is the percentage of the people, you understand, who are lactose intolerant. So meaning that in this case, because this is the percentage, so I'm gonna say 12 divided by 100. That's the meaning of percentage times the 190 million people. So when you get the answer, I think it's around 22.8 million people, something like that. You can calculate and then find out the actual answer. But that's how you're supposed to do the calculation. There is another method, but let's just go with one as long as you get the correct answer. Which country is most likely to be having the least reliability? Check here. Remember when I was discussing yesterday, I said that people, you need to know what is it. So the answer is E. Why? Because we use the same fewer number of people. Remember, if you want to increase the reliability, you increase the sample size, the number of people who are in this investigation. You understand? Yes. So we go to the next question. Yes, we go to the next question. Just a moment. Yes, let's go to the next question. Give a reason for your answer. We say that because you have used this fewer number of people. Provide 2 ways in which the validity of this experiment can be improved. Validity is you are keeping things constant. Yes, use the same number, maybe the age. Remember, age can also affect the people, same kind of individuals. Yes, you are busy increasing the what, this validity. Keep the same type of the food you are giving them, because they might have different concentrations. Yes, meaning that the concentration of food or the type of food you are giving these people, which has the milk, sugar. Yes. Then there are so many. Describe the planning there a lot, because we are using different countries. It depends on where. If you are going to fly to different countries, then therefore you have to plan in advance, maybe requiring permissions or visa things, the budget. Look at the budget. Ask for permissions for these people. Are we going to be experimented about? You have to look at the suitable time for these individuals. So there is a lot of planning steps you can consider. Then let's go to question number 3. Yes, question number 3. Yeah, we are almost done. That was question number 3. Okay, it's too much of talking than writing. Flies lay eggs in urine and fish that stuck on the wall of the what? Of the sheep. The eggs hatch and then larvae damage the sheep's skin, leading to the infections. To reduce this problem, farmers in New Zealand have been developing sheep with no wool growing on their legs. Then they're saying that the back side, that's the bottom, or the under side of their abdomen. Farmers have crossed different breeds of sheep to produce offsprings with desirable characteristics. And they are saying that the characteristic of other breeds of sheep are shown in the table. Okay, they're saying that use the evidence from the table to describe how the farmer could use selective breeding to develop. So if you look at our distinction material, it shows you what exactly you're supposed to do when you are dealing with the selective breeding. Yes, it's here. Sorry. Yeah. There is basically what you're supposed to do. In this case, the first thing you're gonna do is to select the individual who have what you want. So basically, you want these legs. You want bare legs, ne? Did they say bare legs here? Yes, the farmer crossed breeds, okay, of springs with desirable characteristics, and then with other sheep. Okay, they're saying that produce. Yes, he said it here that use the evidence to describe how farmer could use selective breeding to develop the sheep with bare legs. You see, bare legs, and then backside, bare backside, not abdomen. They're looking for backside. So meaning that this individual is gonna cross this, because this has but they doesn't have the bare backside. Therefore, it's gonna cross the one which has bare legs and then the backside, and then it will deny sex to this. So meaning that this one will not be given sex. Only this one will be interbred, and then yes, you keep interbreeding. This is what I'm trying to say. So what happens is this is what you can say. For example, to increase the quality of is here. Suppose you want a variety of cow to produce a lot of milk. Now, in this case, suppose you want to have this sheep with these characteristics they are looking at. So you see now you are writing. Choose the cow in the herd to produce, that can produce most milk. Choose the sheep that is this, the one which can have bare legs, and also, so these two, because they asked you how did use the evidence, describe how the farmer could use. So we did select this and this because they have these desirable characteristics. So after that, then allow these offsprings, let only allow these cows to reproduce. These ones which have the desirable characteristics, these sheep, you allow them to interbreed. That's why I'm saying that you are denying this one sex. And then after that, what happens? The one when they get the offsprings, yes, the offsprings that produce these desirable characteristics, the one who has bare legs, and then this, you cross them again. You see, you cross the offsprings that can produce this characteristic. You let this offspring reproduce again. You understand? So at the end of the day, you have animals with this and this together. One animal with those two characteristics. That's how you're supposed to explain. Then give two tabulate. Here you're supposed to draw a table. I told you guys, you draw a table, complete table. You don't draw a T table. This is wrong. You get a 0. So don't cry when you find a 0 there. So tabulate the difference between selective breeding and natural selection. Selective breeding, basically, this one occurs naturally. So the nature will select. Here it is human needs. Yes, then basically it's like artificial selection versus natural selection. Environment will do the selecting. Human will, is the selective force. You understand? Yes. It occurs within a specific species. This one can occur in multiple species. Multiple species, yes. Because you are just crossing, yes. So you only select what is best, what you think it is good for you. So basically this is the differences you're supposed to write about. Explain one disadvantage of selecting breeding of sheep in this question above. Basically, when you do the selective breeding, this sheep might lose important characteristic which might help it to be able to survive in the wild. You see? Yes. Maybe the hair, and then you don't want this hair, and the hair is there to protect this sheep so that it can sustain in different temperature conditions. And then when you just want to wipe it out because it is doing the caca on the wool, because you don't want it. So at the end of the day, that character might get extinct. So that's, whenever you're doing selective breeding, you are not leaving the nature to select against those who are not best suited. Then you are doing some disadvantages to the animals. Yes. Then when we go to question 3.2, the diagram below shows the modern swordfish. The ancestor of had short sword that yes, the modern swordfish have long sword. Yes, the modern swordfish have long swords. Yes, the swordfish use their swords to kill the prey, meaning that they're using it to kill the prey too much. The injured prey is easier to eat. You see, it's easier to eat and it's easier to catch and eat. Something is important here. Okay, they are saying that the diagram below shows one theory of how the length of the sword it got evolved. Here is many generation saying the sword grew longer each time the swordfish uses its sword more and more. The more you use the structure, the more it becomes bigger and stronger. So it means that that's Lamarck's theory. They're asking a question. Suggest which scientist suggested a theory above. That's Lamarck. Explain one reason why this theory is rejected by most of the scientists. Because organisms don't evolve because they want to evolve. This sword did not evolve because the fish wanted it to evolve. and then there is not enough evidence to show that this acquired characteristic during lifetime is passed to the next generation. You understand? Yes. Describe how natural selection could result in modern sword. So here you're using Darwin's theory. So what are you going to write? So here you talk about the variation, that there is variation among the swordfish. Then you tell us the variation. Some swordfish had long snout. How did they call it? They call it sword. The sword, yes. Some swordfish had long swords, and some swordfish had short sword. Yeah? Yes. Those with the long swords, here they said it. They say that they could. Yes, check here. Those with the long sword, yes. What happened here? The one with the long sword, what happened? Yes, they are able to kill the prey and catch it and also eat it. Therefore, they survived in large numbers. While those with short sword, they couldn't get or catch the prey and eat. Therefore, they died in large numbers. Those with long sword, they passed their genes. This is called natural selection. Those with the long swords, they pass their genes to the next generation of the long sword. After many generations, all swordfish had long sword. See? Yes. So when you're explaining it, you're supposed to link it to this example. Yes. Then the next question they're saying that describe. Yes. So we are done with that. So variation. Some swordfish had short sword and then long sword, and then those with long sword could catch the prey and kill it, and then they survived in large numbers, while those on a short sword, they couldn't, and then they died in large numbers. Those with the long sword, they passed their genes. This is called natural selection. Those with the long swords, they pass their genes to the next generation. Yes, after many generations, all the swordfish had long sword. So that's what you're supposed to explain in that regard. Then when we go to 3.3, 3.3, they're saying that the table below shows the average cranial capacity in hominids fossils. Okay. State one Australopithecus africanus species you have learned about and give. Yes. So what are you going to give? And give the common name, and then so basically you just supposed to choose maybe Australopithecus sediba, that's Carabo, who discovered it. So all those things, they're saying Australopithecus africanus. They're saying, did they say africanus? It is wide. Afarensis, so it's only Adipithecus, Australopithecus Lucy Johansen. Then Australopithecus africanus, you can says place Robert Broom, yeah, Littlefoot, Clark, and then Australopithecus sediba, Carabo, Idebega. Then all this you can choose whatever you want to choose, and then just tell us Australopithecus, then what is it, and then you tell us who discovered it. So you choose any of these Australopithecus from here up to there. So you choose any. Yes. And I was emphasizing how do you memorize that. Step 2, advantage of having a large cranium. Large cranium is going to bring about more brain cells. And then these more brain cells are going to is going to bring about intelligence. And then if you have more intelligence, meaning that you can think more, you can reason more, you can have more skills. All those are some of the advantages of having a large cranium over a small cranium. Then that's what you're supposed to write. Then they're saying draw a bar graph. Draw a bar graph to compare the average cranial capacity of Homo. Guys, this question was a little bit not supposed to be rushed on. You know why? Let me show you. They're saying Homo species. Some people are busy touching on their head. Oh, I drew everything. That's why I say that don't rush. Don't finish the paper in 1 hour and then you say, yeah, I'm done. You think that the examiner is a fool. No. Okay, they're saying Homo species. So it means that your bar graph was supposed to be only those individuals, because those are the Homo species. You see? So from there, after drawing that, then what are you supposed to do? You come here and then you tell us, you draw. You're supposed to draw the bar graph, but now what goes on x-axis, what goes on y-axis. Hominids, they go on the x-axis. So you tell us here the hominids, and then the y-axis, they are saying number of fossils, they are saying cranial capacity, because they are saying cranial capacity. So meaning that the cranial capacity is supposed to be here. Did you give the caption? Did you give what is here? You put the names, the spaces are correct, the scale is supposed to be the same, depends on the scale used. Yes, the small one is, the smallest is 640, the biggest is 1350. Maybe you can start from 0, 500,000, something like that, or you can be 250. Yeah, 0 to 50, 500 to 550, 600, 750, something like that. Yeah, so like that, 0 to 50, then 500, 750, 1000, 1250, 1.5. So like that, it must have the same range, so that we come here, we say caption, we give a mark. We say correct type of the graph, we give a mark that it's a bar graph. Yes, it doesn't matter. And then caption, you put the caption, you give a mark. The spaces are correct. It must be correct, same spaces, same size. And then scale, we see the scale on x and axis. The scale, we give a mark. Have you labeled it correctly? X and Y, you have labeled it correctly, give a mark. And then plotting, maybe we can give you 2 marks there, 1, 2, 3, 4, 5, 6. So maybe that's how we're gonna be marking it, so that now you get the 5 marks, all of it. Then they're saying that explain the genetic evidence to support the out of Africa hypothesis. I was explaining it even yesterday, and then I was giving an example with these kids, and then people. So this is what we are supposed to say, that inheritance of only inherited only from paternal line. Analysis of mutation of this mitochondrial DNA show that the oldest female, yes, the oldest female, yes, the oldest female, what happened to it? The oldest female ancestor. Just a moment. Thank you for, yes, I was explaining that the mitochondrial DNA. So here, if you look at this question, check how the question is saying that explaining how the genetic evidence support the hypothesis that ancestor of Chinese Homo erectus fossil originated in Africa. So here you are showing that this mitochondrial genetic evidence, and we have mitochondrial DNA. It is inherited only from the what? The maternal line. You understand? And then analysis of mutation of this mitochondrial DNA show that the oldest female ancestor with the were located in Africa, and all look, and all now look, and all these Chinese, and all these this came from what? From Africa. So basically, that's what they are looking for in that regard. Yes. So the next question is saying that study the phylogenetic tree. I was clear yesterday when I was trying to explain this. Study the phylogenetic tree showing the evolution relationship between the birds. These are the birds, the eaves and the crocodiles. Ne? Answer okay. The following questions. When did this branch? When did the Eve or the birds or branch crocodile? When did they branch from each other? So it will depend on this will depend on, if you look at this, I don't know whether I see it properly. I don't have the original again. If this is 1 year, 2, 3, 4, 5, 6, from here to here is 1 year, 2 years, 3 years, 4 years, 5 years. Basically, they separated here. This is when they separated. If it is in terms of years, they separated in million years, meaning that's 5 million years. But this is where they separated from. I don't know whether they forgot to put the numbers here, 0, 2, 4, or depends on what we're supposed to be here. So they will see how they're gonna mark it out. But it was supposed to have a mark there. Yes, it was supposed to have figures there. Let me check from the original paper. Then the original paper has nothing. Okay, it has nothing. So I think they will put a concession there. But if it is regarded as 1, then we would have said that then it would have been 0, 1, 2, 3, 4, 5, meaning that they separated 5 million years, meaning that is 5 million years ago. That's when they separated. But they're saying that give the term that describe the structure shown in the symbol of the forelimb that was inherited in common ancestor. Which structure? Oh, this one. Yes, forelimb, this one. How do we call this? How do we call this? We call it pentadactyl limb. Let me show you where I put it in the booklet here. The distinction material. Yes, it's here. We call it pentadactyl limbs. You see? It's here. Pentadactyl limb. This one. This is what they are looking for. So pentadactyl limb. And then they are saying that that is the answer for that question. Then the next question, the second last question, they are saying that too much reading. Okay, that imps and ostrich are both large flightlet birds. Imps are only found in Australia continent, and then ostrich are found in Africa. All right, tell us the story. We are listening. Okay, this is the imps, this is ostrich. The original hypothesis about the evolution of the imps and the ostrich was that they share a common ancestor. They share common flightlets. This is a very important thing. Share. Okay. The sea separated the ancestral population during this So meaning that this is the force which separates the continents. The Australia and Africa, yes, more evidence has resulted in a new hypothesis that the ancestral population of emus and ostrich was highly flying small ancestral birds that could fly long distances and migrate from Central Asia to continents in the southern hemisphere. The new genetic analyses show that mutations in DNA cause modern birds to lose the ability to fly eventually, and then describe the question how the high-flying small ancestral birds evolved into different species of large flightless birds, ostrich and emus, on the other continents. So how are you going to explain this? So basically here this is speciation, yes, and this kind of speciation is the allopatric speciation because there is a geographical barrier which is separating them. So what are you going to say here in the so you can say that at first there was one big mass with a common ancestor of flightless birds, because they said it the originals, they're saying that we're sharing a common flightless bird, and then separated into this by this, so meaning that at one time there was one large mass with a common ancestor with a common flightless ancestor. You understand? Yes. Or this flightless ancestor lived on one big mass at the beginning. And the mass was divided into Australia and Africa, whatever. Yes, due to continental drift. I see. Yes. And then these organisms which were in Africa, where exactly? Which organisms were in Africa and Asia, they could not interbreed because they experienced different environmental conditions, and because of the no gene flow, the natural selection occurred independently. Therefore, they became genotypically and phenotypically different. If you remove this water mass, they say the sea, they say it somewhere. Yes. These birds cannot interbreed. When they mix, they cannot interbreed to produce a fertile offspring, therefore resulted into large flightless birds, ostrich and emus. Yes, so this must be written at the end because this might be a compulsory mark, because this is what you are looking at. And also a common ancestor of flightless birds could be a compulsory mark, meaning that if this question is 7 marks, you find out that 5 marks is for free, meaning that you can score it anywhere. But you saying a common ancestor is a marking point which is compulsory, and also you saying that mentioning these birds, yes, because that's what we are looking for, might be a what? It might be a what? A compulsory mark. Yes. So if you're a teacher and you are here, please try to look at it on the memorandum and then find out where to put a mark, not just giving marks. Yes. So that's how you explain that. Then the last question, but not the least, is the last one. Identify. Here you have yesterday I was at one of school. I was in 3 schools, but there is another school where my colleague called me. It's called Wiseman. All right, yeah. As I was about to leave, a student asked me the same question about punctuated equilibrium. How do we differentiate between punctuated equilibrium and gradualism? I don't know why this always disturbs students. when it comes to explain, but let me explain it. Here it is punctuated equilibrium. The diagram you see here is punctuated equilibrium. It has periods of rapid change and long periods of no change. Short period of rapid change and then no periods of change. You see, short period of rapid change from here to there, and no period from here. No periods of what? Change. From here, you see, rapid change, rapidly they change without seconds and seconds, you understand? And we say that this is caused by a mutation. Imagine from here, from this kind of rat to white, immediately in the morning you find yourself that, and then from this kind of rat to a dark one, from a gray to a dark one, we don't have, it's caused by a mutation. So that's number one. Punctuated equilibrium is caused by mutation. Number two, it has no intermediate at all. Because how is this linked to this? Nothing. It's just rapid change. Then number three, it takes place within a short period of time. Short period of time. If you compare it with gradualism, which moves slowly, you'll find out it's caused by natural selection. The nature must select, and the nature, it's slow. Select small. Then when you talk about it has intermediate. From here to there is an intermediate, you see them, and then it takes long period of time. So now if you come here, identify the concept of evolution illustrated above. So here it is punctuated equilibrium, and then give the definition of the concept evolution as shown in the diagram above. Basically, this is evolution with rapid period of change and long period of short period of rapid change and then long periods of no change. Yes. So this also is a type of speciation. It's trying to show you how speciation occurred. So guys, I think that was the paper. How was the paper? How was the paper? Ne? Yeah, I think the paper was not that bad, but it had too much of reading. Even paper one had too much of reading, and we know that our kids, they don't like to read too much. They're not straightforward like other provinces I've seen. Yes. So if I look at the analysis of these papers they have written in terms of life science, paper one, it was not that difficult, but too much words. Was too much words. Paper two, the same story. It's not too much. Our students don't like to read. Ne? And I know you are there, you know that you don't like to read. Keep reading. It's not, you don't like it. Ne? So you guys, thank you for being here. Don't forget to get a distinction material book from the description below. Tell us what you need us to talk about. If you have any challenge, let us know so that we can help you out. And then also, don't forget to tell your teachers to help you out. We are always here to help you out, you guys, so that at least you have a better life. Yes, so that when I get to my retirement age, at least I know that I helped maybe 1 billion people. Yeah, 1 billion people. People maybe in South Africa, at least 50% of them, at least they pass through my hands or somewhere. I helped them a bit. At least I will be in my retirement with happiness. Anyway, I wish you the best in your finals. I'll be here still to help you. I've not stopped from helping you. I'll help you even if you have done writing your paper. Welcome back to our class. Today we're gonna be looking at the paper 2023 final exam. Yeah, so share the video so that everyone is here. We can discuss this paper. No one should miss out. This paper was really a very easy paper for those people who did the study, and for those people who did use my book for distinction material. Yeah, if you want it, you always get it from the link below. So this paper was a little bit easy, was just a little bit of easy, and we're gonna see how we can really see that those people, they can mark themselves and then they see, and also those who want to prepare for the next exam. All right, let's start without wasting time. Saidi, as usual. According to Lamarck's theory, I know most of you don't like Lamarck because of his theory, but me, I like him because he started the story. All right, according to Lamarck's theory of evolution, or for evolution, acquired characteristics are not inherited. Obviously, we say that Lamarck says that acquired characteristics are inherited to the next generation. If you have a person, maybe your father gets an accident or is a bodybuilder, and then you're gonna produce kids who are having muscles already. Acquired characteristic is inherited. So here the key point is not. So you have to take note of that. So this one was off. And then they are saying that organisms evolve because they don't because they adapted to their environment. No, here they are saying that organisms evolve because of the adaptation to the environment. How do organisms evolve due to the adaptation of the environment? Is it natural selection? Because adaptation will require that those organisms which are able to adapt will evolve, and those organisms which cannot adapt will die. Yes. And then number 3, they're saying that there is variation among organisms. Lamarck doesn't talk about variation. Yes, so this one is off. Then they're saying environmental change has no influence on the species diversity. Here, they're saying that environmental change has no influence on the diversity of the species. And then here they're telling you that organisms evolve because they adapt to their environment. So how are these two different? How are these two different? Here it means that environmental change have no influence. For example, that if there is scarcity of food, yes, and then the one which is on ground die out. This is opposite of this, né? And they die out, and then there will be no influence on the species, how they diverse each they are different from each other. So that is wrong, because species will change based on the what they want, but still based on the environment. If they want to be, for example, they want to stretch their necks, they stretch their necks until they reach, for example, the tortoise. If we are using the what? The giraffe. Yeah, you call them giraffe. Yes, because that's the best example most of the people are looking at. So the best answer here is gonna be B. And then they're saying that genotype for species characteristics. When you talk about the genotype for species characteristics, what does it mean? They're saying that contain 2 chromosomes. No. Physical appearance, they're saying genotype. Physical appearance is a phenotype. No. Is the composition of a gene pair. Look, they're not saying that genes. They're saying gene pair, meaning that you have. Remember that when you talk about an allele, is also a gene, but is half of a gene or alternative form in which a gene can exist. For example, tallness is an allele, and also shortness is an allele. For example, capital T, small t, for example. Yes, so this is an allele, this is an allele, but this is a gene, meaning that it's a pair which makes a what? A gene. And then is represented by the genotype of a specific characteristic. Here, characteristic. Here they're saying is represented by one allele. It's not represented by one allele. Even if it is shortness, it will be represented by the 2 alleles. Yes, for example, capital R, capital R, or small t. They are saying a characteristic. Yes. So when you talk about the characteristic, it means that they are looking for the complete set of that characteristic. So the best answer here becomes C. And then they're saying after discovery of fossil, scientists classify it as after discovery of a fossil, scientists classified it as an African ape because it had, okay, what do African apes have? Large cranium? No. Small cranium? Yes, they have small cranium. Small cranium? Yes. Large cranium? No. So meaning that A and D are out. So these two are out. So let's look at this. Small cranium, non-prognanthus, meaning that they don't have a protruding jaw. Apes have protruding jaws. Yes, prognanthus means protruding jaws. So prognanthus, this is correct. This is wrong. So it means that the best answer is C. They're saying that large canine. This one is correct, this one is correct, but this is wrong. Therefore, B is wiped out. The best answer is C. So C is going to be the most correct answer. Yes, let's go to 1.4. The statement below describes the step in the process of cloning. I explained this, the process of cloning yesterday very well, but let's ignore that you are not there. So now let's look at it. Remember, they are saying that embryo, okay, let me just show you here so that number one, you get one cell, you get a cell A and cell B. So this is animal A, animal B. Get a cell, ovum. I'm just summarizing it very fast. Somatic cell. So here is somatic cell, meaning that is 2N. After that, remove the nucleus of the ovum. Yes, throw it away. Take the what? The casing. And then here, remove the nucleus because I want the characteristic of this. Bring the nucleus, throw the casing. So bring the casing here and bring the nucleus, fuse them together with electric shock. Yes, after that, bring these two, once it is fused together, it looks like a zygote-like structure. Therefore, it becomes, I'm just explaining so that because this is the process they are trying to show you, it undergoes mitosis. Bas. Then, let's go back to the question. They're saying that the statement below describe the steps involving in the process of cloning. Embryo in the implanted is implanted into the uterus of an adult female. Embryo is implanted. This must be around this stage 4. Okay, we shall find out. Nucleus from the somatic cell of the donor cell is extracted. This could be 1. Nucleus. Check. Nucleus. They're saying the nucleus or the cell. Nucleus from the somatic cell. That's not step 1. You can't extract the cell before you get the cell. You can't extract the nucleus from the cell before you get the cell. So we need to first get the cell. We are looking for these 2 steps of getting these 2 cells. All right, we go back. They're saying that the nucleus from the somatic cell is inserted into the ovum. The nucleus from the ovum of another is removed. The ovum from, okay, now we have to look for it. They're saying that the nucleus from the somatic cell, meaning that, okay, they obtain the cell, yes, but now after obtaining the cell, in this statement, they're not talking about this. So meaning that they're starting from this. They're not talking about this. They're starting from 1, 2. Okay, let's go back. They are saying that the nucleus from the somatic cell of the donor is extracted, meaning that the nucleus is removed, is brought here. So this could be step 1. Okay, we shall see if it is the most correct. Step 1. So this is covered. They are saying that the nucleus from, yes, the nucleus from the somatic cell is inserted into the ovum. The nucleus, this is done. The nucleus is of the somatic cell is inserted into the ovum. Do we have the ovum? The nucleus from the ovum of another individual is removed. This is step 2. Yes, this should be step 2. Because I removed here the nucleus, so I remained with this. So now I inserted this into this. This should be step 3. The nucleus from the ovum of another cell is removed, is done. The ovum of the new cell is given, the nucleus from the somatic cell is inserted. Now this is step 3. So meaning that now I have 2, 4, 3. Then I'm remaining with this and this. So they are saying that the ovum is now, the ovum with a new nucleus is now given electric shock. Now we are here. We are giving it electric shock, meaning that now this becomes 4. Yes, and then now the embryo is implanted into, so meaning that I'm going to have, yes, it's going to be 2, yes, 4, yes, 3, 4, 1, meaning that 2, 4, 3, 5, 1. So this is the most correct answer. So the answer is A. Let's go to the next question. Then they are saying that 1.5, shrubs, these are short grasses, of the family, this family, they are saying that can be found in Australia, South America, Indochina, and parts of Africa, as shown in the map below. It is hypothesized that all continents were once one large continent called Pangaea, and that they separated due to continental drift. They said, okay, sharp. They're saying that this is evident. This evidence that the family, yes, this family, what about it? All belong to the same species. No, they don't belong to the same species. They don't belong. That's not an evidence to show that they belong to the same species. Number 2, they're saying that they are equal distributed in all continents. Who told you that? They are not equally distributed. Are you trying to mean that what is here and what is in South America and what is in Africa, they are equal? No. Wrong. They're saying become extinct. They're still existing. Arose from a common ancestor. Simple, because at first they were together, yes, but they went to different environment due to the separation of the continent. And then they developed into different species. That's why we say that they don't belong to the same species. Therefore, D is the most correct answer. 1.6. Yeah, these ones, they take some time, but when you reach there, we're going to be faster. The diagram below shows the DNA profile of a girl that her mother and 4 males are about the males. Okay, maybe they are claiming. Which male is a girl's biological father? I have to look for all the bands which correspond to the mother, because the mother is known. This goes with the mother. Done. This goes with the mother. This goes with the mother. Now, the remaining bands from the girl must be matching with the guy who is claiming. Okay, let's go. This one matches. Cool. Yes. This one matches. Is there any? Yeah, because this one is matching. So is there any other for the girl? No. So it means that this is number 3 is the biological father. All of them must be ticked for the case of the girl, but they must be the remaining one, which did not match with the mother, must match with the father. Hence, number 3 is the most correct answer, which is C. Okay, the following are the characteristics of group of animals. Yes, animals. If you're saying animals, just a moment, guys. Okay. Just a moment. Okay. Animals. They're saying that which characteristics of group of animal are able. which are the characteristics of group of animals able to interbreed, occupy the same habitat, produce infertile, belong to the same species. Which combination correctly represents a population? Oh, which combination represents a population? Yeah, it means that you can't know which group represents a population. Just a moment, guys. Let me increase here so that I can see clearly. Yes. Yes. Which population does the population is being shown by what? Is being shown by the able to interbreed, and they must produce a fertile offspring, occupy the same habitat. Yes, they must be able to interbreed, but interbreeding they must produce the fertile offspring. And the only word which can help us to bring about to produce a fertile offspring is one word. Which word is that? Same species. They belong to the same species. So the answer is B. Same habitat, same species. Yeah. So we go to the answer becomes 1, 2, 4. 1, 2, 4. Where is 1, 2, 4? Is there any 1, 2, 4? 1. 1, 2, 4. Yeah, it's B. Yes, it's B. So yeah, it gives me the correct answer. Let's go 1.8. Which diagram? Let me see. Which diagram? Not which diagram. During anaphase 2 of meiosis, the two chromatids of the chromosomes are pulled apart, each moving towards the opposite poles at a rate of 1 micrometer per second. This question? Okay. I see it as a new content to students. Anyway, but it's just an application question. Okay, they're saying that, yes, two chromatids of the chromosomes are pulled apart, each moving towards opposite poles at a rate, this is the key point, at a rate of 1 micrometer per second. Okay, each second, 1 second, it means a distance moved is 1 micrometer. Let me say 1 like that. The distance in micrometers between the chromatids after 20 seconds. Obviously, if it's 1 second, 1 micrometer, after 20 seconds, they'll have moved 20 micrometers. So that is a direct question. However much is just an application question. So the answer becomes B. Yes. So the answer there becomes B. Yes, let's go to the next question. The next question, they're saying that, which one of the following indicates the type of variation? I gave you examples yesterday. Anything which does not change since you were born is continuous variation. Anything which changes as you are growing up, it's continuous variation. For example, here they gave you height. Height changes, therefore is continuous. The way you were born and the way how you are now is not the same way. Height is continuous. So is where there is this continuous is, I think I have the answer now. Because the rest are wrong for the height. And it can't be correct for the next one. Okay, skin color. I don't think most of us, we are born yellow bones. Check us now. Hey, we are original indigenous. Yeah. Yes, color changes, skin color changes. You check the kind of people. Some people, I want the black one, I want the light skinned one, I don't know what you're gonna use for them for, but it changes among the individual. All right, this is continuous. Check. Earlobes, the way those earlobes are since we are born, unless you get an accident and then they change, but if not, it's the same thing. So correct. So let's not waste time there. Answer is B. Let's go to terminologies. I know sometimes the terminologies, they make these students a little bit of a problem with it, but I don't think these terminologies were that hard. Ne? Let's go. The first terminology, they are saying the type of inheritance which produce intermediate phenotype. Intermediate phenotype. Don't forget that we said it in the what? In the distinction material book, our distinction material book. Ne? Yeah, for those people who were not there. But okay, there we go. Intermediate. How do you call this? Incomplete. Incomplete dominance. Intermediate. Yes, we talked about it. So the answer here is incomplete. Complete dominancy. Yes, done. The point at which adjacent chromatids overlap during crossing over. Chiasma. Chiasma. Guys, spelling is mandatory. The process by which information is copied from DNA to RNA. Transcription. Told you. Transcription. Let's go to the next question. What are they saying? They're saying that sugar found in DNA. Sugar. Thymine. They're saying sugar, not base. Gone. Thymine. That's a base. They're saying sugar found in DNA. Ribose is found in RNA. So for the DNA, it's going to be deoxy. That's why he's saying DNA, deoxyribose nucleic acid. So the sugar found there is deoxyribose. The bond that occur between nitrogenous bases in DNA molecule. The bond that occur between nitrogenous bases in DNA molecule. So how do you call that bond? That bond is weak hydrogen bond. Yes, you must say weak hydrogen bonds. Most of the people, they wrote peptide bond. Peptide bond. In some cultures, they say that sometimes you can laugh because of sadness. Not because you are happy, but you are sad. The same way someone can cry because of happiness. When I laugh at because someone is because of the sadness. So this is weak hydrogen bond, not peptide bond. Yes, because I highlighted it that these are the mistakes students do. Yes, okay, let's go. Manipulation of organisms' gene to obtain the desirable characteristic. How do you call that? Manipulation of a gene to get a desirable characteristic. Whenever you talk about manipulating in science, whenever you say manipulating, what did I say? I said that is engineering. Engineering. Manipulating. But you are manipulating genes. So, what are you manipulating? Genes. Therefore, genetic. Simple. Genetic engineering. Guys, I told you, you don't need to cram. You don't need to cram. You just understand life science. Tell the people to join the channel. If you want a distinction, Thundereduc. Yeah. Use the distinction material. There is no way how you can fail a paper. Never. Yes. Okay. Unless you don't want to. All right. Oh, unless you found me. Or you found the channel late. But if you want a distinction, tell the whole world. Thundereduc. Okay. They're saying that representation showing the arrangement of diploid set of chromosomes. Hey. Representation. They are looking for how do we call that representation? Which shows the diploid set of chromosomes, meaning that chromosome number 1, chromosome number 2, chromosome number, but they are saying, see, diploid. Like that, we call it a karyotype. Karyotype. Done. Terminologies, out of 7. Okay, source of variation. How do you call random? We talked about this yesterday. Random mating, random fertilization. Source of variation for the species. I told you that a man produces over 500 million sperms. That is a rich man. That's why they tell you that you are 1 in a million. It means that your sperm was 1 in a million sperms which were released by your father and 2 there, and then only 1 you fertilize the ovum. So random fertilization is also random mating. You don't just find people come and say that, okay, now I want this one. You just go and then marry. You just say, I want a small one, I want a big one, I want the light skin, I want the dark skin. You see, you're not doing it randomly. So you are minimizing variation. So variation, it must be random mating. When you are chasing this one and then another one comes into your eyes, you just go with that random mating, like a chicken. Yes. So this is both A and B. The answer is not A and B. The answer is both A and B. I explained it yesterday. Both A and B. Organisms that possess two factors which separate so that each gamete contain only one of these factors. Law of dominance. There is no domination here. Law of independent assortment. Organisms possess two factors that which separate that each gamete. Law of independent assortment. This is done in dihybrid cross. We call this law of segregation. Law of segregation, not principle of independence. So the answer here is none. Because this is a law of segregation by Mendel. You remember that guy Mendel discovered the law of segregation in 19. He was there in 1865. Yes, Mendel, Georg Mendel, that priest. Yes, Georg Mendel. Yes. His life science is nice. So what you should do is be nice to it. Once you are nice to it, the subject is gonna be nice to you. If you are not nice to it, forget about. Okay, guys, just 1 second so that yes, Mr. Ramaphosa has given back electricity to us, so now we are happy. Yes, it's back. Yeah. All right. So if you see some disconnection a bit, just know that you're just switching the system. But we are continuing. Reproductive isolation. Reproductive isolation. Is my mouse? Okay. Reproductive isolating mechanism in species. You separate organisms through reproductive isolation, breeding in different seasons of the year. Correct. Infertile offspring. This is not correct. This is not correct, Baba. Look, same. This word, these people are confusing us, ne? Breeding in the same season of, no. It's supposed to be breeding at different seasons of the year. Let me show you what these people are trying to trick you up, ne? Let me just show you using the distinction material so that you see exactly how these people are trying to this is a real fake. They are faking us, so that we are off. We are sober. All right. Let me show you what they are showing you. Breeding, yeah, is here. You see, breeding at different times, not breeding at the same. So that answer is wrong. So this answer is wrong. You see, when you're saying the same, the answer is supposed to be breeding. This distinction material, there is no way you can go astray. Yes, so this is wrong. Infertile offspring. Correct. Check here in the book. Check here. Infertile of the way it is without even changing anything. Yes, infertile offspring. So you see, we say that mechanism of reproductive isolation. Now there are some people saying that I wish I knew this booklet. Sorry. Yeah. But you can refer to someone else so that you can get rewards. All right. So the answer here is B only. Yeah. Let's go to the next question. What does it say? They're saying that this is a phase. What I've seen in this paper, as I'm going, I don't know that much what is down, but what I've seen it here is they are using small words to take you off. If you don't read these questions very well, they're just using very small words just to take you off. All right, let's go. So we have to be keen, guys, when we are trying to answer. Yeah. So even may have to read too much so that I don't go astray. I might go astray. I give you a wrong answer. No, man. All right, let's go. They're saying that the diagram below represent a cell in early stage. You see these people. The word early. That word, there is early and late. Late stage, mid stage, early stage. So now they're saying early stage. Okay. So if you look at this, which stage is this? This is not interphase. This is early prophase. Chromosomes which are homologous, they align together. Homologous. Yes, crossing over is not yet there, but they have aligned together. And you see that these dots shows that the nuclear membrane is starting to disappear. The nucleolus is already disappeared. You see? These people. So this is prophase. Prophase. Which prophase? Prophase 1. If you don't say prophase 1 is 0. Number of chromatids. So how many chromosomes do you have? 1, 2, 3, 4, 5, 6. So you have 6 chromosomes, but each chromosome has 2 chromatids. So 6 times 2, which is 12. So the answer here is going to be 12. Number of homologous pairs. They are saying number of homologous pairs. They're not saying number of chromosomes. Pairs. So it means that 1 pair, 2 pairs, 3 pairs. 3 pairs. Someone is going to write 6. Ask you, do you have eyes? Yeah. Do you see 6 there? No. I see 3. Why did you write 6? I don't know. Okay, let's go to the next question. Identify what is R. Yeah. R is nuclear membrane. Nuclear membrane. What is S? S is cell membrane. Membrane. Yeah, you know, I told you last time that you guys, you confuse nuclear membrane and cell membrane. T. What is T? What is now T? T is nucleolus. Nucleoplasm, not nucleolus. Nucleoplasm. So, the nuclear, okay, maybe they will slash it with the nucleus. Nucleus, yes. But based on this is nucleoplasm or nucleus. So if you give nucleoplasm, yeah, they might give you. If you give nucleus, they might give you. Based on what they have shown us. Ne? Yeah, don't say that. No, me, I wrote nucleus. Yeah, you might be right. Ne? Yeah, correct. All right, let's go. 2 organelles in animal where meiosis takes place. They're asking your parts. The most important structure on you. Which structure is that? Testes. Testes. That's 1. Oh, if you wrote it like this is also correct. Because this is singular, this is plural. Okay? I'm not saying that you write 2. No, just write 1. Hey, students. That's number 1. Number 2 is ovary. So they give you a tick here, they give you a tick there. Simple. They're saying that, you know, they write these funny words of these plants. Okay. Who knows this? I don't know. Let me just quote. The plant carries edible fruit. Scientists have been studying the inheritance of these 2 genes. One for the stem texture, and then another one is for the fruit shape. Okay, sharp. Let's go. They're saying stem can be okay, smooth. Smooth is represented by a capital letter. And then prinkly means that they have those yes, small letter. And then the shape round is capital letter, elongated is small letter. The type of cross, dihybrid. Dihybrid. Dihybrid cross. They're saying state the dominant characteristic in the stem texture. Texture is here. Ne? Smooth. Answer here is smooth. Smooth. Boom. Done. This goal. Recessive. Recessive for what? Fruit shape. Fruit shape. Elongated. Elongated. Let's go. 1.5.3. Give there. The genotype. The genotype. It means that you are supposed to write in terms of gene. Prinkly, stem, and elongated. Prinkly is small letter. So I can't be with the heterozygous. Small n, and then elongated. Elongated is small r. Listen carefully. If you wrote NNRR is wrong. Ask me what? Why? Because of this space. I explained it yesterday. Yes, a space must not be there because this is a genotype. These are not sperms. It's a genotype. So it's supposed to be 1. Yes. They're saying that phenotype of plant with capital N is what? Because the small n would be masked. Capital N is smooth, and what? Capital R is what? Round, smooth and round, sharp. If you have the space there, is cool, but the genotype is supposed to be no space. Yeah, let me know if you have questions. If you have questions, if you have challenges, yes, I'll smash them. Yeah, I'll smash them. Yes. Okay, let's go. Let's continue to the next question. Which question? All right, they're saying that the table below shows the information selected on hominid fossils. Okay. All right, we are hearing. What are they saying? What are they telling us? They're saying that P. What is P now? P. Oh, the common name. Australopithecus sediba carabo. We say this is a transitional fossil, missing link between Homo habilis and the. Okay. Who discovered Dihybrid? I don't know why they like carabo too much. They brought it in 2017. Yeah. It was the second or third objective. Yeah. Is read the bagger. Lee. Beggar. Yeah. Like that. This is Sediba, Carabo. Yeah, you can and you can find that someone called Carabo is feeding this Carabo. All right. They're saying that Taum child or Taum if you're coming from northwest. Yes. How do we call? Remember I told you don't go the paper without knowing the 3 Australopithecus africanus species. I said Taum child or Taum child, little foot, Mrs. Place. What are they looking for here? They're saying this is Australopithecus africanus. Cool. Just saying A africanus, they would have given a tick. Yes, because there is no Adipithecus here. So A africanus. But they have already given you the spelling here, so there is no stress there. They're saying that who discovered? Who discovered the Taum child? Raymond Dart. Yeah, it's Raymond Dart. Raymond. I don't know whether you call it Raymond or Raymond. Raymond. Dart. Raymond Dart. Check that booklet, the distinction material. Check this distinction material. I organized it nicely for you guys. And even I created a video. Check. Australopithecus, Carabo, read the bagger, Malapa. And then they're looking for the 3 Australopithecus africanus species. Check bone. Raymond that. Taum. Sharp. Let's go. They're saying name the fossil. P. We have said it. P is Carabo. Then they're saying identify species R. We said Australopithecus africanus. Let's go. They're saying that give the name of the scientist who discovered Q. We say that read the bagger. Yes. Well, you can also give that one brief Ilof. Yes, Ilof. Where is he? Yeah, this one. So it depends on whom did you give. Yes. Then they're saying S, we say that it's Raymond Dart. Now, by this time, you have 50 marks out of 150. This one is enough for you to shift from a failure, meaning that you're above 30 now, 30%. However, it's not enough for my students like you. Guys, don't have too much pressure. like now I'm failing. That's life. Still doesn't mean that if you fail this, then life has stopped. No. All right, let's continue. It just make you to prepare, and I pray for you guys so that you pass this, so that you get enough gelete. All right, let's go. They're saying that X, what is X? That is amino acid. I told you guys that you must always label these things before you start. Amino acid. Simple. Let's go. X, Y. What is Y? Y is anticodon. Z, messenger RNA. Let's go. X, amino acid. Z, messenger RNA. Give the nitrogenous base of sequence, nitrogenous base sequence of DNA base triplet, complementary to complementary DNA base triplet, complementary to mid codon of Z. Oh, they're saying DNA, meaning that we have to convert it backward. Let's say this is 3, this is 3, this is 3. So we are looking for this. So, but we are converting it backward. You have A, U, G. So we're going to be, say A goes with T, U goes with A, G goes with C. So this is the answer. That's what they are looking for, because they're saying mid in the middle one and for DNA. Then they're saying that give the nitrogenous base of Y. Where is Y now? Oh, the anticodon. So meaning that they want you to change this into transfer RNA, meaning that A goes with the, remember A transfer is an RNA. RNA doesn't have T. Therefore, we're gonna say U. U goes with A, and then G goes with the C. So the answer is this for this question, and this for that question. Yes. Describe the process shown in the diagram above. Hey, what is this? This is translation. I told you guys. How many marks are this? Guys, don't tell me that you disappointed me unless you are not trusting me. Don't tell me that you disappointed me. There is someone who told me in the comment, I was so sad, and but I didn't stop. He told me that you lost me in the first 5 minutes. I knew that this one is looking for attention. Yeah, because I talk too much. Okay, I talk too much, but I wanted you to understand the concept. Yes. Anyway, what do we mark? We can have different ways how we can mark. But this one you are describing, name and describe. The process is called translation. That's one mark for the word naming. And then according to the messenger RNA, the transfer RNA with the complementary anticodon bring the required amino acid to the ribosome. The amino acid are joined together by the peptide bond to form a required protein. Simple. That's how I have been describing. But still in my book, the distinction material, I showed you exactly what you are supposed to do. Now the mouse is jamming. Okay, doesn't want me to this mouse doesn't want me to give you okay, let me use this. Okay, this mouse doesn't want me to give you answers, ne? Sorry. Yes, born is there. Each transfer RNA is a marking point. Here is a marking point. This is a marking point. Each transfer RNA carries a specific amino acid. We give you a tick there. When anticodon on the messenger matches the codon on transfer RNA, we give you a tick. Specific amino acid, we give you a tick. Matches the anticodon. Yes. Then you're saying that this brings the required amino acid. Yes. Where does it go? To the ribosome. The amino acids are joined together or attached by peptide bond. Guys, we call them peptide bonds. We don't call them polypeptide bond. We call them peptide bond. I was very clear when I was explaining this. And then to form a required protein. That simple, short as that. But you can find a person writing a full page. So you're okay. That's short as that. That's what we wanted. Let's go. So this description thing, I'm not going to waste too much time because it is open thing, and you can find them in any book. Yes, you can find them in any book. Yes. Let's go to the next question. What are they saying? Just a moment. Yes. What are they saying? They're saying that the table below shows the codons. I explained this. Guys, take note of this because I explained it and I said this is one of the confusing subtopics. The table below shows the codons of codons that code for some amino acids. Okay, sharp. A mutation caused by DNA triplet to change this. Okay, so it means that we changed this to this. But they're saying this is DNA. But what is here is messenger. Meaning that to read this, I have to change it first. So I'm going to say that, okay, this was C. C goes with G. T goes with A. And then G goes with C. But change it to C goes with G. T goes with A. And A goes with U. Because this is messenger RNA. Wabona? Yes. Now I come. I go to, you go to what? I go to this table. I look for it. GSC. 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 GSC is aspartic acid. What about this? Does it correspond to a different amino acid or the same amino acid? Let's go. And then you're saying that GAU, aspartic acid again. Wabona? Ntuli. I can call you Ntuli. Yes? Isn't it? I'm just saying Ntuli because Ntuli called me and said that, thank you very much, sir, for teaching me. Yeah, I think I managed to pass. I pray for you guys, so that you pass. Sometimes when you're marking you're marking your students, and then you get something, you think of something, you end up giving a wrong one correct. You never know. Make sure that you pray until the results come back. Yes. Anyway, so since they code for the same amino acid, yes, so it means that the protein will not change. But these are 4 marks. Where am I going to get the 4 marks? You're gonna say that this change of this, yes, this changed into this. You writing it GAC changed to GAU. Those are marks. Those are 2 marks. This changed to this. Why add? Why this one is a mark and this is a mark? Is because you changed it. You see, you changed this. That's 1 mark, and you changed this. Those are 2 marks. And then this code for the same. This code is for aspartic acid as well as this code for aspartic acid. Meaning that these two, they code for the same amino acid. Is another tick. Those are 3 marks. Then the 3rd mark, the 4th mark will say that this will not change the protein formed, or it will not change the functioning of the protein formed. Simple. Why? Because it's like calling you with the first name, calling you with the second name. You are the same person. Yes. Or sleeping on your bed when you are straight and sleeping on your bed when you have carved somewhere, or does not mean that you're a different person. It's the same person. So they're just saying that, yes, however much the mutation occurred, but the mutation did not change the amino acid. Simple. This question, they have brought it back again. Tabulate 2 difference. I emphasized this question of mutation, and I told you that I know what students find when it comes to this question of mutation. Anyway, this question. Tabulate two differences between DNA nucleotide and RNA. I explained it before. Yes, those people who were on my channel yesterday. They are they are not asking you the differences between DNA and RNA. No. They are saying DNA nucleotide and RNA nucleotide. Clear? So there are two differences. The first one, DNA nucleotide, has thymine. And the RNA nucleotide has uracil. The second one, DNA is made up of, nucleotide is made up of deoxyribose sugar. And RNA nucleotide is made up of ribose sugar. There is no any other difference. They are looking for the nucleotide. They are not looking for DNA. They are not looking for they are not looking for just those nucleotide for the RNA and for DNA. That's all. So if you wrote a lot of things there, but there are 5 marks. Where is the fourth mark? Fourth mark is for the table. Just drawing a table and say DNA nucleotide, nucleotide. Even if you didn't write anything here, you draw a table, a complete table, you will get a mark. At least you are not getting 0. Yeah, at least. Yes. Let's go. Down syndrome. I talked about it also. Down syndrome. Okay, let's go to Down syndrome. What are they saying about Down syndrome? Okay, let's go. Down syndrome is the result of individual having extra chromosome on chromosome number 21. Okay. The genetic variation that can be caused by Down syndrome are, okay, trisomy 21, all somatic cells are individual, 3 copies of, okay, chromosome number, all. This word is a key point. And then they're saying that mosaic means something which is moving. Yeah, okay. Individuals have only some cells, not all. So this is a key point versus this extra chromosome, which is caused by abnormal process of cell division after. This is after, is caused by after fertilization, but what occurs during gamete formation. This is during gamete formation. This is after fertilization. You see? Yeah. That's the major difference. The type of mutation lead to trisomy 21. The type of mutation that leads. You can say chromosomal aberration. Those who wrote chromosomal aberration, correct? And then chromosomal mutation. Because these are chromosomes. Chromosomal mutation. Yes. Or aberration, correct. Abnormal process during gamete production that lead to 3 copies of abnormal process that lead, name the abnormal process that lead to the 3 copies of non-disjunction. The type of cell division that occurs after fertilization, it is mitosis. The saying, describe how the process mentioned above, which above B here, non-disjunction lead to trisomy 21, is 5 marks. This is a general question. Yes, go back to my distinction material so that we don't waste too much time there. Yes, I talked about it, but sometimes people they have challenge. But before I left meiosis, I said that please don't go without knowing this process, because this process, they will ask you. Here is a question. Non-disjunction, failure of the chromosome to separate is they are looking for this. You see, this page. Each nucleus has it contains 23 chromosomes after meiosis, but non-disjunction when occurs, okay, one nucleus will contain 22 while the other will contain 24. Yes, either of the resulting gamete will fuse with the normal gamete, but they said yes, with the normal gamete. They said yes, they said what will happen. Yes, so the normal gamete is 23, then the resulting, the 20, if the 24 fuses with 23, it's going to be 47. The 22 fuses with the 23, it's going to be 45. Yes. So in this case, they are looking for 23 because they're saying trisomy 21. It means that the 21, it has an extra chromosome, not short of a chromosome. Yes, let me explain to you. Yes, and then they are saying that, let me show you what they are trying to say here. Yes. And remember, during non-disjunction, this is a cell, né? Under metaphase. Yes. So this is what happens. So during non-disjunction, the whole set is going to go to one side. So meaning that I'm going to have 2. So the normal one is going to be 2 cells. I'm going to have 2 here and 2 here. But during non-disjunction, the whole of it is pulled to one side. And then this one, yes, it will go to one side. And then this one, another one will go to one side. Meaning that here, yes, here I will have 1, 2, 1, 2, plus this one, 3. And then this side, I'm going to have only 1. So this one has extra chromosome. 2, 3 instead of 2. And then here it has short. So they are looking for this. Because they are saying, yes, if it occurs on chromosome number 21, it becomes trisomy 21. Tri means 3. 1, 2, 3. You understand? So now, when this one fertilizes a normal ovum, then it's going to have, now the cell is supposed to have 23 chromosome. But this one is not going to have 23 chromosome. It's going to have 24. So if this fertilizes a normal ovum, which is 23, it's going to result in 47 instead of 46. So that's what they are saying here. So go to the distinction material. Yes, get there. So that's what they are trying to explain, or they're asking you here in this question. Let's go to the next question so that, yes, this describe 2 differences between trisomy 21 and the mosaic Down syndrome. Yeah, I think I talked about it. Trisomy 21, all somatic cells in the individual have 3 copies of trisomy of chromosome 21, you see, all. But here, yes, make sure you draw a table. I think they don't want a table. They're not saying tabulate. While here in mosaic, the individual has only some cells with extra chromosome 21. But here, all the somatic cells. The number 2, the process, when does it occur? It occurs during gamete formation. While here, it occurs during or after fertilization. Those are the 2 differences they are looking for. It's just as something from there which you can use to get the answer. All right, I see ourselves trying to finish soon. Yeah, so we are going to the question number 3. How is the paper? Let me know. How is the paper? Let's go to the next question. Yeah, I think the paper was simple. The paper was simple for those who read. Yeah, let me say like that. And I think I should come back and I teach you how to read. It does not stop only here, even when you go to the university. Good. All right. The diagram below shows the inheritance of type. Or if they like this funny name. Anyway, I'm not going to remove my teeth because of these names. Okay. Is a rare disease which lead to this destruction of the neuron. It is inherited as a autosomal. Okay, autosomal means that it occurs in the body cells, meaning that it's non-sex linked. It's occurring in these other cells, these other chromosomes, not on chromosome number 23. Yes. Okay, TT. So meaning that you must not use X and Y. This is not sex linked. It's autosomal disorder. Anyway, let's go. Describe what is meant by is a disorder which is caused by a gene carried on non-sex chromosomes. Yes, non-sex chromosomes, meaning that from chromosome pair number 1 to chromosome pair number 22. It must not be sex. Yes. How many sons do individual 1 and 2 have? 1 and 2. Yes, sons. Son is a box. It's 1, is 2. But this son is not belonging to these 2. You understand? This guy, yes, just married the daughter of these 2 guys. Meaning that the answer is 1. You understand? You see? Yes. This is just an intruder who came to marry the daughter. Yeah, the beautiful daughter of 1 and 2. Yes. So the answer here is 1. Using the individual 3, 4, 5, explain why it can be concluded that this disorder is controlled by a recessive allele. 1 is 3, 4, 7. 3, 4, 7. Okay, let me clear up here so that at least yes. Okay, let's see. 3. Okay, this is the affected individual, né? And these 2 people who are, if they produce a person who is affected, it means that these 2 people were heterozygous. Meaning that it is capital T, small t, capital T, small t. You see? Since they carried that what? Since they carried that gene, this gene, recessive allele, and then this recessive allele and this recessive allele gave rise to this. Meaning that, what is the question? What are they saying? Which question is this? 2.5.3. They're saying that it means that individual 3 and individual 4, they are both normal individuals. I see. That's the first mark. But they gave rise to individual 7 who is affected with the disease. You have to tell us the disease, né? A female who is affected with the disease. Yes, that's the second mark. This means that individual 3 and 4, they were heterozygous. Meaning that they are heterozygous because they are both normal, but they gave someone who is sick. Meaning that they were heterozygous, they were carriers. That's another mark. And then they pass their recessive allele. 1 recessive allele from 3 and 1 recessive allele from 4 to individual 7. That's another mark. You're able to get all the 5 marks. 1 recessive allele from 3, 1 mark. 1 recessive allele from 4, another mark. Yes, to form this one who is affected with the disease. So, yeah, it is an application question, whereby you have to apply and explain exactly what happened to inform individual 7. Then they are saying that individual 1 and 2 can produce children with 3 possible genotypes. Okay, list those 3. Individual 1 and 2 can produce. Yes, individual 1. 1 and 2 is the same thing. 3 and 4, they are both heterozygous. Okay, they can produce. If they are both heterozygous, it means that, look, it means that this is capital T, small t, heterozygous, capital T, small t. So it means that when they produce, they're going to produce capital T. This is what they are trying to show you. Capital T, small t. This is capital T, small t. And then small t, small t. You check these 2. They are both heterozygous. This is homozygous and this is a homozygous. Let's go. The question. They're saying that list all the possible genotypes that have 25% chance of offspring. Then this is 50 and this is 25. This is 25. Meaning that I'm going to come here and say capital T, small t, small t. A comma. That's all. That's what they're looking for. They give you ticks. In human, hemophilia is caused by a recessive allele of X. Of X. Anyway, it's caused by a recessive allele of X. Let's go. Of X. They're saying that in humans, hemophilia is caused by a recessive allele of X. Okay. This is carried on X chromosome, meaning that this is sex-linked. The moment you say that that's sex-linked, Hemophilia, yes, we know is a sex-linked disorder. A woman who is heterozygous for hemophilia marries a man who is heterozygous for hemophilia. That's a woman. It means that it's like this, capital H, small h. Yes, marries a man who is hemophilic. Yes, meaning that the man is definitely is like that. Is use the genetic cross to show the percentage chance of couple having having a daughter who is homozygous for normal. Homozygous for normal and is a daughter, meaning that this person is gonna be like this. There's no way. Check, the father is not normal. Then it means that the answer is gonna be 0. 0. Yes, but they are saying use the genetic cross to show the percentage chance. The percentage chance is 0% because there is no way. But they are saying use the genetic cross to show. So you're saying 0% is 1 mark, but you have to show. Don't forget to show all the steps in a genetic cross. Let me see, is it P1? Let me see. A man who is, yeah, they didn't talk about anything. So I'm going to come here and say P1, F1. P1, parent, phenotype. You go to the question, you'll get the phenotypes there. I told you that this one is simple, and a man who is heterozygous, a woman who is heterozygous, meaning that this is a normal woman. Yes, marries a man who is hemophilic and a man who is affected. Then you go to genotype. Genotype, I'm just rushing so that a lady was like this because they say that heterozygous, marrying a man who is like this. Yalla, let's go. They're saying like that. Don't forget that these are gametes. I always tell you that they are not at the beach, so they must be circled. If you didn't circle, it's still fine. Don't stress. Give you a tick. These are gametes. Wabona. Yeah, but how do you call this process? Meiosis. You saying P1, F1, we give you a tick. So that's the second tick. 0% is a tick, but it's a second tick. This is a third tick because we got it. So you have 4 marks. Mahala. The question is out of how many? 7. So we are searching for the 3. This is the next tick. It's the next tick. Then after that, you can draw your Punnett square here, or you can just cross. When you cross, you'll find out that this, look critically. This goes with this. So it's going to be X, capital H, X, small h. Then this goes with this, capital H, small, capital H, yes. And then this with this, small h. This with this, small h, and then y. Is there any probability of having a child who is homozygous normal and is a female? No. There is no way. Therefore, this is offsprings. F1 offsprings. And then you remember you have to cross, ne? Yeah, show cross or the Punnett square. So, and this process is called fertilization. So we give you another mark for this. But what I know that the marks are more than what we stipulate. But you saying that therefore the percentage chance is 0%. This is going to be a compulsory mark because the question is emphasizing that percentage chance. They want a percentage chance. Do you understand? Yes. So it's 0% having a child who is normal, homozygous, and is a female. Yeah. Let's go to the next question. I think I'm clear. Yes. Let's go to the next question. These papers are simple if you read. If you don't read, yeah, they become sour. But I don't expect you to. Let's go to the next question. The next question. Question number 3. It means that now you have 100 marks. 100 marks. See now, you are approaching, ne? Slowly, ne? A distinction. You're approaching a distinction. I know some of you just made small mistakes, which can be acceptable. but there are some mistakes which cannot be accepted. So yeah, anyway, let's go. Yeah, let's go. This is year 2023. Yeah, I know some of you gonna be successful. I pray for you so that you become successful. Anyway, learners conducted an investigation to determine the blood group was the most common in the community. I talked about a pie chart yesterday. Yeah, they conducted information about, okay, 200. It's a big number, né? It means that the sample size. Take note of that. In each of the 3 types of blood group. Okay. Yalla, let's go. They're saying that in their committee, they did not include the first donor in the investigation. Why? Comparison of results. Okay. Yeah. The pie chart below shows, all right, let's go. The percentage of each blood group in the sample. I know you read all these questions, né? You know them. So I'm not going through too much. Né? State the aim of the investigation. Ooh. The aim must start with the word to and is always put in the first statement or second statement. That's why always I tell my students, if you can't find it, then you have to figure it out. Learners conducted an investigation to, that is the aim. From to, the investigation. The aim is to determine which blood group was most common in their community. Yes, in the learners' community. Conduct investigation. Yes. So the aim is done. Answer the following question. State 3 planning state. We always ask you this, but you go back and read the question and see the possibilities for you. What are you going to plan? What are you going to do? So, but I need these people who are going to be giving me blood. So you have to organize them. Yes, get those 200 people. Yes, you are not just going to just get them. Yes, you have to get permission because this is a community thing. Get permission where you're going to do this, or the clinic, or try to go inform them so that they know and they accept. You are not going to force them. Get permission so that they can do that. You are not going to just collect the blood just like that. Get the necessary equipment. Planning before you do the activity. Yeah, that's what they are looking at. Yes, something like that. State one way in which learners ensure. Yesterday I told you the word language and I emphasized it. If they ask you, don't ask you, how did this person ensure, how did the person do increase reliability? Or how do you ensure reliability? Now they say that, how did the learner ensured? That is a past tense. Your answer must be in a past tense. The learner ensured reliability by using two. Remember, reliability is to increase the sample size by using a big sample size by using 200 people. 200 people. You see? So, this learner used the 200 people. Sharp. So, that is done. Give the reason why give one reason why they did not include the, yes, we said comparing the result. Calculate the percentage, the number of participants that had blood group B. Show your working. Blood group B. They use 200, but only 15% blood group B. So you just come here and say 15 out of 100 times 200. So the answer you get is the answer, around 30%. Done. They give you your 3 marks. Give you a mark, give you a mark, give you a tick. Done. They're saying that name the blood group that has only recessive allele. Only recessive allele, blood group O. Remember, it is recessive allele. Remember? Yes, I said it yesterday. Results of codominance is AB. I said it yesterday. Remember, it's A and B. Give the possible genotype of the blood group represented by 25%. 25%. Where's 25%? Is A. But they're saying the possible genotypes. If you remember, A is either homozygous. Go to the distinction material. You will see what I was saying yesterday. A, heterozygous. I explained this yesterday. I explained it very well. very well. Yes, I hope you guys did not disappoint me. Ne? I don't want the marks to come back and then I'm disappointed. Say, oh, why did I do this for my learners, these learners of mine? Ne? No. I don't want that. Ne? I want everyone to pass. Ne? Yeah, I want everybody to pass. Anyway, let's go. Question 3.2. We are almost done. We are almost done. Anyway, question 3.2. These are dogs, man. Do you like dogs? Let me know the question in the comment below. Do you like dogs? Do you have a dog at home? Sometimes they call you a dog. And a dog. Okay. Dogs have been selected and bred over many years to produce approximately 340 different dogs breeds. They have been bred for certain characteristics desirable to humans, saying that analysis of 786 okay, gray wolves, only direct ancestor, present day dogs bred. Okay, all the dogs belong to, I'm just reading so that I get the idea, ne? But you know you can read it for yourself. Okay, I've got the idea, ne? Very fast. They're saying that name the process whereby the different breeds of dogs were produced, artificial selection. Yes, these are the things we are talking about here. Where am I? Yes, artificial selection. Is it? Yes. Where is it? Okay, this is meiosis, ne? Yeah, artificial selection is they are looking for you. These dogs, even this question has these dogs we are talking about. Exactly the same dogs. Don't disappoint me. Don't disappoint me. Don't disappoint me, guys. Dogs, wolves. They're there. Born. Check. They are dogs, the wolves. That's what are the wolves we are talking about. Okay, they're saying that name the artist. What did we say? Is what? Artificial selection. Human, they select desirable traits. You see, that's what they said here. Desirable, they're desirable to humans. Yeah, yes. Distinction material. Get it? Yeah, get it. I'm talking about those who, yeah, your sisters, your brothers. Yes. Anyway, let's go. And we're saying that name the process whereby different breed of dogs were produced. Artificial selection. Describe how human carried out the process named above. Meaning that they got the characteristics they wanted. They let that individual which has that characteristic to mate with the other characteristics they want, and then the other one which doesn't have the characteristics were not allowed to give to have sex, meaning that to pass their genes to the next generation. So after many generations, they had to be with that desirable characteristic. That's what they are saying. So if you look at this example, check, it was about milk. Choose or select the cow in your herd, or now they're talking about the dogs to produce which has that characteristic. Yes. Then they are saying that let only those dogs to reproduce, the one which have the specific characteristics, then select the offspring that produce that characteristic most. Yes, let those only those offspring reproduce, allow them to have sex and have offspring like that. Keep repeating this process of selection and then the breeding until you achieve your goal. That's it, to get the desirable dog you want. Explain how it can be confirmed that all the dogs breeds belong to the same species. Same species? It means that they can interbreed to produce a fertile offspring. If they are not producing fertile offspring, then they don't belong to the same species. Yes, they must produce to produce a fertile offspring. Hence, this answer becomes right. Yes. Explain how present-day dogs may have been disadvantaged. Yes, in the relation to their common ancestor. This common ancestor wolf, Baba, it could resist diseases. Now, you have to take it to the how do they call it, the veterinary doctor. They were staying in the wild, so these dogs are susceptible to diseases. Less chance of these dogs being adapted to the environment. Yes, these dogs, the one which are new, yes, they have less chance. This one, it could adapt, Baba. Yes, meaning that some genes are being lost as you are doing this selection. Yes. So the dogs are being disadvantaged. Yes. Let's go to the last question. Describe the process of speciation by geographical. Geographical barrier. Guys, I told you don't go to the paper. Speciation is here. This is what they're looking exactly. This formation of new species. Exactly. There is a population of a particular species. A population of a particular species becomes split into two by geographical barrier. As a result of, you don't talk about the geographical barrier. Okay, you can say river, mountain, or whatever, but geographical barrier. As a result of two parts of the population cannot interbreed. There is no gene flow. Yes, between the two. The natural selection will occur independently. Yes, due to different environmental conditions. As a result, the population became genotypical. You start with genotypically, then phenotypically different over time. Even if you remove you bring these two populations, you mix them together, meaning that you remove the barrier and you let them mix and they interbreed. They cannot produce a fertile offspring. They cannot produce a fertile offspring. Therefore, one or both population have become new species. This is exactly without changing any word or what. That's what you're supposed to write. That's what you are supposed to write. Someone is gonna tell me that what if I used a scenario? That's not KO because they didn't give you a scenario. KO is when the examiner sees, just give you a tick, not arguing with him or herself. Yeah, should I give no? Something like that. Don't like it. Just when examiner sees your paper, is marking it as you are. You know, when you're marking someone, you see that someone is getting all the ticks. Yeah. You feel good, Baba. You feel good. You feel good. The same way you feel good when your marks are coming back and you have distinction. How do you feel? You feel like you are not fitting on the when marks are nice. All right, let's go, man. You feel good the same way I'm feeling good when I'm teaching you. Yeah, I feel good, ne? I feel good. All right, let's go. They're saying that. Name the type of diagram represented above. Okay, the diagram will represent the evolution of the family Hominidae. Okay, sharp. They're saying that. Name the diagram. I showed you these things. Phylogenetic tree, and I showed you the questions you're supposed to answer, and I don't think that they skipped those questions here. Let's go. Name the diagram shown above. Phylogenetic tree. Done. Give the letter of organisms, which is the common ancestor for all the hominids. P, because all of them, they come from here. Is P. They're saying that. They're saying that. What is common? Okay, share most recent common ancestor with the gorilla. Gorilla is here. Gorilla is there, but they share a common ancestor. What shares the common ancestor is here, meaning that is this. But what shared is R, because gorilla and R, they share a common ancestor. So the answer here is gonna be R. Give two organisms that have S as an ancestor. Two organisms that have S. Where's S now? S is this. Is this point. Stop it. Stop it when technology. Okay, this is S. So meaning that you're gonna say this and this. Those are the two individuals. Let's go. Name two organisms that have S as a common ancestor. We have said it. Quadrupedal. These ones. Gorillas. You know them. Is chimpanzee quadrupedal? I think so sometimes. But these ones, they are quadrupedals. Okay? They're saying that Describe 3 anatomical features of the skeleton of a quadrupedal organisms. Skeleton, when you talk about a skeleton, you're gonna talk about the spine, the cranium, because that's a skeleton, the spine, the cranium, and then the pelvic girdle. Yes, I showed you here when talking about. Yeah, for the case of the skeleton, here, you see, it's a skeleton they're talking about. They're saying about quadrupedal organisms. Foramen magnum is in forward position. Yes, it's a skeleton. Yeah, that. Yes. We talk about the spine is C-shaped, and then long and narrow pelvis. That's what they are looking for. Simple. Simple. As simple as eating ice cream. No bones. Yeah, let's go. Second last. Yeah. Fossil evidence is used to support out of Africa hypothesis. State out of Africa hypothesis. Modern humans originated from Africa and migrated the rest of the world. I told you when I was concluding, don't go to the paper without knowing this. Modern humans originated from Africa and migrated the rest of the world. And I showed you this. That take note of this. Unless you do not want to trust me. The modern humans originated from Africa and migrated the rest of the world. That's what they want. The word modern must be there. Modern. Don't just say humans migrated the rest from Africa. And Homo habilis, Homo naledi, Homo neanderthalensis, all those are humans. But the modern ones, Homo sapiens. Yes, they're looking for. Okay, let's go. Last question. Describe how the fossil evidence is used to support out of Africa hypothesis. How is this fossil evidence? How is this fossil evidence used to show out of Africa hypothesis, guys? How is it used? Describe how this fossil evidence is used to support out of Africa hypothesis. You guys, I told you this and then you didn't write. Okay. You talk about all the Adipithecus, Australopithecus, Homo habilis. You say that only, the word only must be there. Only. Adipithecus, fossils of Adipithecus, fossils of Australopithecus. You can talk about the examples. Fossils of Homo habilis. Only found in Africa. Then when you go to Homo erectus, you talk about the oldest fossil of Homo erectus, distinction material, and the Homo sapien. Homo erectus and Homo sapiens. Here you talk about oldest fossil of Homo erectus and Homo sapiens were found in Africa. Don't say that only found in Africa. Nope. You have to say oldest. The key point there is oldest. That's why I put it in bold italic and I was supposed to underline it so that your eyes can open when you are reading it. I think, guys, the paper was easy. Yeah. Don't forget to subscribe, like, and share. It has been Saidi, has been taking you all over this paper. I think, yeah, the paper is a little bit easy. It was a little bit easy. Okay, they had some challenging questions, but it was not that bad. It was not that bad. Yes. Thank you very much for being with us. Yeah. Thank you very much, guys. It has been a wonderful paper. Don't you think so? Let me know in the comment below to know how was this paper to you, based on you. How was the paper? Yeah, let me know in the question in the comment. Thank you for being with me from the beginning until guys. Yeah, I just love you so much. Stay blessed. Stay blessed. Yeah. Maybe I just a small note that even physical science, yeah, even other papers we will be doing discussing them immediately after you have done them. Thank you very much. Ne? See you. Very much, guys, for those who have been supporting us. Thank you very much for those who tried to be on our channel, who shared our content. I really appreciate, or we appreciate for that. Natasha, please don't be scared. Everything has the beginning and has the end, and this is today's end for this life science. Let's start, guys, without wasting time. Are you ready? All right. All right, guys. All right, guys. Don't be scared, ne? All right, guys. Hey, question 1. Various options should be provided. I don't read these instructions because everyone knows that. The base pairing in DNA was discovered. Base pairing. Don't forget that. Here they are talking about that DNA is made up of complementary base pairs. Who discovered that? When I was teaching this, I tried to show you exactly what they said, and I talked about this, that they would just try to mix up these names. So basically, it is James Watson and Francis Crick. So number 1 is going to be the best answer. I'm not going to go to the distinction material all the time, unless there is a challenging question which talk about maybe you don't understand something, and then that's when I'm gonna talk about the distinction material. So this is what you're talking about: James Watson and Francis Crick. Yes, so basically it is Crick and Watson. So number 1 is yes, the gene code for production of the gene code for the production of what? For the production of protein, DNA code of life. I was explaining that the DNA code of life, that DNA code for life because it codes for proteins. So the answer, the best answer here is D. I hope you guys, you can hear me very well. No echo, no what, everything is right. If it's not right, let me know. And then they're saying that which one of the following is a characteristic? Which one of the following is a characteristic of stem cells? We talked about stem cells yesterday. And then we say that stem cells have ability to change into other organs. So let's look at that point. They are easily obtained from any other organ, not any other from any organ. No. They're saying that because we saw the sources, division divided by meiosis, no. They are haploid, no. Then they're saying that they can be stimulated to form any other type of cell needed. Correct. So basically it is D. Then someone is saying that the chance of having female in a human, guys, the chance of having a female in a human is always the same. Boys, girls, the same thing, 50%. If you want to do the calculation, we can do the calculations, or you go the distinction material. So basically this one becomes B. You see, now I'm thinking about something and then I'm writing something. Okay, B. Which one of the following is a part of reason why color blindness is more common in males? We talked about this yesterday, and then we said that the allele for colorblind is recessive and located on X chromosome. Yes, don't forget we said that males have one X. If a recessive allele appears on it, it cannot be masked. So the individual is already sick or affected with that disorder. While females have two X chromosome, if a recessive allele appears on one, can be masked by a dominant allele. Therefore, it is very common in males, because the moment it is there, definitely the person is sick. Colorblind is made of two copies of no in male, because Y doesn't carry, so it's out. The allele for colorblindness is recessive, located on Y chromosome. Never, Y doesn't carry. They're saying the father pass the alleles of colorblind to their sons. The allele, no, remember the father give them the Y, doesn't give them X. So this is out. So the most correct answer is A. I think you are counting your marks, né? You're counting your marks. We are doing for the distinction. I think our prediction was 95% correct, né? Yeah. So I expect you to get 95%, because that's what we talked about, that our prediction is always 95% correct, né? Yes. So make sure that you count until you get 95%. Okay. We talked about this yesterday, and I showed you how exactly you're supposed to indicate it. DNA profile of 4 individuals are given below. Okay, sharp. Let's look at it. Saying that which one of the individual is possible members of the same family. So it means that they have to be with something which is common almost throughout. Okay, first one is correct. First one is correct. Correct. Second one, no. Correct. Third one, correct. No. Correct. Fourth one, correct. The fifth one, correct. No. Correct. No. Correct. Correct. No. Correct. I think now you can identify. Correct. No. They're saying correct. I see. Then this one, no. Okay, if you say I take this, no, nothing is there. All right, let's see. This one is 1 2 3 4 5. 1 2 3 4 5 6 7 8 9. You see, 9. 1 2 3 4 5 6 7 8 9. You see, so it means that this has 9 bars which are similar to this, So the similarity here is only 5. Therefore, we can conclude by saying that it is W, Y, and Z are family members. Do we have W, Z, and Y? W, Y, and Z. So yeah, so C becomes the most correct answer. Kill. When two plants homozygous for the characteristics are crossed, expected ratio is, remember, for example, capital T, small t. So when you talk about that, it's capital T. They're saying phenotype. This with this, it's going to be capital T, small t. This with that, capital T, small t. This with that, small t. Therefore, these are tall, for example. Tall, 3 of them, and then 1 short, for example. So the ratio is 3 to 1. We look for it. So A becomes the most correct answer. Then we go to the next question. Someone said that in Thunder we trust. I think, guys, Thundereduc we trust. Yeah, if you want a distinction, tell your friends, tell your brothers. Next year they must join, subscribe, so that they don't miss out the everyday classes we release on this channel, so that we help everyone. Subjects which we have not tackled this year, for example, agricultural science. Next year it will be here full. Fortunate enough, I'm the teacher for the agricultural science, so we will be dealing with that subject also. Thank you. Let's continue. 1.1.8. The diagram below represents the structure of chromosome at different stage of meiotic cell division. Which one of the following chromosome would be found in the cell during late? They're saying about late and anaphase 2. So remember that in anaphase 2, you expect the chromatids. The chromatids are supposed to be what? They are the one which are moving to opposite poles. Yes, but because they are late, it means that they could be straight. So we are looking for chromatids. Therefore, the answer becomes S. Therefore, we look for it, so it is C. So it is 2 marks there. Why not this? Because they are not chromatids. Why not this? Homologous. Why not this? Homologous. So it couldn't be the what? The answer. So they're saying that scientists who discovered Littlefoot. I talked about this yesterday, and then we said that the person who discovered it must be starting. Remember this all the Australopithecus africanus species. The people who discovered them, they start with R. So we say that it must be among these ones. So it's not really the bagger, no. So now it is Ronald, yes. Littlefoot, actually it's Ronald. The one who has the L, Littlefoot, Ronald Clark, or Ron Clark is the same thing. Ron J. Clark, whatever. It depends on what they have given you, but here they have given you Ron Clark, but actually it is Ronald J. Clark. All right, so C becomes the most correct answer. I hope you guys, you made it to 18 out of 18. Those who made it, congratulations. Yes, so we say that thank you very much for bringing about that. All right, so let's go to the next question, which is terminologies. I think let's talk about terminologies and then we see. Then they're saying that position of a gene on a chromosome, we talked about this yesterday, locus. The type of evolution where there is characterized by a long period of change and little period of no change. We talked about this yesterday. Punctuated equilibrium. Yes, punctuated equilibrium. Yes, they're saying that the natural shape of DNA. Yes, we talked it about yesterday, and we say that is double helix. And we emphasize that, please don't say double helical. No, it's double helix, nothing else. Type of bond found in amino acids, we talked about it yesterday. We said it's peptide bond, and we said that don't say polypeptide bond because we cross it. Peptide bond. Yes, so we talked about it yesterday. Then you're saying that the type of version shared by but these people, they just did what we did yesterday. But thank you very much at least, because that was our prediction yesterday that these things are coming. Make sure that you go through them. All right. They're saying that the type of vision shared by primates and allowed deep perception is stereoscopic vision. Vision. Someone can ask me, what about binocular? I hope so. They will also credit about binocular, because stereoscopic and binocular, they mean almost the same thing. But here, because they talked about deep perception, then you have to talk about stereoscopic. But let me just show you in the distinction material what I'm trying to say, that they could credit you. However much, this book is not written by the department, it's written by Saidi. So it's the same thing. I hope, let me check here, not yet there. Let me just go through here. I'm sorry about your eyes if it is hurting your eyes. I just want to show you something here. Yes, here, yeah, 2 minutes, 2 seconds, 2 seconds, 1 second, and boom, we are here. Saying that the eyes in front, stereoscopic vision. This is what we are talking about, because they didn't say that eyes in front. So the word binocular might be a little bit of not being credited, but stereoscopic, definitely it will be credited. But if they go under memo discussion, they could credit it, because they are talking about deep perception. So I think those who wrote binocular, you are halfway. But those who wrote stereoscopic, 100% correct. Because they didn't just say eyes in front, they talked about the perception of, yes. All right, the type of however, binocular also brings about that deep perception. So Yes, someone is saying that you wrote binocular. Don't be worried. So you could be credited about that. Ne? Yes. The type of dominancy which result in intermediate. When we talk about the intermediate phenotype in heterozygous condition, here we are talking about intermediate. The moment you talk about intermediate, you are talking about incomplete dominancy. Remember, you form, for example, capital R and capital W, meaning that if you have maybe red and white, you produce maybe gray, whatever. Yes, an intermediate, not red and not white. So that's what they are talking about. But because it is in this form, therefore it is in heterozygous state, different letters. We go to the next question. Just a moment. Just wanna check here something. All right. Yeah. All right. Okay. We are writing. Someone is saying that wrote codominancy. The reason why they might not give you codominancy is because they say the intermediate. And there on our distinction material, let me just clarify for you, so that because also this one could be a disturbing factor, that people could mix it with the codominancy, and the supposed to be at the beginning here of yes, at the beginning here. Okay, yeah, supposed to be around here. Yes, so you see codominance in complete dominancy. No, that one is fine. In complete dominancy, here we say that they say that this word, you see this word, it's what they said, intermediate. You see, intermediate, and then the title here is incomplete dominancy. And then if you talk about codominancy, it produces a mixture. It produces a mixture. Both allele are, yes, expressed. So it means that here you produce a mixture of two colors. So that's why I'm saying that incomplete dominancy is the best correct answer. Then we go to the next question. We are saying that the fluid fluid of the nucleus where free nucleotides are found, we call it the nucleoplasm. Plasm means fluid, and then nucleo from the word nucleus, therefore is the fluid of the nucleus. Then you are saying that the tangled mass of chromosome located within the nucleus. So we call this one chromatin network. So here is chromatin network. If you have any challenge, I can just show you on the distinction material. As I told you that distinction material, there is no question which cannot be answered by that book. Yes, so I wrote that book with the great time so that it can tackle any question they will set before and after you have written. So if you use distinction material, now I'm talking about those people who will be watching, not you now, definitely you will get a distinction. All right. Division of the cytoplasm, we call it cytokinesis. We talked about it yesterday. Say that and they're saying that the name of the someone is saying they're saying that is talking about the chromatin network. We talked about the chromatin network. It is here. We talked about it here. You see, entangled. You see, this is what you call chromatin. So it's a chromatin network. Yes. So that's what they are talking about. Here is the thing. Chromatin with chromosome consists of DNA and protein. So that is what they are looking at. This entangled. Yeah. That's what they are talking about. Ne? Yes. All right. We go to the next question. The name of the X and Y chromosome in male. We talked about this gonozome. So I think we are clear there. Someone is giving me the answer already. Chromosome is correct. Let's go to, I hope you guys, you got 10 out of 10. If you got out 10 out of 10, congratulations. If you lost one, at least you tried. Yes, we're still in the range of 95%. Genetic disorder characterized by chromosome mutation. I don't know how many people got this right. Let me just say it because I have no choice, because I have to tell you what is the most correct answer here. They're saying that genetic disorder caused by chromosomal mutation. The answer is none. I'm sorry if you wrote something or both. It's none. Yes. Why none? Because these are gene mutations. They are gene mutation, not chromosomal mutations. When you talk about the chromosomal mutation, we're talking about the whole chromosome changing. Therefore, we go back to Down syndrome. And if you check on Down syndrome, sometimes we call it chromosome mutation, sometimes we call it chromosomal aberration. So when you look at Down syndrome, that's where you can find chromosomal mutation. Let me show you what I talked about Down syndrome. About Down syndrome, when you come here, yeah, I think I didn't indicate it here, but it is chromosomal mutation. Yes. I don't remember where I wrote it, but it's there, whereby you talk about the whole chromosome is changing. Yes, when you change the number or the structure of chromosome, then we call it chromosomal mutation. Sometimes we call it chromosomal aberration. Yes, aberration or chromosomal mutation. But if you just change the base, then we call it chromosomal, we call it gene mutation. So these two are gene mutation because as a result of exchange of a base, a specific base. Then we are saying that importance of meiosis is both A and B because there is formation of gametes and then having the chromosome number. Yes, so this is both A and B. And then I'm saying that organelle where DNA is found, both A and B. We talked about this, and yesterday I was tricking you, and then someone got it right. I asked a question that give me any other place where you can find DNA other than a chloroplast. No, other than a mitochondrion. And then someone saying that chloroplast. And then the person couldn't tell me that nucleus. Yes, so but what I wanted to tell them that think about mitochondria and think about the chloroplast. You understand? Good enough that this question came, and I hope that person tried to get the answer right. So the answer here is both A and B. Don't forget that in paper 1 I told you that you must say that both A and B, not A and B, not both. If you wrote both, I'm sorry. If you wrote A and B, I'm sorry, you did not follow the instructions. However, maybe with the mercy of God, then maybe they might correct it, but you have to follow the instructions. Now I'm talking about those who are going to be writing the next paper. And then we are going to question 1.4. I hope, guys, you got 6 out of 6. Yes, question 1.4. Oh, blood group. We talked about blood group yesterday, but let's see what was here. Name the type of diagram represented above. This is pedigree. Pedigree, yes. So this one is correct. We talked about it, and someone was asking what's the difference between pedigree and the phylogenetic tree. So now we are clear there. Give the number of the allele that control blood group. We talked about this yesterday. We said there are 3 alleles. Alleles. Yes. Alleles or alleles, whatever case you call it, is fine, man. Just a moment. Sorry, this Samsung is here disturbing me. Just a moment, guys. I don't want to Samsung. Yes, I'm back. I'm back. I didn't want this Samsung. Okay, so we are saying that the diagram below represents the blood group in the family, or blood group O. Okay, sharp. Then we have name the type of diagram represented above. We said that the answer here is pedigree, so it is fine there. Then they're saying that give the number of alleles. We said it is three alleles. How many generations are represented in the diagram? We said we count 1, 2, 3. So the answer becomes 3 generations. Yes. They're saying that Lena's genotype is this. Lena's genotype is A. Yes, you remember what we talked about? Okay, now let's go to the question. State the possible phenotype of Vusi. Possible phenotypes. Phenotypes or genotypes? They want the genotype of Vusi. So if you are talking about the genotype of Vusi, where is Vusi? Vusi is here. But now, if Lena is A, small a, Vusi, this person I don't know, is small I. Remember, I asked a question that how is a person of blood group A and a person of blood group B can produce a kid of blood group O? Then I said yes, because if they both have heterozygous, they are in heterozygous state, they have the recessive allele, therefore they can produce a kid of blood group O. Therefore, how many marks they give it to Vusi is 2 marks. And they are saying possible. Therefore, Vusi can be A, small I. Yes, that's number 1. Vusi can be B, small I. Vusi can be I, small I, like that. Whereby if this combine with this, you produce O. If this combine with this, you produce O. If one of these combine with this, you produce O. You see that any of that can lead to Vusi. So they want 2 of them. If you wrote this and this, cool. If you wrote this and this, cool. Any of this, they wanted 2 of them. So I think, yes, you will get it right. Ne? Yes. Someone is saying that 4 generations. I don't know where did you get the 4 generations, but here is showing you 3 generations. Ne? Yeah. All right. Then they're saying that give the genotype of Bob. Where's Bob? Bob. So if this person is O, if this person is also O, definitely there's no way how Bob will be A or B. It will be O. So they're saying genotype of O, of Bob. Therefore, Bob is gonna be II. So I think that one is also correct. Give the name of the individual who displayed the codominancy. Codominancy means that you have blood group A and blood group AB. That's what they are looking for. So the first person I can think of here is you see here now, this person is blood group O. You see, blood group O. But the kids they produced is blood group B and blood group A. Definitely Anna is blood group A, B. Do you understand? Yes, so this one is here, can combine with this to produce B, to produce A. So this can combine with this to produce A, and this can combine with this to produce B. Therefore, Anna is definitely answer. But here it's showing you, so it's Anna. Is it Anna? Anne. Ne? Yes. So here they're showing you 2 marks. Then you have to look for another one. If not, if they don't want one, you have to look for another one. Another one, do we have another one? We can get another one just in case that Lena and Anne are kids of these 2. You understand? But Lena is having blood group A. And then Anne is having blood group AB. Definitely, if this is blood group A, ne, and this is blood group A, how will they produce this? Maybe one is blood group AB. You understand? Whereby this combine with this to produce blood group AB, which is Anne. You understand? What if What if? Because they said, did they say give the name of individual which displays codominancy? So the Anna is the perfect answer. However, based on this and this, so either this, because I can have this here, or I can be, maybe this can be A, because if I combine this with this, still I'll produce this person. You see, if I combine this and this, I'll produce this person. So is either any of this is either A like that, and then B like that, or is like this. Either of these could be. So it means that based on the analysis, deep analysis, not just easy analysis, it could be Curis and this, one of the two, not both. You understand? Not both. Both can't be AB. One of them must be AB. Then the other one has to be a heterozygous. You understand? Yes. So one of them could be. So the next answer, if Anne is the obvious answer, then the person who wrote either this or this could be also another correct answer. Someone is saying Vusi. What about Vusi? Let me see. Vusi is having blood group AB. No, there is no way how you can get blood group AB when someone is having blood group O. You understand? Because you don't have the second recessive allele. You understand? Yes. Yeah. Someone is telling me name. Thank you very much. Yes, I know that you are saying name. This is the obvious answer. Yes, it's a name. But don't forget that this could also be one of them correct. So the memo could have an extended opportunities after the discussion. However much, they could not see this maybe before they do the memorandum. But when you go to deep analysis, then you find out that this and this, there could be some student who wrote it and it could not be discredited. So should be credited if it results in that. You understand? Yes, obvious answer is that. However, even those, either of this could be an answer. We go to the next question. You're saying that the next answer is question below is a representation of protein. Okay, protein synthesis. We know that what is here is being protein synthesis. So now remember guys, we discussed about this and I told you that please don't make a mistake to go without knowing this. I told you that one of the paper, depending on which section, which paper they have brought, for example, if set 1, set 2, set 3, whatever, if it happens, one set must have at least transcription, maybe another one must have translation, another must have DNA replication. One of it must come. And then that's what I said, and say that please don't go to the paper without knowing it. All right, let's continue. They're saying that 1, 2, 1, okay, sharp. Let's go to the question and then we see Z. What is Z? Name, identify Z. Where is Z? Am I lost? Where is Z now? Oh, sorry. I couldn't see Z. The process Z. Process Z is, what is this? Remember, this is uracil. And then this is DNA. Definitely, this is transcription. Transcription. Remember, transcription is the formation of messenger RNA. So, molecule R. Where is R? Oh, molecule R is messenger RNA. Then they are saying that organelle Q. Organelle Q. Where is this? Where is organelle Q? Okay, organelle Q is the ribosome. Yes, ribosome. So guys, I used to tell you that whenever they think of organelle, think about two things, either nucleus or ribosome. So that the two organelles we talk about here. don't have any other organelle. Give the collective name for nitrogenous base O. This is O, ne? O is gonna be anti-codon. Remember, we said that code for DNA, codons for messenger RNA, anti-codon for transfer RNA. And I told you how to identify that this is a transfer RNA. Just write a T, transfer RNA. It's a T. And then you call it messenger. When you see it's a T, it's a T. So it's a transfer RNA. So the codes which found there, we call them anticodons. Determine the sequence of the nitrogenous base at S. At S. Where is S? Oh, it's here. And remember, this is, let me check, this is T, and also let me check this T. This one, it has T, even this one has T. Therefore, this strand is for DNA. Therefore, I've just said T goes with A, and then C goes with G, and then A goes with T. So basically, that's what you're supposed to write there. Here, the sequence. Which strand 1 or 2 is used as a template for formation of R? So we come back to R. So we have to mirror this. So this A goes with the T, U goes with the A, C goes with the G. So I have to look for what is this. I can continue. TAG. TAG. So strand 1 is the one which is used as a template so that this can be formed. Some people could have, maybe they have written strand 2 because this thing is facing that side, but that's just a confusion. It's just a confusion to see that some people are off. All right. So the answer here is gonna be strand 1. Strand 1. Yes. Someone wrote spinal cord. Wait. Okay, sharp. Someone said it's a spinal cord. Aibo. No, man. You guys, you're acting in the paper. All right. AGT. Okay, sharp. Then they're saying that which amino acid 3 or 4 will be brought to area P. Area P. Where is area P? Okay. Guys, there is something which was hidden here. Let me show you that hidden part of it. And I hope you guys got it right using the elimination method. Okay, all right. If we follow this, following this, ne? Yes, I hope we can get it. But if you just follow what is here, because there are 3, and here you have the bases. So definitely this goes with A goes with U, because this transfer A, U goes with the A, then this goes with the G. So UAG, so this one is gone, so we don't talk about it. G goes with the C, G, C, and then A. G, C, C, A, C, C, A, then this is gone also. Definitely this one will come there. You see? So you can see how you can identify it correctly. Ne? So basically here we come here and we say that amino acid number 4. Amino acid number 4. So that's the correct answer there. Then they're saying that name the type of sugar that forms part of structure molecule R. Part of sugar molecule R. Part of sugar. Name the type of sugar, not part. Molecule R is messenger RNA. So here they are talking about sugar. So it's gonna be ribose. Don't say ribo. No, is a ribose because they're looking at the sugar. So guys, I think you got 50 out of 50. Congratulations if you got it. Ne? Yeah, so it looked to be a little bit simple. Is there anyone who got 50 out of 50? Let me know in the comment section. Someone is saying 1.5.2. The collective name of nitrogen. They say the collective name of nitrogenous base. The collective name of. They are not talking about what is this. No, they are saying nitrogen. The key point here is nitrogenous base. So someone is talking about tRNA. tRNA is only this, but they're talking about the collective name of these three bases which are found on the tRNA. Therefore, the answer becomes nitrogenous bases. I think I'm clear there. Let's go to question 2.1. 48, 35, 40, 18. Still, 18, you can still do it. You can still do it. Let's go to section B, and then you see how much you're going to collect with the 50. Yes. Section B. Then they're saying that name the structure. Name the structure. Name the structure. So here is, I told you about it. Yes. Name the structure B. What is B? We come here, we look for B. Is a century. Remember yesterday I told you that please turn it so that it runs east to west. Is it east? Is it east way? Is it west? Is A? I'm not a geographer, guys. Ne? Don't quote me wrong. Ne? I think it's east this side and then west this side. I hope so. Ne? Yeah, so it must be running east-west or in the horizontal way so that you see it clearly. For example, it's supposed to be like this so that now you see. It must be running in that direction, ne? So that you see the actual phase. Yes. So I hope you guys, you did it and identify the phase correctly. Okay, let's go through it and then we find out. Okay, now we are saying that B is centriose, spindle fibers. These are homologous chromosomes, but I don't know whether they're asking you. Okay, spindle fibers, we are done. Identify the phase in diagram 1. Diagram 1, what is happening? You are seeing here crossing over is taking place. There are 4. It is prophase 1. Prophase 1. Give 3 reasons for your answer. Why are you saying it is prophase 1? First of all, you are forming here a bivalent. Chromosomes are under crossing over. Homologous chromosomes are under crossing over, you see. Yes, that's number 1. The nuclear membrane is starting to disappear, you see. Nuclear membranes are starting to disappear. The centrioles are moving to the opposite poles. So you can talk about that. Yes. Yeah. If there is any other, it could be accepted. I don't know what you wrote, but as long as it is correct. Describe the process that takes place at A. They're talking about the process of crossing over. Homologous chromosomes come together or align together, and then chromosomes of homologous chromatids, chromatids of homologous chromosome, they cross over at a point called chiasma. Yes, they exchange the genetic material, and then they split to form 2 genetically different chromosomes. So basically you just explain the process of crossing over. As I say that you cannot go to the paper unless you are insulting me when you don't know the process of crossing over. Yes, I talked about that. I said don't go to the paper without knowing the process of crossing over. Don't go to the paper without knowing the mera phase. Don't go to the paper without knowing anaphase. The rest you can skip, but those 3, don't make a mistake to go there. I think those who read, you did not. Someone is telling me that spindle fibers are formed. Spindle fibers are formed, they are moving, centrioles are moving to opposite folds. Spindle fibers are not formed. Spindle fibers are forming. They're not yet, you see. Yeah, it's also correct. It's also correct. Spindle fibers are forming. Yes. Yeah, because if they are formed, they have already formed like this. So they are forming. Remember, in prophase, they are in the process. Everything is in the process of forming. All right. That's English, but in most cases, we also mark English. All right, let's go to the describe the process. We are done with that. Identify the phase represent diagram 1, diagram 2. This I said you swap it because they're in pairs. Therefore, it is meiosis 1. Therefore, because they're in the middle, you see they're in the middle, you swap it, you turn it. Ne? Yes, as I told you, east to west. Ne? Therefore, it is metaphase 1. So the answer there becomes metaphase 1. They're saying that describe the difference in the event that takes place in phase mentioned above. Describe the differences in the events that take place in the phase mentioned in A and the same phase during mitosis. Or they want the differences between this phase, metaphase 1 and metaphase. Remember, mitosis doesn't have metaphase 1, metaphase 2. Here, chromosomes align along the equator, yes, in homologous pair randomly. You see, randomly. Chromosomes align along the equator in homologous pair randomly. While here, individual chromosomes align along the equator singly. In actual sense, they're asking you meiosis 1 and meiosis 2 of the same phase. Remember, we said that meiosis 2 looks like mitosis. Yes. So basically, chromosomes align along the equator randomly in homologous pair. That's the key point. Homologous pair. Yes. While here they align along the equator singly. Yes. So they're saying differences. They're not talking about tabulate. So they don't need you to write the table or to draw the table. Describe the results at the end of meiosis if chromosome D fails to separate. Chromosome D. Okay, let me just turn it so that you see it clearly. What happens if chromosome D does not separate? So basically, this is not a human cell. So don't talk about 47 chromosomes. Here, what you say, okay, definitely it's going to be like this. Technology is jamming. Okay. Still jamming. Okay, it's supposed to go that way. Just a moment, just 2 seconds. As it's back. It's back. Okay, now you're saying that it's gonna go through like this, and the whole thing, this becomes one cell, and then this one becomes another cell. So it means that what you describe here, say that one cell we have 1, 2, 3, 4, 5 chromosomes. Yes, one cell will have 5 chromosomes, while the other ones instead of 4, because it's supposed to be 1, 2, 3, 4. Then the other one will have 1, 2, 3. So you have 3 chromosomes instead of 4. You see? Yeah. So basically, that's what they are looking at, or they are looking for. Yes, that's what they are looking for. So that's why they ask, describe the result at the end of meiosis. At the end of meiosis, yes, if D fails to separate. Yeah, so it means that one cell will have an extra chromosome, 5 chromosomes, while the other ones will have 3 chromosomes instead of 4, and then the other one will have 5 chromosomes instead of 4, meaning that you have one extra chromosome and then the other one will have one short of chromosome. That's it. Then number 2, those people who wrote about 46 chromosomes, they didn't talk about here that this is a human what? They didn't talk about that this is a human cell. Yes. Number 2, look critically here. Let me show you something here. If this cell, these two cells, this one is going to receive X, 1, 2, 3, 4, 5, ne? And then this one is going to receive 1, 2, 3. So the fourth one is going to be empty. I see. Yes. So it means that now when this one divide, this cell is going to have 1, it's going to have 5 of them, and then the other one, when it divides, they're going to be 2 of them, but each will only have 3. Will only have 3. While this one, each of them, remember it's meiosis 2 now, each of them is going to have 5 because it's supposed to split equally. Each of it is going to have 5, and then this one, each of it, because there are 3 now, this one is 0, it's going to be 3. You see, it's going to be 3. That's another option, but still it gives you 3 here, 5 here, 5 here. 2 cells will have 5 each chromosome, while instead of 4 each, and then this one will have 3 each instead of 4. I think I'm clear. If you wrote it in that way, still they will credit you. Describe the process of DNA replication. Perfect. I think I was clear here, emphasized, and even said that if you could see my face. All right, DNA replication is a process by which DNA makes identical copy of itself. You understand? Yes. So you now tell us that DNA double helix unwind, weak hydrogen bonds break, and then weak hydrogen bonds breaking is the same thing saying unzip to form 2 templates, each. So to form 2 separate strands. What you want to hear the word separate. 2 separate strands. Each is acting as a template for formation of new strand using free-floating DNA nucleotide. Remember, if it is transcription, messenger RNA nucleotide. But here we say DNA nucleotide from the nucleoplasm, whereby a complementary strand is formed whereby A goes with the T and C goes with G. Is it clear? And then 2 new DNA strands are formed. 2 new DNA are formed, each with 1 original strand and each with 1 new strand. This process is controlled by enzyme. It could also be right. So this question has more than 6 marks. They have given it to it. So it means that they only credit 6, and then we write maximum, meaning that you have reached the maximum, therefore you got it. People are always asking me about the spelling. Okay. Tando is asking me about the spelling. Tando, what if I call you Tondo? Will you feel good? Instead of calling you Tando, I call you Tondo. I replace A with O. Will you feel good? Will you be the same person? If you are not the same person, why do you want to make mistakes with the spellings here? All right, it will depend on them, whether it means something else or it's the same thing. But we always encourage you to write correct spellings. All right, let's continue the next question. Read the passage. Gene, okay, code for blood clotting in humans. This gene made up of that 163 amino acids. The mutation occurred and result in amino acid 123, 128 sequence. This changes to this, and then this changes to this. Mutation has been transmitted as autosomal. Yes, it has been transmitted as what? Autosomal. And they are saying that they're saying that the mutation has resulted in a resistance of warfarin drug. Warfarin drug in human. Warfarin is used to treat prothrombosis. I'm used to the process of clotting. Prothrombin, fibrinogen, fibrin. Okay, prothrombin is the one which produce thrombin. So here thrombosis is the disease. Okay, treatment of thrombosis. Thrombosis results in the formation of blood clotting in the arteries, and then this causes the thin of blood to break down and the blood clot. Name one piece of evidence in the information that show that mutation for this gene occurred in DNA molecule. How do I know that mutation occurred in DNA molecule? Check here. Yes, it changed from here to this and here to this. Where do you find the T? Definitely find T in DNA. Therefore, that is the correct answer. So you talk about that, yes, the sequence change of this, change to this, and also this change to this. This shows that it was DNA. You understand? Yes, because DNA is the only one which has the thymine. Yes. Then number 2, they're saying that how many nitrogenous bases code for this gene? Hey guys, I talked about this. I even showed you how to calculate these nitrogenous bases. Remember, each nitrogenous base, each amino acid is coded by 3 nitrogenous bases. Therefore, I will say that. Yeah, someone is saying it has thymine. Yes, correct, because of this thymine. Yes, presence of thymine. Yes. Okay, saying that how many nitrogen? You're gonna say 163 amino acids times 3. So the answer you get is what you call let me see, how much is it? Okay, you calculate. Let me know the answer in the comment. Yes, describe what is meant by autosomal dominant allele. Autosomal, it means that is a non-sex chromosome. Non-sex, it is not carried on X chromosome. You understand? It is not carried on X chromosome. This allele is not carried on X chromosome and is dominant. Yes, it is dominant. That's what it means. It's an autosomal dominant allele. It's dominant and is not carried on X chromosome. So this disorder is caused, or this condition is caused by a dominant allele which is not carried on X chromosome. Yes. Then they are saying that the table below shows the amino acid and their corresponding codons. Hey guys, when I was trying to explain to you these mutations, I told you that you need to take note of this. Yes, I said it. The table below shows the amino acids and their corresponding codons. So meaning that this is messenger RNA, because they said that codons. They didn't say transfer RNA or anticodon. How will the mutation on this result in a resistance of this? Check 5 marks. What are you supposed to do now, man? What you are supposed to do here is you come back here and read here. A mutation occurred affected the amino acid this, and the sequence of this to this is changed, becoming this. This mutation has been transmitted as an autosomal dominant characteristics throughout the generation. This is the problem now. It has formed another protein. You understand? But how do I know that he's forming another protein? Okay, now the first step to score those 5 marks, you had to tell us what we started with and what we ended with. So we started with G, T, G, which changed to C, A, G. You understand? And then you have T, A, T. Someone said that is 489. Here is 489. Guys, if it is correct, you calculate it, then that's the perfect answer. Then you have to tell us that this one changed to from what? Okay, let me just make a big space so that we can know exactly what we are trying to show you. Okay, this is the first amino acid, this one, né? So first one, and then the second one, let me write it here. It is T, A, T, and then T, C, T. But now, what do these ones indicate? They're saying these are codons. Yes, so I don't know, I can't read it here unless I change this. So change it. So this is the first mark by changing G, A, C. And then this one is G, U, C. What does GAC mean? GAC, let me first remove this so that I see clearly. GAC is leucine. It indicates leucine, this one. And then GUC indicates glycine. Yes. Now, what about this? Let's convert this. This is gonna be it's got A, U, A, and then this one is gonna be A, G, A. So what does this one indicate? This one indicates this one indicates AUA. AUA is phenylalanine. Phenylalanine is this one. And then this one, what does it indicate? AGA. AGA is arginine. Guys, these are just names, né? Yeah, you don't need to know them by head, né? Me, I'm saying them that arginine, what? Yeah, because we find this in biochem, né? Yes, in biochem. So this is it. These are the amino acids, which you know that, yeah, the essential and non-essential amino acids. So as long as you write what they gave you. All right, now we have translated. You see, we have what? Translated. Here we will get a mark. We will get a mark. It's 1 mark, 2 marks. Then also changing this is another mark, 3 marks. Then changing this to this is another mark, which is 4 marks. You understand? But now put them in words. The codon, the first codon, AGA, ACGS, CGA was changed to ACG, cool, which indicates that this was changed to this, okay? And then this produced leucine. It produced glycine instead of leucine. So you have changed the amino acid. And then also this one, the second phase, we change the amino acid from phenylalanine into arginine. So different amino acids will be changed. This will be changed into this, and also phenylalanine will be changed into arginine. You understand? Which makes a different protein. It won't be the same protein because these codes which change, they change the amino acid. So therefore a different protein is formed. That's what it means. So you guys were supposed to show us that this changed to this changed to this, meaning that new amino acid is formed. So it will be coded for glycine instead of leucine, and then here it will be coded for arginine instead of phenylalanine. Therefore a different protein will be formed, and then this protein is resistant, because they said the mutation has resulted in resistance. Yes, this protein which has been formed result in the resistance to this drug. So basically that's what they wanted. I explained it yesterday, but now when I'm seeing this question, the way how they try to trick it, when I was explaining it, I explained using one example, and I told you that don't make a mistake to go to the paper without knowing this, because I know they're going to bring it, and I know that people are going to have a challenge to explain this. And these people, you know what they do, that if they find out that this question is hard for these students, they keep on bringing it. I don't know why, but they keep bringing it. Maybe they want to make sure that everyone knows that concept. But if they see that now you can know this one is easy, they don't bring that one anymore. They bring more, which is a little bit difficult to see that, yeah, you can apply. But I hope you guys, you got this question correct. Let's go to the next question. Let's go to the next question. They're saying that they're saying that the effect of this mutation has explained the effect of this mutation has with the prothrombic. This one you just come back to the extract and then you read here. The mutation has resulted in resistance, you see? Yes. And then in the human of, okay, treatment of used to treat this, okay, the thrombosis result in the formation of blood clot in artery. So this will result in the, yes, you see, the effect of this mutation on human with this. So meaning that it will result resistance. First of all, the mutation will bring about resistance. Number two, it will also bring about this formation of blood clot in the artery, you see? Yes. And then also will cause this to break down, you see? So basically, no, this one is out because this is a drug, né? So it means that it will not allow this blood to break down, the clot to break down, because now it is now resistant. So meaning that the blood clot will remain there. Why? Because of the mutation which has developed. Therefore, you talk about the resistance, and then you talk about what this you see here, this formation of the blood clot in the artery. Therefore, these clots will not be broken down because of the resistance which has been developed. So basically, the answer was supposed to be obtained from there. Hemoglobin. Someone is saying that if I could say that it's okay, has deleted it. Okay, it's fine. So guys, I think this question was one of the challenging questions, just challenging questions, and I knew that it's coming. And then when I was emphasizing it, yes, I tried to see that everyone can pass it. Yeah, we go to the next question. That is question 2.4. Question 2.4, we are saying that polydactyl, these are the people who have extra finger. Maybe they have 6 instead of 5, ne? They have 6 instead of 5, lead to extra finger on extra finger or toys. It is caused by a dominant allele. This word, you remember I told you guys when we talk about dominancy? Yes, a man who is heterozygous for this, meaning that is capital R, small r. Yes, because they said use R, ne? And a wife who is not, meaning that it's a recessive, so is homozygous recessive. Use the letters R and the RR, do the genetic cross to show the percentage chance that their children will have polydactyl. The percentage chance of having the polydactyl. So here, what are you going to write? So it's simple, guys. Here is very simple. Let me just talk about it here. P1, F1. I told you about that, and I said we give you a mark. Then we say phenotype, phenotype. I think you guys wrote it that. You go back to the question, you find the phenotype. They say that homozygous, a man who is having the condition, a man with polydactyl. Yes, polydactyl. So you're gonna have to say a man with, yes, polydactyl crossed with a lady without polydactyl. Yes, meaning that here I'm gonna have a lady wife with no, yes, here with, yeah. So you finish. Then here is genotype, but they say the man is what? They say the man is heterozygous. Therefore I'm gonna say capital R small r crossed with a lady who is recessive. So here I'm gonna say meiosis occurs, and then I'm gonna say capital R small r small r small r. I say these are gametes. If you didn't circle them, it's fine. If you circle them, it's fine. Yes. Then now you can use the Punnett square method, yes, or you can cross. Either way, as long as it's correct, you write the word fertilization if you use the Punnett square, or you write fertilization here, fertilization here, and then you say C, and then you start capital R small r, this with this, capital R small r, this with this, small r, then this with this, small r. So these are offsprings, you understand? Yes. So basically this is what you're supposed to do to get it right. But now we come here, we mark. If you don't know, if you got this wrong, then automatically we shall come here, we say fertilization, it will give you a mark. And also writing this, you see now you have 3 out of what? Out of 6. But if you got all these correct now, but the question is asking you, there might be a compulsory mark. Let's find the compulsory mark here. They're saying that use the letters, do a genetic cross to show the percentage chance. Yes, the percentage chance of so percentage is a key point. Is a key point of having the kids with the disease. So basically we have 2 people normal is caused by a dominant allele. 2 people with the disease and 2 people without. So basically it's gonna be 2 out of 4 times 100, which gives you 50%. You see, this is a key point. We mark you. So we're gonna mark you here, we mark you here, we mark you here, but we're gonna first count this. How many marks are there? How many marks we are given? Yeah, we're gonna give you this one plus any other 5. Yes, or it will depend on, but this is the compulsory mark which they're gonna give. Someone is asking me about agriculture science. So many people asking me about agriculture science. Guys, I told you that unfortunately this year I'm not sure that you're gonna work with agriculture science, but next year 2024 we will work with agriculture science. Yes, it's because currently we don't have a school which has which does the agriculture science in this area. Unless you send it to me, you scan it, you send it to me, then we'll do a memorandum for you. Yes. All right, yeah, saying that below shows the graph below shows, I think guys you got all the marks here. I'm skipping, I'm sorry. Okay, so we are done with that, so let's go in summer squash plants. White fruit is dominant, so we need to know that white is dominant over yellow, and then round is dominant over oval. So this is dominant white over yellow. Okay, now let's start. Summer squash plants that are homozygous for white and round is crossed with plant that is homozygous yellow. Okay, okay, sharp homozygous. Remember, yellow is recessive, ne? And then this one is white is dominant, and they are saying that dominant around, oh, dominant over crossed with recessive. Okay, give the genotype of parent one, parent in the first generation. Okay, parents in this case, they say that white, therefore capital B, because they said homozygous, and then white, and then round is capital D because they said homozygous. So that is the first, and then the first mark, and then the second mark could be is the crossed with the homozygous. Yellow is, we said, is small b, and then oval is small d. So that's the second mark. So that's what they are looking at here. The phenotype of F1 generation, because all of them are dominant, therefore is going to be capital B, small b, capital D, small d. So the phenotype both is going to be white and yellow. So it's going to be white, correct, and oval. Did I say? Not oval, man. It's not oval. The one which is dominant, because I have capital D here indicates round, right? Round. So it's going to be white, a mark, and round, another mark. Then they are saying that two plants that are heterozygous for both characteristics were crossed. Okay. What if I use, someone is asking me, what if I use X and Y? Guys, I told you that in this generation, in this, you only use X and Y if they have told you to use X and Y. So they didn't tell you that this is sex linked, ne? So the answer will be wrong. But if you wrote this, and this, then automatically you will get the answer. Those remarks, and if you got the percentages 50, you'll also get another answer. At least you'll get 4 out of 6, ne? At least you did not come out with a 0, ne? Yes. They're saying that the two plants that are heterozygous for both characteristics were crossed, meaning that I have capital B, small b. I have capital B, small b, capital D, small d. So this is what I have. So if they are crossed, all possible genotypes of the gametes. So here I'm going to find out the gametes. This gonna go with this, as I told you yesterday. This is exactly the same thing I told you yesterday. This we're gonna be go with this. It's gonna be capital B, small d. Then this with this, it's gonna be capital small b, capital D. This with this is gonna be capital B, small d. So these are the gametes which are possible. So 4 of them, maybe 2 mark by each. So you get that correct. Then we go to the next question, guys. If there's any question I've skipped, you let me know, ne? Sometimes I skip when I don't see. All right, how many plants in, okay, so how many plants in the next generation are likely to have yellow and oval fruit? How many plants in the next generation are likely to have yellow and oval fruit? Yellow and oval. So we come back here and look at for yellow. Where is yellow? Yellow and oval. Yellow. Okay, now they're saying that, okay, both were crossed, heterozygous. So automatically when you cross, you'll find out that because both are homozygous recessive, meaning that they are going to be small b, small d, small d. So the answer here is going to be one plant. Yes, or maybe 1 out of 16. Yes. So one plant out of 16. I think let me just show you here from the distinction material what they are trying to show you without doing the genetic cross. Yes, This is what I'm saying. You see, they are looking at this. It's only one which has both letters when they are homozygous recessive. So basically, that's what they are looking at. When you come back here, that's what they are looking at. It is small b, small b, small d, small d. That's what they are looking at here. Capital small b, small b, small l, small l. Yes. So and the percentage here, if you count all of them, they are 16. Therefore, it is 1. It's only they are looking for this. Yeah, one individual, or 1 out of 16 individuals. So I think I'm clear there. Then we go to the next one. Yeah, someone got it right. I think, guys, you are collecting marks now. Ne? You are collecting. Give a possible genotype of both parents that must be crossed. A farmer wants squash, okay, that way, white and oval. Yes. So the two, if you want white, basically, if you want, all of them are supposed to are white, which is capital letter, and then oval is a small letter, meaning that the resultant must be capital B. For example, it must have a capital B, and then it must have 2 small d's, because they say the oval, and the oval is recessive, you see. So basically, the genotypes of the parents must be this: capital B, small d. That's the first person, and then the second person can be capital B, small d. That's the second person. Or the first one can be like this, and then the second one can be capital B, small b, capital D, because this B will be somewhere. Yes. So basically, if you wrote this, they will give you a mark. I think they want to. Let me see. Possible genotypes. They want to. This with this. Yes. Or this with this is cool. Or this with this is also cool. So yeah, that's what you were supposed to write, so that at least you get the correct mark there. So the options there, if I have not mentioned another option, then automatically it means that it will be marked correct. Who got 50 out of 50 in section question 2? 50 out of 50. Let's go to question 3. Someone is asking me, you describe, I don't know, what was that question? I don't remember what question was it. Okay, let's go to the next question, and then they're saying that the graph below shows the results of artificial selection for protein content in the meal plant over 50 generations. Describe how the farmer did artificial selection. Here you just had to explain the artificial selection. Remember, in artificial selection, you identify the character, describe how this farmer did. So he identified, you see, they identified the character of producing protein in the meal, and then after that, he identified those plants with that character, yes. Then he crossed those plants with those ones which have that character, which have that character. He crossed them, yes. Then after that, he selected only those which produced more in the first generation, and they were allowed to cross-pollinate, because they don't do sex here. This is close pollination. It's pollination. So he cross-pollinated those which could produce large number of protein, you understand? And then at the end of the day, he found out that the plants were able to produce a large amount of protein. What was the average percentage of protein content in the meal in at the 15th generation? So here you had to draw a line touching there, and then you draw a line touching there, and then you find out maybe 12.8, and then you put these units, kernels. Yes, so that becomes the answer. And then someone is going to ask, what about 12.89, or what about 12.9? Yeah, they will give a range. Yes, someone got 38. That's still good mark. 42, still good mark. 34 is good mark. All right, guys, thank you very much for getting those good marks. At least you are promising. But by how many times did the average percentage of protein content in meal grain increased between this, meaning that they are looking at for percentage increase. They have brought the question again. By how many times? By how many times? By how many times? Okay, the average percentage of protein, yes, increased between 40 and 45. I come here, I find what is final. The final is 20. 20 minus the initial, which is 14. So you get how much here? You get 6. 6 divided by the initial. The initial, which is 14, times 100. So the answer you get here is the number of times it increased. Yes. In terms of percentage, saying that by how many times did the average percentage of protein, yes, okay, you had from 14 to 20. So someone might have a different version. So you guys, you have to calculate for me, and say it's only 1 mark, so you how many times from 14 to 20? Basically, that's what they are looking at. So when you do the percentage increase, then you'll be able to know how many times. And then here, you guys, you calculate and you get the answer. Yes. Describe one way in which the process of artificial selection is different from genetic engineering. We talked about this. Artificial selection, there is no manipulation of genes. You only have to select those organisms which are desirable by the human, and they are given a chance to reproduce. While in genetic engineering, you don't have to cross these organisms. You just have to select using, there is manipulation of genes. That's why it's called genetic engineering, manipulation of genes. While here, it's only you cross the organisms. You don't touch the genetic composition of an organism. Yes. Number 2, this one is a little bit of expensive. This one is a little bit of cheap, in which this process they differ. Okay, this one takes short period of time, while this one takes generations and generations. Yes. Someone is saying 12.5. Someone is asking me, is it fine if I don't put the units there? Hey guys, those are the discussions we do, or the discussions they do when it comes to the discussion. Should we correct this? Should we not? So basically, what you need to do is you must not always forget about the units, because the units are very, very, very important. All right, let's go to the next question. Darwin's theory of evolution by natural selection. I explained this and I told you that unless they bring it as like this, then I also explained to you what you're supposed to say. Natural selection, you talk about there is variation among organisms, those which are favorable, those with favorable characteristics, because they didn't give you here an example. So you don't pick an example. Those with favorable characteristics and those without favorable characteristics. Those with favorable characteristics, they reproduce in large numbers. They survive and reproduce in large numbers. And they pass their genes to the next generation. And those without favorable characteristics, they die in large numbers. This is called natural selection. So those which pass their genes to the next generation, yes, they reproduce in large numbers, and then after here you also supposed to talk about, just in case, because it's 7 marks, maybe you can talk about natural selection is what is a survival of the what of the fittest, which organism is best suited. Sometimes when students, they are trying to define this, because this is a block question whereby it is no example, no what, you just have to explain just what you know. Yes, you on natural selection. So basically, this is what I'm trying to say. Yes, I'm almost there. Yes, here. Natural selection. Yes, it's natural. Organism produce large number of offspring. There is a great deal of variation. Some have favorable characteristics and some do not have. Those when there is a change in environmental conditions, yes, if there is competition, the organisms with favorable characteristics will survive, and then those with unfavorable characteristics will die. Organisms with those which will survive will reproduce in large numbers, thus pass their alleles to the next generation, to their offspring. So basically, that's what they are looking at. Just describe the process of natural selection. That's it. Then we go to the next question, guys. That's what they wanted from there. 3.33, investigation was done to determine the relationship between height and the head and the bite force. The head sorry guys, there was some technical problem with the sound, but now it's back. Okay, they're saying that what I was saying that the investigation was carried out to see the relationship between height of the head and the bite force in lizards. Ne? Yes, the bite force in lizards. Scientists, okay, collected all of that, so you are supposed to read there. Ne? So now, what is the first question there? What is the independent variable? So the species will determine the head and also determine the force of the bite or bite force. So the species becomes I, D, X, Y. I told you that. Ne? So this becomes the species, becomes the independent variable. And the independent variable, then the bite force. The bite force. Yeah, someone can ask me that, what about that? Height determines the force of the bite. You understand? How much force do you need in neurons? Yes, the height will, because as the height increases, the force also increases. Yes, but what determines this is the type of the species. So yes, if it was this and this would have been the independent variable, dependent variable. But now we have this which determines this and this. You understand? Yes, so the species will be the independent variable, and then this bite force is the most correct, but I hope they also credit for those people who wrote the height of the head. Yes, because if you are to plot a graph, you'll find out that this is A, this is B, this is C, and then these two will have two graphs in that case, whereby this is the independent. And someone talking about full marks by Darwin, that great man. Yes, state two factors what were kept constant. Factors which were kept constant here, they talked about, there are a lot, so you have to talk about in a factor, but here they talked about that each species were kept in a cage with environmental conditions similar to that of the habitat, meaning that you kept maybe same temperature, same humidity, same light, same, talk about environmental factors which you could keep constant, or any other factor which you read there and then you find out that it was kept constant. So they're saying that apart from the sample size, state one way in which the reliability of the experiment was ensured. So when you come back here, apart from the sample size here, what did they do? Did they repeat the experiment? Yes, they say that the height, was measured for each lizard and the average. So they calculated the average height of each lizard. You see, this is also increasing reliability. Yes. Then they are saying that school name the type of variation which is displayed in this characteristic. I told you that whatever you are born with, and it changes since you are born up to where you are now. How big was your head before when you are born, and how big is it now? How big is your size, and how big was it when you are born? So things which change like that, then it is continuous variation, because it's all individuals, they have the same. But if it does not change, like blood group doesn't change, so it means that it's discontinuous. So in this case, name the type of variation, because the size keep on changing, therefore we will say that it is continuous variation. Describe the relationship between height and the head of the lizard in terms of the bite force. As the height of the head increases, the bite force also increases. You see, 10, you see, so the bite force also increases. Which species A, B, C would be expected to be feeding mainly on rough fibrous plant? So we are looking at the one which has the force which is strong enough. So we have this. So its answer is C. Why? Give a reason for your answer. Because it has a strong bite force, so that it can chew these tough fibrous plants. Yes, so it has a strong bite force. Yes. Then they are saying that explain, so we are done with that. Which species A, B, C would be the most suitable to live in narrow areas of the rock? The one which has the small head, because it can go through the narrow or the crevices. So basically it is species A. They didn't ask you the reason, but if they had asked you the reason, then you'd have said that because it has a small head. Yes, it has a small head. So that is the correct answer. Another question is 3.4. 3.4. Guys, are you getting it? If you have any challenge, let me know. Maybe you are not so sure about the answer I talked about. Yes. Let me know. Yes. Let's go to the next question, and then we try to do the last one. Okay. Which species in the diagram? What is this diagram? Is a phylogenetic tree. Yes, which species in the diagram above existed or survived for the longest period of time? You look at how long the length of the bar. Check, it's this one. So the answer is Homo erectus. Then we come to calculate the period of million years. Australopithecus afarensis and Australopithecus africanus coexisted. So here, remember I told you this and I gave you those questions that you will never be asked a question and it will never be fail to answer it. Remember when I was going through this and I told you here period of existence, you see, competed for resources. So that's the same question which they are asking you here, that which give two pair of species which competed for resources. So now what they did here is they gave you that, now they're asking you here, you see, they coexisted. In other words, they competed for resources. But how long did they do that? They did that if this Australopithecus africanus is here, and then this is afarensis. Did I say afarensis? Wait. Afarensis and africanus, yes. So this is the time when they competed for resources. So basically that's the time. So I have to calculate here. So it is 2, 3, 4, 2, 1, 2, 3, 4, 5, 6, 7. So it's 2.7. Remember we count from this side because the 0 is this side. And then this one is 3, 1, 2. So it's 3.2 minus 7, 2.7. And then the answer you get here is million years. Not million years ago, because here they are talking about the range. Is million years. So guys, I think when you subtract here, what is it? What is 2? Yeah, make sure that you subtract correctly, né? But it must have that word million years, because in most cases we mark the what? The units. Name the species that was also known as handyman. Homo, I told you this. Homo habilis. Remember when I was going through many schools trying to explain these skulls and stuff. I hope you guys you got it right, ne? Got it right. State 2 uses of tools in developed. State 2 uses of tools that was developed in 2.6 million years ago. 2.6 million years ago. 2.6. So it's 2, 1, 1, 2, 3, 4, 5, 6. So something which, yeah, it's this one. Yeah, the other one tools. Yes. So what is it? You just copy from here, because they said this for So that's, there is no cramming there. Then so we got the answer there. Identify 2 species that use the most complex tool. 2 species which use the most complex tool. The most complex tool is this one. So if you look at it here, it is the Homo sapiens and the Homo neanderthalensis. Homo sapien and Homo neanderthalensis, or Neanderthals. Sometimes you call them Neanderthals, but Homo neanderthalensis. Yes. Is there anything I've skipped? Yes. Okay. Let's continue. Name 2 species. So we have done that. Explaining how the change in the brain size over time relates to developing tools. I explained this also, that increase in the brain size increases the number of cells. Yes, so this number of cells, if they are increased, then automatically it means more intelligence. And these sophisticated tools require thought. They require what? Thought. Therefore they will require more intelligence. So basically that's what they are looking for is here. We talked about it here. Cultural evidence, using tools in cutting or improving in nutrition. Developing of tools become more sophisticated as hominids evolved. Tools require planning and thought. You see, yes, this is what they are looking at. Tools require planning and thoughts. This is what they are looking at. Development of tools become more sophisticated as the hominids, yes, as the hominids develop. You understand? Yes. Or as the brain size changes, yes. And then they become more sophisticated, and these tools, they require planning and thought. Therefore, you have to increase the brain so that you can increase the intelligence, so that you can plan and you can also think about what you are planning. So basically, that's what they are looking at there. Then we go to the next question. They're saying that name two species used to, okay, explain the size. So we are done with that. Let's go to the last question. Yes, the last question. Someone is saying that what if you calculate it differently but you got the answer? It's cool. It's cool. Maybe they will give you a mark. How many marks are there? Which one is it? I don't know. But if you got the answer correct, maybe, because here they're saying show your answer, you're working, maybe you'll just only get one mark for the correct answer. Yes, and then you lose the 1 mark for the Let's go to the next question. 3.5. State the genus of Adipithecus, did I say Adipithecus? Australopithecus sediba. Australopithecus. It's this one. You have to write it in full. Australopithecus, because they're looking for the genus, the first name. Yes. Describe, it must start with a capital A. Describe the shape of the spine. We talked about this. Is S shape. They're saying describe. Just is S shape. That's all. S shape. Then they are saying that state what is Hosi Dube. Thank you very much. You are also the best because you kept yourself on the channel, and we pray that you get that distinction you are crying for, and I know you will get it. You will get it. There is one person I want to thank. Has been a member since the beginning. Oh, I've forgotten the name. Maybe I'll thank her in the next video. Yes. Yes. Okay, state what is meant by the transitional species. We also talked about this. Transitional species is a species which has both characteristics of the present species and the most recent common ancestor, and the most recent species. You understand? And the most recent species. Don't say and the ancestor. Ancestor where? And the most recent what? Species, or before it. This is what I'm trying to say. When I was yes, here is transitional species. This is a transitional species. It is having characteristics of this and the recent ancestor, or the most recent what? Ancestor. Yes, so that is a transitional species. That's how you're supposed to connect it both in front and backward. So you're supposed to explain that in two versions. That's why it is 2 marks to talk about in front, and 2 marks. Yeah, but in most cases here we use the all or nothing law. You either get it all or you lose it all. What if you misspell the Australopithecus? Yeah, you guys, spelling. Maybe they'll correct you if it doesn't mean something else. But if it means something else, they won't correct you, ne? So what is this? And then what is this? Do they mean the same? They have the same letter, but differently. So it depends on how you have spelled it, ne? Yes. And they're saying that give the letter of the pelvis that would be represented by, okay, give the letter. Remember, I said that Australopithecus sediba is a transitional fossil. Remember, I talked about it and I emphasized it here. Check. Australopithecus sediba is a transitional species of fossil. You see the distinction material? Yes, those people who used it correctly, I hope you're going to get a distinction, because there is no way you're going to fail to get it. No question can be failed to be answered by that. Okay, they're saying that give the letter of the pelvis that would be represented by Australopithecus sediba. In this case, you look at, this is more wide, but it's not that wide. This is more narrow, but it's not that narrow. So this is like 1, 2, 3. So this is in the middle. So this is Australopithecus sediba. Therefore, the answer becomes A. Explain your answer, because now you have to relate these two. That Australopithecus sediba, yes, this A, the pelvis is not as wide as C and is not as narrow as B. Is not as wide, yes, meaning that is wider than this, but still is not as narrow as this. This one is much more narrower, and not wider compared to this. So it's not as wide as this, but it's not as narrow. So sorry, it's not as it's not as narrow as this, ne? Yes. Or how many marks are there? 2. Yes. So basically you would have said that is not as wide as C and is not as narrow as B. That's the first mark. If you also wrote like this, you will get a mark. This is not as narrow as this. No, not narrow. This is not narrow. Is not as long. This skull, this pelvis is not as long as B and is not as short as this. You understand? This one is shorter compared to this, but this one is longer compared to this, which means that this is in between these two. If you look at how long it is, how long it is, how wide it is, how wide it is. You either use wide to explain, you have to compare this with this and this, or you have to use how narrow it is. So not narrow, how short it is, short and wider, isn't it? So it must be not as wide as this and not as narrow as this. Or you say not as short as this and not as long as this. This is not as short as this and this is not as narrow as this. So basically, either way or whatever you have written, then you will get those 2 marks. We go to the last question, the last bullet in the gun. The last bullet in the gun to see who got 150 out of 150. Explain the significance of change in the prognathism from Australopithecus sediba to Homo sapiens. Prognathism meaning that protruding jaws. But there's something which you need to know. When there is protruding jaws, check if this from here to here, check if this protruding jaws, basically the prognathism is reducing. Yes, because we said that this is prognathus, it's not prognathus, so it's reducing. So they are not saying increase or decrease, they're saying explain the significance of the change in the prognathism from Australopithecus sediba to Homo sapiens. So the prognathism here is reducing. So when you become more straight, flat, it means that it creates more space for the cranium to accommodate more brain. But here, because here it is not, because he has to have too many muscles so that this is being supported. Therefore, the first answer is to create or to have more brain, which indicates brain cells, and then you link them to the intelligence. Also, when it is more pregnant as like this, the position also keep on changing, the position of foramen magnum. You see, if it is straight, if it is flat straight, then the position of foramen magnum indicates like this. And if it is more protruding, then automatically this position is going to have to go a little bit back, so that it brings about the shape. You see, it brings about the what? The shape. But here the shape is straight. Therefore, it also allows the shift of the foramen magnum, so that it can bring about bipedalism. Bipedalism, which means that you're walking on two hind limbs, and then this will create more faster. The organism can move more faster. So the significance here, you have to talk about the cranium becoming more bigger, creates more space for the cranium to be bigger, meaning that more brain cells, so the intelligence is linked here, while here is small. And then number two here is you talk about you can talk about it makes the position of foramen magnum to move a little bit forward, so that it can allow the organism to walk on two hind limbs, or to allow bipedalism to take place, so that now the organism can walk faster. So basically it brings about changing from the slope face to a flat face. Yeah, so that's what you are supposed to explain there. Guys, I hope you got. So guys, so what about their deity? You're talking about, how about the deity? Yes, there are some things which I've not talked about, but you look at when you are changing from prognathism, meaning that even the jaw, the shape of the jaw will change. You understand? And if the shape of the jaw changes, because it is like this, let me show you in the book, the distinction material. You see here, ne? So the more it's checked, ne? So even this is gonna change. You see how it is slanting. When this thing is out, then automatically this one, you see. So the position is cool. So the position, we have seen it, because now it's not protruding out, so it means that the position is cool. You see now, when it is straight, it creates more space for the skull. You see, it creates more space for the skull to accommodate more brain. But here, because it is a little bit flat, so it means that the skull, the cranium becomes smaller. You understand? Yes. And then also prognathism is basically you change. Remember, it's the jaw. This jaw, which could be also from big teeth. You can talk about the teeth becoming more smaller. So it means that you are shifting away from the rough, tough food to rough, raw food to one which is more cooked. So that, yeah, so you have to link. Your answer was supposed to be linked. You know, I told you yesterday that once it comes to this describe thing, you don't know, but you think about something which is correct. Describe thing, you write everything, and I caught it that write all your rubbish. I call it all your rubbish. But in other words, I mean that you write everything you think it is correct. When you're marking, we will look for what is correct in these things whereby they tell you that describe what. Yes. So you guys, thank you very much for attending our class. You have been good people. You have been supportive. And then I wish you the best. I wish you the best in your life. If you have sisters, brothers, and stuff, please refer them to this channel. If you have not subscribed, subscribe, like the video, so that it can move, and then others can also benefit. Yes, benefit. They can judge themselves, so that they go for these holidays when they are happy. If you have not made it, just this is a simple advice. If you have not made it, guys, listen carefully. Not the best in class will be the best in the world. Is it clear? So it must not make you to be too much stressed. No, I don't think, I'm not insulting, but I'm just trying to show you this. Ne? I don't think Mr. Cyril Ramaphosa, our president, ne, was the best in class, but he's the president. So that's what I'm telling you, that not the best in class is the best in the world. You understand? So that must not influence you. You understand? Keep focused. I don't think Mr. Let me say Musk, Elon Musk, whatever, yes, was the best in class. I don't think so. You understand? So not the best, I repeat, not the best in class is the best in the world. Those are two different things. I repeat, those are two different things. However much we want us to be best, but please don't lose your hope. Yes, you should not lose your hope. Is it clear? Lastly, don't treat others bad because you are the best in class. Because you don't know that this person you don't treat right might be your boss. It might be your boss tomorrow when things... we managed to reach our conclusion today. And then we see that soon we're going to be going to the university. Let's go through the paper. And then we see how did we make it. Yes, any challenge, if you have any complication, if you think your answer is not correct, let me know in the comment section before I go offline. So don't wait for me to be offline. Yes, many people they tend not to comment when the time is still there. So please make sure that you comment when the time is still there. Okay, let's start without wasting time. Yes, what was the first question? What was the first question? I thank those people who have been here trying to follow us from the beginning until the end. Those people who watched our last minute discussion, all of you, those people who watched our prediction, I know that yes, you have made it. Let's go without wasting time. Saidi, as usual, don't forget to subscribe, like, share, and also tell other people that, oh, there is a channel called Thundereduc. Yes, where we focus on quality. Question number 1. I don't have time to go through the objectives. Which one of the following organelles is a site for protein synthesis? I told you yesterday that when they talk about organelle, you think about two things. Number 1, that is ribosome nucleus. The rest, you don't think about them when it comes to protein synthesis. So the answer becomes A. So that is 2 marks. We go to the next question. In humans, gonosomes, remember these are sexy chromosomes. Gonosomes determine the gender. Which combination below shows the correct gonosome for male? You know that males are X and Y, while females are XX. So you are looking for that. So if you come there, you see that X and Y is C and A, while XX is C. So this one, because of it has no XX, so it makes it to be ruled out. So the most correct answer becomes C. So you get your 2 marks in there. An organism that has capital letter TT, that is the genotype. The correct term to describe the organism's genotype is capital T, capital T. It is because the same kind of letters or alleles, therefore we shall call them homo. But because they are dominant, if it was small letter, then it's a different story. Because it's dominant, so the answer is supposed to start with the word homo. So I have homo and I have homo. So these ones are out already. So here it becomes what is the first homozygous because it's dominant, therefore D becomes the most, so C becomes the most correct answer. So you get your 2 marks there. Yes, Down syndrome is a genetic disorder where an individual has an extra copy of chromosome number 21. Yes. Hey, you tell me even the comment and then I see if you guys you did follow our predictions. Yes. All right, which one of the following may lead to Down syndrome? Failure of the chromosome to replicate, out. Failure of the chromosome pair to separate during meiosis, looks to be correct. Failure of the chromosome to form pair during fertilization, nope. Failure of the chromosome pair to cross over during meiosis, out. So B becomes the most correct answer. We go to the next question. Gradual change in the characteristics of species over time is known as. Remember, we say that increase in the complexity of an organism is called what? Is called evolution. So since we are dealing with the biological evolution or biological or individuals which have life, you're not talking about the sun, the moon. So it's about life. Therefore, it becomes biological evolution. So D becomes the most correct answer. Yes. We continue to the next question. Which one of the following is a reproductive isolation mechanism? Reproductive isolation mechanism. This one deals with the things which make organisms not to have sex, not to have, or if they have sex, they are not able to have babies, to separate them reproductively. That's the meaning of reproductive isolation. You separate reproductively. Now, mechanism. So number one, adaptation to the same pollinator. That is out. It cannot separate you because you have the same pollinator. Species courtship behavior. I don't think humans have the same behavior of courtship like dogs or like cats or like but humans, you are weird. Because you have different, you have dog style, you have cat style, you have the humans. Let me use another example. I don't think the dogs have the same style like cats. You understand? Of courtship. You understand? Yes. So it means that the most correct answer here becomes B. Courtship species specific courtship behavior. Production of fertile, it is fertile, meaning that it can breed. It can what? It can produce into another baby. Breeding at the same, not different times of the year, same time of the year. That one is also, it's fine. You understand? So the most correct answer looks to be B. Analysis of mitochondrial DNA is an example of a line of evidence. We talked about this and emphasized it, and I say that when you talk about the evidence of evolution, we said that genetic evidence, the mitochondrial DNA is the evidence of out of Africa hypothesis. Yes, we say that mitochondrial DNA is an example of genetic evidence. If they ask you the evidence, you have to say genetic evidence. An example is mitochondrial DNA. You understand? Or Y chromosome. But Y chromosome, we don't talk about it anymore. So here they brought the example and they wanted the evidence. Therefore, mitochondrial DNA belongs to genetic evidence. Is it clear? Yes. So D becomes the most correct answer. Hey, let me see in the comment section. People are busy talking. All right. Don't lose hope. You'll make it. Yes, don't lose hope. You'll make it. All right. We continue the next question, which is which option in the table below shows the correct comparison between mitosis and meiosis. Which one is the correct comparison? If you look at meiosis and mitosis, they are looking for which one is the best. Okay, we find out that mitosis produce fodora cells, that is out. So it means that this is also out, because once one is wrong, then the rest is wrong. Produce genetically different, that is out. Also is out. Chromosome number remain the same. Yeah, looks to be good. Division occurs 2 times. Nope, it is 1. Eh? Do we have mitosis 1 and mitosis 2? Nope. We have mitosis. We don't have mitosis 1, we don't have mitosis 2. All right. will result in 4, so meaning that this one is also out. However much it result in 4, not 4 division, 2 division. Yes, therefore that one becomes the most correct answer. So the answer becomes to be C. If you have a challenge, let me know in the comment section. Comment section. You know, some people, they just comment. Nah, your answer is wrong. No, man. Check yourself before you say, sir, your answer is wrong. However, if you see that is wrong, yes, sometimes when you are rushing and busy talking, you are gossiping, you end up saying something different. All right, let's continue. Which one of the following science discovered Tom Child? This one I've been saying it that they like it. I don't know why they like it. Is Raymond Dart? No discussion about it. If you want more, you go to the distinction material by M. Saidi, grade 10, 11, and 12. All right, which one of the following is produced at the end of transcription? I said it. I said that end of transcription. What is transcription? Transcription is the formation of the polypeptide chain. But if the amino acids are more than 50, therefore it becomes a protein. And then translation, what did I say? Then transcription is the formation of the messenger RNA. Translation is the formation of polypeptide chain. So in this case, polypeptide chain, if it's more than 50 amino acid, therefore it becomes a protein. So if I see a protein or a polypeptide chain, I go with that answer. So A out, B out, C correct, D out, meaning that C becomes the most correct answer. They say the translation. They are not saying transcription. Transcription is the formation of the messenger RNA. They are saying translation. Thank you so much, guys, who got 20 out of 20. If you got 20 out of 20, congratulations. Who got 20 out of 20? If you didn't get 20 out of 20, still fine, because I know that you're gonna make it in other questions. All right, we continue to the next biological terms. When you go to the biological term, the nucleic acid carrying hereditary information, we talked about it. It is DNA. We said yesterday the functions of DNA. We said that it carries genetic information. Yes. So now they brought the question, and then I think people who watched our prediction, who watch our predictions, you don't regret. Even ask other grades. Some grades, even you bring exactly the same question paper, the way we have predicted it, the way we have set it out, and we release our prediction sometimes 2, 3, 4 months, yes, in advance, so that there is no doubt. You can't say that sir copied or what. Yes, it's because of our analysis, our detailed analysis, and you see the paper is exactly the same. All right, thank you so much. Let's continue. Chromosome condition of the cell that has a single set of chromosome. We call them haploid because the single set, which means half the chromosome number. Therefore, it becomes haploid. Yes. Then they're saying that the structure that hold the two chromatids of chromosomes together. Two chromatids of, check like this, that they're looking for that central part, which is centromere. So that the answer, the type of RNA that carries specific amino acid to the site, according to the messenger RNA codon, the transfer RNA with the complementary anticodon brings the required or specific amino acid to the ribosome. You see? So it becomes tRNA. The transfer RNA brings the required amino acid to the ribosome. You see? Correct. We got the next question, guys. We got the next question. What is the next question talking about? Analysis of DNA sample to identify individual or relationship between individuals. That is DNA profiling. DNA analysis of DNA sample to identify is analysis. They are talking about the technique which is used. So it's DNA profiling. Yes, not DNA profile. There's a difference between DNA profile and DNA profiling. Yes. The next question, they are saying, the group of similar organisms that are able to interbreed to produce fertile offspring. What is the answer there? Is a species. I've skipped. the type, the arrangement of number structure of all chromosomes. If you talk about number and the structure of all chromosomes in the nucleus, whenever I talk about the nucleus, you come with the word karyo. The arrangement, I don't want to go to the distinction material book now to show you exactly. Everything is stipulated there. Yes, karyo. So number structure, so we need to know which type of nucleus is that. Therefore, it becomes karyotype. Which type of nucleus is that? Hey, we got the next question. A group of similar organisms that are capable of interbreeding to produce fertile offspring. So we say that is a species. Yes. Then we are saying that we go to the next question. Yes, we got the next question. Biological species is good. Biological species, as long as they see species, yeah, you get it right. Sir Saidi, I say DNA profile. When you say DNA profile, is a different story. So it will depend on them. You know me, when I'm talking about, I only talk about KO. There are some other answers they accept, you understand, but when I'm teaching or when I'm trying to give a memorandum, I only give out those which are straight, but the other answers they discuss about and then they give you. You understand? Yes. So don't be too like you are too scared. Your answer is not is exactly the same. We are not the same people. You understand? I can't write exactly the same as you. So DNA profiling is a technique. DNA profile is the technique. When you use the technique, what you get is the DNA profile, but it will depend on them. So the correct answer here, the most correct answer, that's why I say the most correct answer is DNA profiling. Yes, I think even they can also give you DNA fingerprinting. Yes, we sometimes call it DNA fingerprinting, but I prefer calling it DNA profiling. Then they're saying that, is there any question they are saying? The phase of meiosis where paired chromosomes are arranged at the equator. The phase of meiosis where paired. You see the word pair, guys. Hey, you see that word pair? This word is a key point. Then chromosome, meaning that they are not single. They are in homologous pairs, meaning that they are like this. They are like this. They are looking for something like that. How do you call that phase? So that is mera, because it is in the middle. Mera phase at the equator, mera phase. But because they're in pairs, therefore it becomes mera phase 1 before they divide. So the most correct answer there is mera phase 1. Is it clear? Then they're saying that the biological process that produce genetically identical organisms, we said it, cloning. So guys, I think you got it right. Who got 9 out of 9? Guys, let's be honest. This year, life science was fair. Based on what we are discussing so far, life science was fair. You can't compare life science they have been doing and life science of today. So we expect to be the best performed subject in the world. All right, we go to the next question. The source of variation in population. Yes, source of variation in a population. Random fertilization, random mating. Yes, this is both A and B. Random mating, random fertilization. Yes. So the answer is A, both. Let me write. Don't ask me questions here. Both A and B. Now someone is going to say, what if I say both? What if I say A and B? I say the most correct answer is both A and B. Who discovered the structure of DNA? Yes, who discovered the structure of DNA? Here, who discovered the structure of DNA? I said it. James Watson and Francis Crick. James Watson and Francis Crick. They're saying who discovered the structure of DNA. So the answer is none. Yes. They're saying that Rosalind Franklin and Maurice Wilkin, yes, they helped in the discovery of the structure of DNA, but it's not them who discovered based on the books. You understand? However much it's them who discovered the structure of DNA using X-ray diffraction pictures. You understand? Hey, they just say, once they say, I said it yesterday, that once they say who's discovered the structure of DNA, you just talk about James Watson and Francis Crick. Simple. Hey, so the answer is none. Occur during meiosis, karyokinesis, division of the nucleus, cytokinesis, division of the cytoplasm. Both A and B. Because the nucleus divides, that's why you have half the chromosome number. Yes, cytokinesis occur, that's why you have different cells forming. Therefore, the answer is both A and B. You understand? So that is it. I think we got it. Who got 6 out of 6? Someone saying simple. Hey, yo, I told you. Whom did you tell? All right, guys, I see you guys. You are getting good marks. I love it. I love it. I love it. I love it. Hey, I love it. Let's continue the next question. We talked about this yesterday, and even I showed you a simple diagram. Oh my God, I love you guys. Hey, oh, and I love this examiner today. Yeah, you just made my prediction on point. Thank you. All right. The diagram below represents the DNA molecule undergoing an important biological process. Yes. Hey, what is, I told you this. People, if you messed up when I emphasize this, hey, I'm going to fight you. Okay, identify the process shown above because we are forming two new DNA, therefore is DNA replication. I expected everyone to get these answers. Okay, chemical bond 1. I said we say weak. Don't ask me that if I didn't write weak. Weak, I told you yesterday. Weak hydrogen bonds. Yes. I said it in the last minute, what called the last I or last minute, that emphasizing all this. All right. Thank you. Collective name, that the same thing. We talked about it. Collective name for 1, 2, did I say 1? For 2, 3, and 4, they are looking for this. How do you call that? We said it's called a nucleotide. Good. Full name for nitrogenous base X. Oh, guys, some people lost marks here. They're saying 5. 5 is the same thing like, thank you so much. So, remember this one goes with this. So, if this is adenine, therefore this is thymine. Therefore, they are saying, if you wrote this, the answer is wrong. I'm sorry. Is wrong. Don't discuss. Don't even put it in the comment section. Is wrong because they say the full name. So they want thymine. So they want you to write the whole word, not short form. Nope. Thymine. Simple. During which phase of cell cycle we said it? During which phase of cell cycle does it interphase? Interphase. Guys, if I told you, if you want a distinction material versus Thundereduc. Simple. Done. Life becomes simple for you. All right. Where is, I think I should even go for a holiday after this paper. Because the happiness I have in the heart, I hope is the same happiness you guys you have. Where in the cell does this process occur? Where in the cell? We say that it occurs in the nucleus. Guys, you make me beat myself, the tongues and stuff. Check what is DNA replication. So you identified it. When does it occur? Takes place. You see the answer is there. Where does it take place? We said it occurs in the nucleus. Why does it occur? I think I also saw that question. They're asking you, why is DNA replication occurs? What is the problem? What is the problem? Okay, we got the next question. We got this. We got the next question. Thank you so much. All right, in certain species of butterflies, one gene control wing spotted color and another control eye color. All right, wing spotted color and eye color. So they're saying that the wing spotted color is red. Meaning that if we come here, we shall say red is capital letter R, or gray. Gray meaning that is small letter R. And then while the color black is capital letter E, and brown black. And then brown is capital letter E. All right. So now, since you know that, then we can answer the question. Name the type of cross represented above. Because we are involving two characters, therefore it's gonna become dihybrid cross. Yes, dihybrid cross. They're saying name the dominant characteristic in each. The dominant characteristics is red, and then it's black. Yes, it's red and black because they said in each, meaning that red for what? The red is the color of what? Color of the eyes, and then black. Red is the color of what? Spotted color, okay. Yes, of spots, and then brown. It's there. Red, and then black. We continue. We go to the next question. If you wrote the answer as scratched it with a pencil, if you wrote the answer and scratched it with a pencil, Nana, as long as you scratch it. My wife was acting up. Who is your wife? Hey, guys. Okay, let's continue to our discussion. I thought that you have question or you have some addition or what. This one saying I wrote hydrogen bond. Hey, Wena, it means you are not here on our discussion, on our last minute and prediction. But maybe they will give you. Don't stress about it. Maybe, I'm saying maybe. All right. The table below shows the phenotype and all possible genotypes of offsprings. Okay, you have red and brown. Red is spot. Oh, yeah. Red is for spot and brown, and then black is for spot. Yes. Red and brown. All right. Here you have possible phenotype. They are saying all possible genotypes. They are looking for all possible. So if you have red, so you can be red, capital R, isn't it? Brown, remember it is recessive, so can be capital R, or saying red and brown, capital R, and then small e. Or it can be capital R, small r, and then small e. These are the two alternatives you're supposed to talk about. Yes. Then we got the next thing. The next question. They are saying that all possible phenotypes of Y. Capital R is red, so it means that Y is gonna be red, and then capital E is black, so it's gonna be red and black. Yes, I think we got it. Someone is asking, where is the 4 marks? Why 4 marks? 4 marks, they will mark this, they will mark that, they will mark that, they will mark that. I hope if you get this right and this wrong, you lose one, you get another one. You understand? Or they can use the all or nothing law, meaning that you either get it all the 2 marks, or you lose all. You understand? Yes. If they are sympathetic to you, then they will say that capital R is a mark, so that you have 4 marks. Then here is black. They are saying black, red and black. So because it is simple, it's easy to get the colors. So this one looks to be a little bit a higher order than this. That's why they give this one more marks than this. So 1 mark. The genotype of gametes produced by butterfly with gray spot and brown. We talked about this. Gray, so we have to look for gray. Gray is recessive. It cannot appear unless it is small r. And then brown is also recessive. It cannot appear also unless it is small r. So it means that all the gametes will be the same. Small r, small e. So that is the answer. Therefore, you get the what? Some people I know that they wrote small r, small e, small r, small e because there are supposed to be 4 of them. Small r, small r, small e. Still you get a mark. Don't stress about it. Yes. But if you wrote just small r, small e, still you get a mark. Yes. Thank you so much. Oh, the question is done. Oh, it was so nice. 9 out of 9. Who got 9 out of 9? 9 out of 9. Someone saying I have got 47 out of 50. Congratulations. Thank you so much, guys. I see 42, 31. Yeah, I see 48, 40. I'm looking for 9 out of 9. Thank you, thank you, thank you, thank you. Yeah, I told you yesterday that you guys don't stress. Paper 2 is gonna be very simple, simpler than the paper 1. People are saying, sir, don't say, I know what I'm talking about. Don't ask me how did you know. I know because I know. I know because I teach you. Yes, I know that what I've taught you, it will come. Unless I've not taught you, then I'll tell you that it's going to be more difficult. But if I've taught you, then I know that, oh, it will be simpler because I've talked about everything. Let's continue. Question number 2. Let's hunt for 50 marks again. Identify molecule Y. Molecule Y is this, which is the messenger RNA. If you wrote RNA, it's 0. It's supposed to be messenger RNA. Describe the process resulting in the formation of Y. You see, I told you yesterday that they're gonna ask you this. Describe the process which result in the formation of Y. You understand? Formation of Y. You mentioning that this is transcription is the first mark. Please, I said it. First mark, because they didn't mention it that it is transcription. So mentioning it, a mark. And it might be a compulsory mark that if you don't mention it, you lose a mark. You understand? Yes. So transcription is number 1. Then describe. DNA double helix unwind, weaker region bonds break, then it unzip to form 2 step. DNA double helix unwind, weaker region bonds break is the same thing like unzip if you use that. Forming 2 separate strands. I said you have to say 2 separate strands. That separate must be there. One acting as a template. That's what one acting as a template is the marking point. For formation of messenger RNA using free floating RNA nucleotide from the nucleoplasm. So you describe that process. Everyone knows that. Yes. So even if I don't go through it, these are the process. I say that if they don't bring this, they bring this, they bring this. Simple. Give one so I'm not going to waste too much time there. So that process, if you don't know, you go back and then you check about it. As long as you know that that process was transcription. Give one structural difference between molecule X and molecule Y. Molecule X, where is molecule X? Oh, they are looking at the difference between DNA molecule and RNA molecule. That's what they are looking for. Did you fail it? They are saying structure, something which is structure. For example, it's double-stranded, it's single-stranded. It has thymine, it has uracil. Those one, differences between DNA and RNA, as long as it is not functional. You understand? What that DNA carries genetic information, RNA carries coded information. You see? As long as it is structural, what you see, what is made up of, then they will give you a mark. Yes. Then they're saying that someone is saying that messenger RNA didn't have to say messenger RNA. mRNA still is fine. Yes, mRNA is fine. If you wrote messenger RNA is still fine. If you said messenger ribonucleic acid, it still is fine. Give the sequence of base Z on strand 2. Sequence of Let me clean up so that yes, they're saying give the sequence of bases at triplet Z. So triplet Z is this. Let me first clear up here. Triplet Z is here on strand 2. Strand 1 is here. Oh, so they are looking. Give the sequence of triplet. They are looking for DNA because strand 2 is DNA. Give the sequence of triplet Z. Okay, triplet Z is this. Oh, is this. triplet Z on strand 2. Okay, so if messenger RNA is this, so what is DNA? So it's gonna be, let's put it here so that you see it clear. Guys, here there is a simple trick. I think most of you got it wrong. Let's find it. It's supposed to be okay. If this is messenger and this is DNA, ne? And this is DNA, you understand? And this, because it's facing this side, it means that this was the template. Therefore, U goes with A goes with A goes with the T, and then T goes with the C, G. You understand? So if this is the first strand of DNA, what will be the second strand of messenger? Therefore, it's gonna be A goes with the T goes with the A, and then G goes with C. So the answer is TC. Oh my God, some people didn't get it right. Some people didn't get it right. TAC. The answer is TAC. Yes. That's why I said it's a little bit complicated if you don't know how to get it. Someone is saying how. Eh? Should I repeat it? It killed you. Sorry. Okay, let me do it again. Ne? The trick was simple. Do you know what is the trick? So that you are satisfied. So, this is messenger. This is messenger RNA. This messenger RNA is facing that side. It means that this was obtained from this DNA. Meaning that if this messenger is U, then this side is gonna be what? Is gonna be U goes with T. U goes with A. You understand? A goes with T, and then C goes with G. So this is DNA, the first strand of DNA. Then the second strand of DNA. Remember this strand, let me call it strand 1 and strand 2, they are complementary. So I need to find out what is this complementary. So A will go with T, and then T will go with A. Are you seeing? And then G will go with C. That's why we say that this strand is TAC. I told you that something which looks simple in most cases look to be difficult if you don't read the question several times. I said read the question 3 times. It shows that you are so sure of your answer. All right. We continue. Give the structure difference. We are done. We are done with that. The table shows some of the amino acid coded for by codon on molecule Y. By codon on molecule Y. So we come back here and we look for molecule Y. Okay. Molecule Y is a messenger RNA. All right. Remember I told you that please make sure that you know how to change the codes. Code, codon, anticodon. So mRNA, molecule Y is a messenger RNA which has what you call codon. What is here is a codon. So meaning that I don't need to change anything. So they're saying identify the first and the last amino acid coded by this section of molecule Y. The first and the last. Because this, you don't need to change anything because this is messenger. I told you that and I said, please, I beg you, always correspond with this. This is codon and then this is also codon. So you are changing it to what? You are changing it to what? To anticodon. Here there is nothing like anticodon, so it's codon versus codon. Therefore it was very simple. You just read it the way it is. AGA, you look for AGA, which is arginine. So the first codon is arginine. Then the second one is CCA. I look for CCA, which is proline. Simple. The first one is arginine, the second one is the third one is proline. So simple. Arginine and proline. Haibana, bafetu. Yes. So that's why I'm saying that something which look to be simple in most cases tend to be difficult because you don't give it time because you see it easy. All right, we continue. And we got the next question. I hope you guys you got 12 out of 12. We got the next question. 2.2. The diagram below represent the phase in meiosis, which is the meiosis 2. So we don't know what is it. So we are just going to meiosis 2. They are saying that the diagram below represent phase of meiosis. Identify the phase of meiosis shown and above. So this is because this is, I told you, you have to rotate it. I told you that please rotate it like this so that this thing, they look in different angles. So the answer is, you see, it's separating, you see. So it is anaphase 2. You don't need even to think whether it's anaphase 1 or anaphase 2 because they said meiosis 2. So it's simple. Yeah, so it is anaphase 2. So the answer is identify the phase anaphase 2. State one difference between phase shown in the diagram above with that of meiosis 1, meaning that they want a difference between anaphase 1 and anaphase 2. For example, anaphase 1, homologous chromosomes separate opposite poles, while here the chromatids of the chromosome separate opposite poles. You understand? Yes. So you look at the difference between anaphase 1 and anaphase 2. That is the answer. If it is there and I've not talked about it, yes, as long as it is correct. State one difference between, okay, we're done. Identify structure A. Structure A, guys. Structure A is centriose. Centriose. Someone is going to say, what about centrosome? I don't know. Yeah, centriose. This is not a centrosome, is a centriole. So if you wrote centrosome, it will depend on the examiner. Then B is spindle. Yeah, spindle fibers or spindle threads. Yes, or spindles maybe. I don't know. But spindle fibers, spindle threads, those are the most correct answer. Describe the role of structure B. Yes, this one in cell division. They didn't say in this phase. They say in cell division, meaning that they hold the chromosome in position during metaphase, so that yes, one is on the different poles. You understand? Yes, along the equator. Or it also is very important in pulling the chromosome to opposite poles during anaphase. So you can talk about during anaphase, what is its function during anaphase. You can also talk about its function during metaphase. You understand? Because they didn't talk about this phase, they said during cell division. So it holds the chromosomes in position, yes, along the equator, so that now chromosomes are on different poles. You understand? Yes. Also, they contract and then they pull the chromosome to opposite poles, or chromatids to opposite poles. So as long as it is linked to that, I think you will get a mark. Sir, please predict Africa. Please predict my guys. Don't say that. I can't predict your future. Your future will be bright. I'm telling you, God willing, your future is gonna be bright. Yes, you'll be bright, you'll have a lot of money, you'll enjoy life, you'll go outside the country, you enjoy different places. Yeah, all right, let's continue, guys. Draw the this is what I said. I want ish, man. Draw a labeled diagram representing the cell above, as it would happen. It would appear. It would have appeared in metaphase 2. Metaphase 2. Metaphase 2. Right now, guys. I think I saw it. Yes, this time they made it clear for you, but in most cases they don't make it clear for you. So they made it clear and they said that make sure you have the correct shading. So a wise person, if you don't know, would have done this. If you don't know, but if you know, then it's fine. Then you turn this paper like this, because now they said what? Which phase is it? They said in Mera phase 2, meaning that I just bring it back. Yes. So these chromosomes were like this because the spindles are like this. You understand? Meaning that this was one chromosome and this was another chromosome. So you guys, you put the caption. So that is the first mark. The second mark, you draw this chromosome whereby you bring this chromosome together, meaning that that chromosome was like that, all of it is black, and then another chromosome was like that, chromatid, but this one is shaded. All of it was shaded like this. So that is the first mark. Yes, it was shaded like this. So that variation was very what? Was very important. Yes. Okay, let me write it like that. It doesn't matter, I'm just drawing so that then this one, so this one is done. Then this one, you come back, you also draw it. Hey, I used a different color. It's fine, but it's supposed to be the same color. Yes, central. But now check this thing is this side, is not this side. So I shade it. I only shaded that part. You understand? Yes. And then now, because they said is meta phase, then you draw that. Holding, you see the function of the spindle fibers, they hold the chromosome along the equator. So now you tell us this cell membrane, then you tell us is centriole. As long as you have 3 points correct, you tell us this is a chromosome, then tell us this is a centromere. Yeah. So that's it. That's what they wanted. Simple. Yeah, as long as you have, if you didn't draw the shadings properly, corner beast, yes, it means that you lose 1 mark. So you keep on losing 1 mark. But if the labels, as long as maybe they are, how many marks is it? Is 5. Maybe they will give you 2 labels, any 2 correct labels, and then they give you the shading also. Then they give you a caption, so 1, 2, 3, 4, and then giving you a mark for the correct what? Correct phase, meaning that it is under meta phase. So basically that is how it's supposed to be marked. Then we continue to the next question. I think, guys, you got all those 5 marks. Then we got the next question. Blood group. Yes. We got the next question. They are saying that the table below shows the percentage of blood donor for each of the group in South Africa in 2018. They're saying that according to the data, yes, which is the most common blood group among the donors. If you come here, you'll find out that this is blood group A, blood group O. Yes, remember, even before you look at it, we say that what is blood group which is regarded as universal donor, meaning that it donates more. It's blood group O. Even according to the data, it shows that the most common blood group that donated a lot is blood group O. So answer is O. Or you say blood group O. Name the type of dominancy shown in the inheritance of blood group A. Blood group A has complete dominancy. Why? Because it can either be capital A and then small a. This one is completely dominant over that. Yes, so it is complete dominancy. Hold the chromosomes toward the opposite poles, as long as it has the word hold the chromosomes. Yes. Then they are saying that you did it by accident. All right, explain how is it possible for a man, even this one I said it yesterday, a man of blood group A and a woman of blood group AB have child with blood group B. You understand? Man of blood group A marries a woman of blood group AB, and then they have so if they say explain, here I think they will give you two different style. You can explain by writing either in bullet form or paragraph form, or you can explain in terms of genetic cross. And after giving the genetic cross, you conclude. Yes, meaning that you conclude. For example, you can say that blood group A and then blood group AB, meaning that you can say P1, you say phenotype, then you say that blood group A heterozygous, blood group A, because blood group A. Yeah, so you can explain, or you can use blood group A. You understand? Blood group A, and then crossed with blood group B, AB. You understand? Then the genotype, genotype, then you say blood group A, heterozygous, then blood group AB. Then meiosis, you have blood group, let me just say it like this, blood group A, blood group A, and then allele for A, and then allele for O, and then allele for A, and allele for B. And then after that, what do? You say meiosis, then you say these are gametes, then you tell this fertilization, then you told us this is this A, and then this with this, you say A, B, then this with this, you say A, and then this is this, you say A, is B, and the recessive, this is offspring, and then this is the F1 generation. So what was the question? Explain how is it possible for blood group, a person with blood group A and a woman, yes, blood group AB have a child of blood group A. So this is possible because the man of blood group, therefore this is blood group B. So you can use this method, or yes. Did I ask the percentage? Nope. How is it possible? How is the possibility? Meaning that you have 25% chance of having a person of blood group B. Meaning that you can explain in terms of this, the cross, and then the recessive allele from the blood group A will be passed to the child from the mother. Was it a father or mother? Who is blood group A? A man. From the father will also join the dominant allele from AB, from the mother, meaning that the mother will give the child AB, will give the child B, while the father will give the recessive allele, and then when they join, you form blood group AB. You form blood group B. You understand? So you can explain in terms of the genetic cross, or you can explain in terms of writing blood form. Still, they will look for those points and then they will mark you accordingly. Yes. Then the last question on this. This question was a little bit simpler. Plot a bar graph of the data. This one, just plot the bar graph. Bar graph, you're supposed to put the caption. You're supposed to put the bars. They say that the table below shows, I say the ID. Meaning that the blood group is the independent. So this is a blood group. Yeah, he said ID, which means XY, the study. And then they say that this is 0, maybe you say 10, 20, 30, 40, 50, and then this is the donor. Then you put in percentage. You understand? Then after that, you start to plot blood group A, blood group O, blood group B, A, blood group B, and then blood group AB. You see? This you supposed to have equal spaces, you understand? Equal bars. This one must be indicated, and then this you say A, or this is A, this is B, and then this is A, B. Then you say blood group. Here you're supposed to say donor, then you put in percentage. Donors in percentage. You put the caption here. Some people, when they draw, they start on the x-axis, like on the y-axis direct. I hope they also mark it. They also mark it. Yes, but it's advisable to leave that space which is equal. But sometimes when this one is, they also mark it as long as you leave a space there, and then the other bars they have that space. So that is it. Question 2. We are winding up with the question 2. Then we go to the next question. Explain in words. I want to see how far. Question 2. Muscular dystrophy is a genetic disorder condition that causes muscles to weaken over time. It is caused by a recessive allele on X chromosome. Yes, recessive allele, meaning the dominant allele is this, resulting in normal muscle functioning. All right, they're saying that how many offsprings do have in individual one have? Offspring individual one, the same question. So those are the one, meaning that the answer here is 3. Genotype of 2, because they produce a kid who has it, and this is a female, therefore is X is a female, but they produce a kid who has the problem, meaning that is a small error, has a small error, and then this one, because this individual is normal, therefore is going to have a capital D. So that is meaning that it's going to be capital D and then X small d. Explain more males have. This is the same thing, and then I used hemophilia, meaning that X female, and then XY male. That females have 2 X chromosome. If a recessive allele of this disorder, yes, you mentioned the disorder, appears or goes on one X, it can, if a recessive allele appears on this one X, it can be masked by this dominant allele on the second X. So the individual is still normal, the one which we call normal but a carrier. But males have one X chromosome. If a recessive allele appears on the X, the individual is sick with this disorder. Don't just say is affected. Affected with what? You have to be saying it that is affected with the disorder. With that disorder, yes, muscular what? Yes. We got the next question. Yes, the next question is saying, someone is saying how about XDD? XDD, it can't be XDD. You know why? Because they produced a kid who is X small d and then Y. Meaning that how did they get this if this person is X capital D, X capital D? You understand? So this can't be applicable. That's why we say that is X capital D, X small d, so that this small d, yes, this X can go here, and then this Y came from the father. Yes. We go to the next one. Parent 5 and 6 plan to have children, another child. Use the genetic cross to show the percentage chance of having a child with this. You see, it's the same story again. Parent 4 and 5. What is parent? Another trick. I'm saying that. Just a moment. Guys, is the network fine? I just want to first confirm. Um, all right. Yeah. Sorry, there was some bit of network issues. So they are saying that yes, genotype. Genotype in this case, what is the genotype here? You'll find out that the genotype here is 5 with without a female without this, and then meaning that 5 without that crossing with the 6, a male without still the disorder. You understand? Then you go to the genotype, so you write it here in full, just that the space is too small. Let me this side. P2 F2, you're saying that phenotype. Now we said parent 5, you tell me what is it here. Whereby the genotype, the parent 5, because they produce kids who are having issues or with the disorder, meaning that parent 5 is X, capital D, X, small d, and then crossed with parent 6 with X, capital D, Y. Then you go to the next, which is meiosis. Meiosis splits X, capital D, X, small d, then here X, you go to the next, you start to cross. You can use the Punnett square, or you can just do capital D, then this and this, Y, then this and okay, guys, sorry. We are just switching the networks, because some people say that the network
Recording in progress.
they are saying that the network was not right. So I think the network is fine now. All right, we go to question number 3. Read the passage below and answer the question. Genetically modified maize, yes, bacterium, bacillus, yes, produces a toxin. It produces a what? A toxin. Okay, yeah. Produces a toxin that kills insect. Yes, this bacterium is used to genetically modify maize to certain to contain Bt toxin. This Bt toxin is toxic to the insect. Okay, describe how Bt maize is genetically modified to insect resistant. Basically, what they do is they have to remove get the what, the gene from the bacteria. Yes, then they insert it in the maize. You understand? So that now the maize starts to produce this gene, and then it is able to fight against this. For example, you can say that a Bt gene from this bacteria bacillus is inserted into a maize genome using genetic engineering. You understand? Yes. So we use the genetic engineering to do what? To 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 do that. So the gene of bacillus, the one which is resistant to or which produce the toxins, yes, is inserted into a maize using which method? Using genetic engineering. So removing the bacteria, the gene which causes toxin, inserting it in the maize, all those are marks, and then which using which method? Biotechnology, sorry, not biotechnology, genetic engineering, meaning that we manipulate the genes. As I said yesterday, that whenever you see that you are manipulating gene, you are touching genes, so the method you are using there is what we call genetic engineering, because engineers, they manipulate. Yes. All right. Explain 2 reasons why farmers might want to grow Bt maize. The reason why farmers they want to grow Bt maize, first of all, um, uh, uh, um, remember Bt maize, we say that it produces this toxin. So this maize will reduce the pest damage. You understand? Yes. Also decreases the reliance on chemicals. You understand? Yeah. chemicals, chemical like the insecticides, because it is resistant, you understand. Therefore, first of all, it's gonna reduce this maize from being attacked and damaged by the insects. That's number one. Number two, it's gonna, so it increases the yield, you understand. So it increases the yield of the maize, you understand. So the farmer gets more profits. The number two, you can also talk about the farmer not using the chemicals, you understand, saving money, you understand. So you don't use these chemicals which you call the insecticides, so the farmer will be saving money. So meaning that you'll be making, at the end, you'll be having a lot of money which has been saved. So you explain, I'm not saying that this is the conclusive answer, meaning that you have other ways in which you explained it, you understand. And when they look into that answer stands to be correct. Is it clear? Yes. So that is it. Someone is saying 2.44. We did this 2.44, guys. We did the genetic cross here, and then we ended up having 25%. All right, then we go to the next question. Yeah, the phylogenetic tree below shows the evolutionary relationship between the sum of the species. Give the letter representing the common ancestor of all species shown in the phylogenetic tree. The letter is V, because everything goes there. This one, when it came, it's split, you understand. Yes. So they keep splitting, you understand. They keep splitting. Then they're saying that if identify the species that is most closely related to old monkey. Old monkey, what is the most common recent ancestor of old monkey? What is the most, if you look at it, you have this and you have this, you understand. Yes, so this is a ancestor of old monkey. So if you go back, you'll find out that the most recent common ancestor is this, meaning that this monkey shared a common ancestor, the most recent, with this gibbon, meaning that this and this are most closely related. So the answer becomes the gibbons. Hey. Then they're saying that give a reason for your answer. Yes, we say that because gibbons share the most recent common ancestor with the old monkeys. The most recent common ancestor with the monkeys. Identify 3 species that share the common ancestor with R. So you come back here. R. This is R. So meaning that the chimpanzee, the gorillas, and the orangutans, okay, whatever. So state 3 characteristics of upper limb that humans share with the group P. Group P is this. So upper limb here, guys, you are looking at what? You are looking at upper limb. Only upper limb, meaning that the lower limb. So you talk about opposable thumb, you can talk about nails instead of claws, you can talk about free rotating arm, you can talk about elbow at 180 degrees. So all those ones, they are talking about the upper limb, meaning that they are talking about only this. Yes, and the others, but they are supposed to be linked to the lower limb, meaning that the upper limb, your hand, the hand. Yes. Hey, someone saying human is correct. What is wrong? The more what is the most common recent ancestor? No, man. Yes, recent ancestor. You see, why do you talk about human? How is human connected to this? No, recent ancestor. You see, where they meet. Yes, meaning that those are the one which are most commonly related. But now these ones are This one, if you look at this is most recent with that. You see, now this and this, you see, humans and that, you see, this and this, then this and this, you see, yes. So you see, and then now this and this, you understand? Then this is the far. Are you together? Yes, that's how we do them. All right. Then we go to someone even saying how humans. Yes. All right, guys. Sir, will you predict geography? Definitely. It's already out. It's already out. Geography is already out. Yes. So just in geography and then put Thundereduc. Right. Then they're saying that a group of researchers conducted an investigation to determine the influence of the type of milk on the height of children drinking milk. The investigation was conducted over a period of 7 years. All these are answers, you see, over a period of 7 years. From age of 3 to 10. They selected the 1,000. This is also, they might, because this is reliability. 3-year-old children are of the same age, you see, same age and gender. Children will children into two groups, splitting them into two groups. One group given 1 liter of milk to drink daily. Another one is given 1 liter of soy, milk to drink daily. Ensure that each group, you see now, why that? Why that? This will help us to improve the validity. Ensure that the same additional diet, similar daily activities, similar living conditions, those are factors which increase the reliability, the validity of the experiment. Provide milk from the same supplier also is a key point. Measure the children's height at the end of the investigation and calculate the average. This one also increases the reliability of the investigation. All right, identify. So you have the below table, okay, 10 years old, okay. Expected average height of a child on cow's milk, average height of a child on sow's milk. All right, identify the independent variable. You go back as I told you that you read the first statement. A group of researchers conducted an investigation. Who? That's the first. To determine, if they ask you the aim, then that's to determine the influence of the milk type on the height of children, meaning that milk type will determine the height of children, or yes. So the independent variable here is going to be the type of milk given to the child, and then the dependent variable, it will be the height of the child, or the average height, because the children, the average height of children. You understand? Yes. The average height of what? Of the children, because they say that on children's, yes, the height of the children is not one child, so they are doing, because they say the average, so average height of children. Now someone is going to ask me, what if I wrote the height of children? I don't know. It looks to be also correct. Some people are saying I'm skipping. I skipped what? 3 point. I didn't skip any question. Guys, did I skip any question? You skipped 3.25. Skipped 3 point. You guys, I don't know why. Maybe you are not, you are not listening nicely. I didn't skip any question. I answered that question, guys. All right. They are saying that name the type of variation displayed by the height of the person. Name the type, because you have short, medium, long, whatever, so it is continuous. And I told you that if you see a character which does not resemble you since you were born, now it's changing, then that is continuous variation. Your height, you're not the same height the same time when you are born, but the blood group you had it, the same type blood group is the same type blood group you're having now, meaning that that is discontinuous. But if it is changing, then if it keeps changing, then that is continuous variation. So the answer here is continuous variation. Yes. Set 2 planning steps. If you talk about the planning steps, basically you select the number of children, ensure that the children have the same age, same race, same gender. You ensure things which you ensure that they, so make sure that you select individuals, yes, large number of individuals, large number of children, you understand. Also, number 2, you can say ensure that all children are of the same age, same race, same gender. They talked about them here. Yes, and I said that this is very important. Yes, they talked about them. Yes, you see. Yes, they talked about them there. Meaning, and also they talked about them here, selecting a large number of individuals. So you are planning. You don't just find the kids and then you just do experiment with them. You have to plan for them. All right. Then you're saying state 3 ways in which the validity of this investigation was ensured. The validity of the experiment was ensured in 3 ways. Number 1, they talked about them here. So meaning that the answer here is that number 1, they use the same additional diet, similar daily activities, similar living conditions. Yes, that was to ensure that the validity is done there. So this is also done. State 1 factor that could have negatively affect the validity of this. What will affect this? The genetics. Yes, you can be provided with all this, but when the genes are not allowing you, baba, baba, you can go to the gym, you do whatever you do, but the muscles can't do anything. The genetics is saying, you are not going anywhere. Yes, you are the way you are. Yeah, that's the genetics. Oh, you can make your muscles, but there is that one muscle you can't make gym. You can't gym it, meaning that the genetics will determine its size. You understand? So the genetics will also interfere with this. So you can say that genetic you can maybe say genetic maybe predisposition. Yes, it can bring about that. I don't know how you put it. Maybe there is also other answers you can also give, and then it will be marked correct. But genetics, the genetic composition of an individual will also affect the results. How did the researcher ensure reliability of the experiment? The researcher used large number of individuals. You see here, large number, 4,146. The researcher, you see, guys, you're answering these things in past tense because the question is being asked. The questions here are being asked in past tense. So the researcher used 4,160 what? 64, 4,164 individuals, meaning that this is a large sample size of children. Then number 2, the researcher repeated the experiment for over a period of 10 years. Repeated the experiment, the researcher repeated. So you see, you repeated the experiment over a period of 10 years. You see that you answering it in past tense, because how did you see all the answers are being asked. Give 3 ways in which the validity of this experiment was. So when you're answering here, you were supposed to say used the same additional diet, used similar daily activities, used similar living condition. So meaning that I told you when I was discussing about this question yesterday that this is the only question they mark grammar. If they ask you in past tense, you have to answer in past tense. If they ask you in present tense, you answer in present tense. If they ask you in past tense and you answer it in present tense, sorry, I don't know what they will say, but I will say for now, I'm sorry, because I rectified it, because I'm trying to say that it must be in past tense if it is asked in past tense. Let's go to question 3.7. State the conclusion of this investigation. The conclusion of this investigation, you see it here. Ne? You see it here. Ne? Which one was higher? So children with the who drank, because they said state the conclusion for this investigation, meaning that children who drank milk, cow milk, yes, they had a higher average height compared to children who drank milk on the children or the child or children who drank soy milk. You understand? So children who drank milk, cow's milk, they had more. They had what? A higher average height than the children who did drink the soya milk. You understand? So that is the conclusion which you can give there. I made a mistake on some of the question. Not that I didn't know the answer. Yeah, I know that those things happen. I know that those things happen. Sometimes when you are rushing, sometimes when you are under pressure, sometimes, yeah, those mistakes are there. Sometimes you can even think that I'm gonna write this and you end up writing something else. That's why I always say that if you do hear this discussion and then you get 50 out of 150, so you do yourself plus or minus 5 marks so that you accommodate there. So you might get more marks or you might get less marks. Yes, sometimes we call it moderation. All right, let's go to question 3.4. Okay, the passage below describes the Homo erectus fossils. Evidence from fossils of Homo erectus show that they appeared in Africa about 2 million years ago. They spread among many parts of Asia. Homo erectus is the oldest known fossil to be fully bipedal. Yes, and human-like body structure. The brain size was smaller than that of human, and the prominent dominant ridge, brow ridge, was present over. Okay, I see something here. It looks to be good. They might ask you, describe how the fossil of Homo erectus are used to support out of Africa hypothesis. Remember, out of Africa hypothesis, we said that modern humans originated from Africa and migrated to the rest of the world. You understand? Yes, during the Homo erectus period. So how is this used? You have to tell us that the oldest, that word oldest is a mark. Oldest, that's what I said yesterday. The oldest fossil of Homo erectus, yes, was discovered in Africa. You understand? Which conclude that modern humans originated from Africa and then they went to the other parts of the world. So that is it. This is what I'm saying. This is what I'm saying. Yeah, this is what I said yesterday. This is what I said yesterday to support the out of Africa hypothesis from our distinction material book. You see, here is the answer. The oldest fossil of Homo erectus, yes, the oldest fossil of Homo erectus, the oldest fossil of Homo erectus. You see the answer here. The oldest fossil of Homo erectus was discovered in in what? Yes, being found in Africa. So that word, the oldest, you see, the oldest fossil of Homo erectus. This word oldest is a marking point. You understand? Fossil of Homo erectus. Yes. Was found in Africa. You understand? Yes. So that's where you get a mark. Then we go to the next question. Describe how all other fossils used to support out of Africa hypothesis. So you come back again. Fossil of Adipithecus was Adipithecus were only found. This word only is a key point. Were only found in Africa. Then you come the fossils of Australopithecus. You understand? Now you tell us either you can say, for example, Karabo, little foot, whatever. Then that's what I said even yesterday. And the Homo habilis were only, this word only is a key point, only found in Africa. That's it. Meaning that you guys, you're supposed to write that answer. So they just split this into just this page. They just split just this page into those sections. Right. Then we go to uh, another another question. Then they are saying that describe how the other fossils were done. Use information in the paragraph regarding Homo erectus and tabulate. Tabulate meaning that you have to draw a table, the features that Homo erectus and Homo sapiens. They want the differences, meaning that you draw a table as I told you yesterday. That draw, you put Homo erectus here, Homo sapiens. Then point number one, I say that Homo erectus, they say that it has a predominant brow ridge. Then you tell us predominant brow ridge. Then here you talk about it has no brow ridge. You understand? Meaning that then you talk about larger brain was smaller than that of human. The brain here, brain size, smaller brain size. Then you talk about large Homo sapiens, larger brain size. I think if you said if you said if you said cranium, maybe larger cranium for humans and then smaller cranium for Homo erectus could also be what? Could also be true. What observation of skull of Homo erectus would have led to science to conclude that Homo erectus was bipedal? So basically, they're saying over their eye socket. So here bipedal is linked because they are saying skull, meaning that the position of foramen magnum was it's moved a little bit forward. You understand? Not that forward than to that of human, but was a little bit forward. So meaning that it was in a forward position. There is no hope for 3.42. Why do you say that? Oh, sorry. I'm so sorry. But we discussed it yesterday and said that, guys, this is how you're supposed to write. But I know that you can't get all marks. You understand? You can't. You can't get all the marks. I understand what your argument, but as long as you get marks which you are supposed to take, you are supposed to go. Because, right. So we talked about, so here you talk about the foramen magnum. Yes. 3.5 is the last question. 3.5 natural selection in wolves. In 1986, there was a huge radioactive disaster in this, and all humans immediately left the area because of the deadly high radiation. Animals exposed to this high radiation developed cancer. Oh, or died. At least 4 years later, 40 years later, scientists have been studying the wolves that live in this high radioactive environment where they are exposed to a high radiation. They found that these wolves have a mutation that make them immune to the cancer. I don't understand something here. We are in 2004. Yes, 1986. When you subtract here, can it make 40 years? Why did they say 40 years? People in the group, can it make 40 years? Why did they say they said almost. Almost 40 years. Okay, Laila. Almost, yeah. Even if it doesn't make 40, then the statement makes sense. All right. Said almost. Cool. Thank you. So almost. So since they said almost, then it makes sense. Almost 40 years. Yes, later scientists have been studying the wolves that live in this highly radioactive environment where they are exposed to high radiation. They found that the wolves have mutation that make them immune to the cancer. Immune to cancer, meaning that even if they have cancer, they are not able. It's not being affected. Yes, being affected by this cancer. So you either have that cancer and become immune to it, or you die. All right, what's a mutation? Is a sudden permanent change on the genetic composition of an organism. Yes, that is a sudden permanent change on the genetic composition of an organism. So we said, remember yesterday, that it can be gene mutation or chromosomal aberration. Down syndrome, we saw it already in the question. What effect does the exposure to sunlight, to high radiation, have on an animal? Here they talked about it. Deadly high radiation. The animal exposed to this, they said, develop. Yes, it will lead to the development of cancer or death. Using Darwin's theory of natural selection, explain the development of immunity to cancer in those wolves. So what are you supposed to talk about here? I just wrote foramen magnum. Okay, as long as you wrote it. Okay. What is the story behind here? Remember this natural selection using Downey's theory. So what are we supposed to explain? The first point is to talk about variation. So there was variation among these wolves, isn't it? So you have to tell us what was the variation. Yes? Some wolves had a mutation, yes, that make them be immune to cancer. Wabona? Some had the mutation, yes, that make them immune to cancer, and the others didn't have no mutation. You understand? That make them immune to cancer. That's pointy. Point number 2, then you tell us that those which had a mutation, yes, they were able to survive. You understand? They were able to survive the radiation. You understand? Therefore, they reproduced in large numbers. They are able to survive the consequences of radiation or cancer, and then they are able to reproduce in large numbers. Therefore, while those without a mutation, they died of radiation or cancer. You understand? Then that's point number 2. Then point number 3, those which survive, they pass their genes to the next generation. Which gene? The gene of the mutant gene that make them to be immune to cancer. You understand? So since you talked about already the mutation, then it means that now they know. Meaning that the favorable characteristic of having a gene, yes, to immune these wolves from cancer so that they don't die, that gene is making them to be favorable. So that gene is passed to the next generation. You understand? I skipped that point. This is called natural selection because they said that one is no longer a big deal, and even they talked about it here. Yes, so they passed that gene to the next generation. After many generations, we will have high proportion of wolves with a mutation. You understand? A mutation that make them immune to cancer than those which don't have a mutation. So basically, that's how we are supposed to explain in that regard. Yes. What if you said immune to cancer and not mutation? Yeah, it could be correct because this question it's a little bit wider. It will have different options. You understand? Don't be stressed. I say that I'm not you and you are not me. You understand? There are some questions which require you to explain based on the scenario which is there. So as long as he's linked to the mutation, those having the mutation, how do they survive cancer and how do they survive these radiations? You understand? So your answer is supposed to be linked to that, but you follow the principle of natural selection, the variation, the environmental condition, competition, yes, those which survive and those which die, yes, then the natural selection will come in, and then passing the important gene or resistant gene or that gene which is favored to the next, or that character which is favored to the next generation, and then telling us that you have a high proportion of this individual who have the favorable characteristic in the next generation than those ones which don't have that favorable characteristic. So it's supposed to be linked to that. I will conclude by saying that I think the paper was fair. I know that question 3 is always like that, but if you look at other questions, because this question 3 requires you to think and write based on what you understand. Who got 130 out of 150? If you got 130 out of 150, 125, 120, all those are excellent marks. You understand? Those who got less than 120 still is good mark. You understand? And that good mark does not mean that I want 120, you also want 120. Even if I got 50, it still is fine because 50 might be taking me somewhere which I want, and I don't need to have 120. But there are some people who want 140, 130, 100 and what? You understand? So all of us, we have different desires for these different marks. I thank you guys. I'm proud of you. Simu. Si sum. Thank you so much, Jordan. Thank you so much, Chilas. Thank you so much. I see you guys. Teki. Thank you so much, Boti. It's me, Sparkstar. Guys, I see you. Thank you, thank you, thank you. Don't forget that tell your brothers, tell your sisters there is this beautiful channel whereby they help students code Thundereduc. Thank you for so much. Now we are going to geography. We are going to geography paper 2 so that you guys will make you proud still in geography 2. You are going to geography 2 with the geography boss. Yes, see you in the next class. Agriculture science, we'll talk about it. There are some subjects which we have not yet started, but definitely we will come in. Thank you so much. Thank you for liking Thundereduc, and then thank you for liking, subscribing, everything. It has been Saidi. If you have any challenge, let me know. Yes. If you have been a challenge, let me know. If you have an answer which is a little bit different from mine, sometimes is as a result of different thinking, but does not make your answer to be wrong. See you again. Hey, when? Does it mean that you guys have gone? Have not? I salute you guys in your future. Have a wonderful, beautiful life. Until we make answers which you were supposed to write. Please take note that this is not the final discussed memo. Yes. So the final discussed memo will be released soon. All right, let's go and then find out what we expected to get in this memorandum.
What we
So let me know
to
if
get in
you can hear me properly. If you have any question, let's discuss about what you have maybe challenges with, so that when it comes to the final exam, you don't miss it out. All right, let's start without wasting time. You know me, I don't go through instructions. You're supposed to do that every time you go through the paper. Saidi, as usual, the first question is saying that various option chart. The diagram below shows the allele. When you say allele, it means this. Allele for the height in the flowering plant, where the tall is dominant and then small t is short, meaning it's recessive. So once you have a capital letter and a small letter, what I told you is that is complete dominancy. But because there are two different types of alleles, there are two different kinds, not types of alleles, so it means that is the heterozygous. So now we come here and then we find out that the plant is homozygous. No, the plant is heterozygous for the height. So that looks to be the most correct answer, which is B. So you get 2 marks there. Then you go to the next question, that which one of the following is not. The key point is not applicable for crossing over. So if you look at what is not applicable for crossing over, genetic material is exchanged between non-identical chromatids of homologous chromosomes. Cool. Homologous chromosomes undergo crossing over in they're saying not applicable for crossing over. Homologous chromosomes undergo crossing over in prophase 2. So we don't have homologous chromosomes in prophase 2. Therefore, and crossing over doesn't take place in prophase 2. Therefore, B becomes the most correct answer, because the key point was saying which is not. Then we go to the next question. They are saying that which changes occur in the sex of the cell, in the gene of the sex cells. These changes lead to changes in the parent phenotypes. No, what happens in the sex cells in terms of genes does not change the genotype of the parent, so this is wrong. Only affect asexually reproducing. No, doesn't affect, because they don't have even the sex cells. Always harmful. Not really can be a basis for evolutionary change. So why? Because this will lead to variation. So D becomes the most correct answer. Then we go to the next question. They're saying that which one of the diagram below represents the different 4 different species of bacteria. Then they're saying that which statement is correctly regarding the survival chance of this species if there is a change in the environment. If there is a change in the environment, so species have the best chance of survival, because it has the most variation. That's what they are saying at A. It could be true, but let's find out if there is the most correct answer. Species C has has the best chance of survival because it has no gene mutation. If there's no gene mutation, then automatically it can't survive. That is too wrong. Neither B nor D will survive because they compete for the same resources. It can't be. They're saying neither B nor D will survive. Neither B nor D will survive because they compete for the same resources. What shows that they compete for the same resources? So it makes it none of these species will survive because bacteria reproduce asexually. They say not really. So the most correct answer becomes A. So A becomes the most correct answer. So if you have any challenge, you let me know, and then I find out that this is Power, the paper was so beautiful. Majority of the things you did on Saturday. Yes, boy, B boy. Yes, I told you that 90% of our discussion is always the paper. So always stick on our channel, you will get distinctions always. And I will salute that boy from Limpopo. I just want to take this opportunity to say thank you so much for my boy from Limpopo who got 140 out of 150. So that's a good. And however, they robbed you with those 3 marks. 3 marks. Their memo, they had some issues of percentage what percentage increase. The formula they gave in the memorandum was not correct, so we claim for those 3 marks. We need them, we need those 3 marks. Yes, I will try to see that that is rectified. All right. They're saying that the relationship between gene and the allele is okay. The relationship between gene and the allele, gene are different form of the same allele. Gene are different forms of the same allele. Each gene are different forms of the same allele. This is wrong. Each person contains two alleles for the gene of one characteristic. Yes. However much the character can be multiple allele, can be controlled by different alleles, but this one, uh, each person is supposed to have two alleles of that character. You understand? Like blood group. You can't be blood group, allele for blood group A and B and then O. Never. You have either A, homozygous, heterozygous, like that. Or if you have for A and B, then it's one allele and one allele, meaning there are two of them. However much there are 3 alleles controlling a blood group, but you only have to be with 2 alleles for one character. That is the key point, and then B looks to be correct. I'm not saying that's the most correct answer. Gene can only have 2 possible alleles. Not related, because we have multiple allele. Because of this word only makes it to be wrong. Allele are phenotypic expression of gene. No, actually this is like misleading. Why? Because it's the gene which determines the phenotype, not the phenotype determining the gene. So B becomes the most correct answer. 1.6, this is a structure which show that there is a divergent evolution. And these structures are called homologous structures. Those people who are using the distinction material book, I think you are very well conversant with this. Study the diagram and the and state the structure below. All right, structure are homologous. Yes, the structure are homologous. This is correct. Animals do not share the no. They share a common ancestor. That's why it is divergent evolution. Four limb of all these animals have the same function. Not really, they don't have the same function, because this is for flying, this is for swimming, this is for maybe making holes and catching rats, and then this is for carrying. So they don't have the same functions. Structure arose due to modification by descent. That's exactly. If you check there, let me show you what I'm saying. Maybe you don't understand what I'm saying here. This is what I'm saying from our distinction material book. This is it. That's exactly, I think if you look at that diagram, it's exactly the same as that diagram. You see, they brought it human, cat, whale, and bat. Human, cat, whale, and cat. I told you, read the distinction material book. Distinction is guaranteed. All right, let's continue and then see. So, and then here we're saying that this is check modification. This is what they are saying, modification by descent. So this is what they are trying to say, that modification by descent. So 1 and 4 becomes the most correct answer. So I have to look for 1 and 4. 1 and 4. 1 is this, and so C becomes the most correct answer. Who got it right? Yeah, who got it that one right? Who got that one right? Okay, guys, let's continue. All right, they're saying that a certain disorder is caused by a dominant allele. I emphasized about this dominant allele. The following combination should be considered. Okay, this and this. Okay, they're saying which one of the following combination arose crosses can result in offspring without a disorder. Certain disorder is caused by dominant allele. So meaning that in this case, we are looking for something which has no. Certain disorder is caused by a dominant allele. So where I have capital F, that isn't it? So when I cross this and this, all of them, some will be like this and some will be like this. So which one of the following combination can result in some offsprings? You see, this is the keyword, some offspring without, meaning that they are recessive like this. So number one is correct. When I cross this and this, I'm gonna produce all of them like that. So number two is wrong. When I cross this and this heterozygous, if this combine with this, I'll form that and that. So meaning that 3 is also correct. If I cross this and this, I don't have any homozygous recessive. So it becomes 1 and 3 and 3. Meaning that I look for 1 and 3. 1 and 3 becomes A. So A becomes the most correct answer. So if you got it right, you need to clap for yourself. Ne? Yes. Hey. Did I say one? Eh? Let me read it again. Someone is saying something. So we are saying that yes, it is one. Let me see, one y1 let me check and see. Yes, well, let me clean here. Y1, because if one recessive allele, my boy, and one recessive allele combine, they form that. And then the 3, if this recessive allele combines with that recessive allele, they form that. And because they say it's caused by dominant allele, automatically it can't be. So it's gonna be 1 and 3, which is 1 and 3 only. So that one becomes the most correct answer, which is A. So I think I'm clear there. Those who didn't understand that concept. We go to 1.8. 1.8, they're saying that approximate, they're saying that they're saying that 1.8 and 1.9 are based on the diagram below. Okay. Approximate how long did the Paranthropus, pan, existed? How long this pan existed compared to its closest ancestor? So it means that how long? How long did this exist? So this is the pan. So it existed up to from there. From there up to there. So what we need to do is we need to subtract the recent ancestor. Where is the recent ancestor? They're saying compared to the closest relative. So who is the closest relative? So if you look at it, this was the relative it had. So it means that we have to subtract this from that, and then we get this minus this, and then we get remaining with that, meaning that that's how much it existed from that more than that point. So based on that, if you subtract, I saw if you drew them correctly, I saw that B was the most correct answer. But you need to use the paper with the ruler, and then you measure properly. Yes. So I'll leave that as question marks if you measure properly. Yes. So if you measure properly, you need to use a ruler. Yes. So that you see exactly if it is pointing here or it is pointing in the 5. Yes. It is pointing in the middle or it's a little bit down. Yes, so your paper will show you exactly what is, but when I measured from the screen, so that what it gave me. So they're saying that how many genera are represented in the diagram above. How many genera are represented in the diagram above? So we say that genera, based on genera, we told you that yesterday that genera, you look at one, that's one genus, you don't repeat this one again, so it's the same thing. So it's one, it's two, it's two, is three, yeah, it's four. I'm seeing Pan 1, Paranthropus 2, Australopithecus 3, and Homo 4. Do we have a 4 there? Yes, so C becomes the most correct answer. Let me check and see if you have any challenge there. Yes, no challenge, and then we continue. All right. So, I think, guys, you got 18 out of 18 there. Yes. So let's look at 18 out of 18. Yes. So they're saying that, or who got at least 14 out of 18. Yeah, because if you got 14 out of 18, you are still a lie. You might still lie in the same distinction area. Terminologies. Allele that does not influence the phenotype of inheritance. We talked about this. It is recessive allele. It only expresses itself in the presence of a dominant allele. Simple. They are saying that the bond that form between the amino acids. We talked about these bonds so many times. We call it a peptide bond. If you wrote polypeptide bond, 0. Portion of the chromosome that code for a particular characteristic. We say this is a what? It's a gene. Biotechnology process that produce genetically identical. We say it is cloning. Yes, we talked about it when you're talking about the cloning of the dolly sheep. They're saying that the type of variation where there is a range of intermediate. We said it, and we say that it is a continuous variation. And I told you, yes, I told you that anything which you are born with is called what? Continuous variation, because it has a range of characters. That's what I say, and they brought it here that they have a range of characters, or what you call the intermediate. Yes. Let me show you what I'm saying. This is what I'm saying. Yes, this is what I'm saying, and this is the example I used when I was talking. That's what they are talking about. And they're saying that the study of past present day distribution of living organisms across the continent. Yes, living organism is bio, then distribution is geography, so it is biogeography. So that's when it's done. They're saying the scientists who argue that acquired characteristics obtained by the parent could be passed to the next generation or offsprings. Who is that scientist? And that is the Jean Baptiste de Lamarck, or Lamarck. Even if you say Lamarck, I think they will give you a mark in that regard. Hey, yes, we continue. Example we continue to the next one. Yes, Danko, Citro. Hey, I don't know how you read your name. Yes, guys. I hope you got 7 out of 7 there. 7 out of 7 there. Then we go to both A and B. Yes, characteristics of African ape. Prognanthus. Yes, they are prognanthus. They have protruding jaws. So they're saying less pronounced brow ridge. No, they have pronounced brow ridges. So the answer is A only. The word only must be there. Characteristics of blood group inheritance. So what are they saying here? Characteristics of blood group inheritance. Multiple alleles. Yes, it is multiple alleles. And then not all the blood groups are codominants. The characteristics of blood group. They are not saying blood groups, meaning that it could be that one blood group. So both A and B makes to be the most correct answer. Yes. Why? Because blood group A, B is co-dominancy. However, blood group A as A is not codominant. It is complete dominant. You see, so it is codominant, but is caused by multiple allele because there are 3 alleles which control blood group. So both A and B, both A and B becomes the most correct answer in that regard. Then they're saying that in a cross between the bull, remember it's a male cow. I don't know whether I'm saying it right. A male more right is heterozygous for long eyelashes. Long heterozygous. The moment you say heterozygous and brown coat, it means that long eyelashes and brown color are both dominant. So meaning that, let me say that eyelashes, I'm gonna say it's capital E, and then color I'm gonna say is capital B, for example. But because it is heterozygous, then it's gonna be capital E, small e, capital B, small b, without a space. They're saying that the cow has short and yes, the cow has short eyelashes and white. It means that those are recessive. So I expect them to be like this and to be like that. They're saying that the gamete of the cow will be the gamete of the cow. In this case, they are looking for the gamete of the cow, not the bull. If they're saying the gamete of the bull, definitely it would have been this. So since it's this and that, so it's gonna be E, it's gonna EB. All of them are going to be EB. EB, isn't it? So because now here's the cow is not having any dominant character, therefore this one becomes wrong also, and this one is wrong already, so answer becomes none. So here it becomes none, here becomes both A and B, and then this one becomes A only. Is it clear? I think, guys, I hope you got also 6 out of 6. So I expected you to get a lot of marks. I think, guys, you are counting and you're also checking and see that things are moving well. Guys, let me know. Let me know what if you have any concern of what we have discussed now. Don't wait when I'm offline and you start to say, sir, now what? Same. How will I say now, sir, when I've already gone offline? It's now downloadable. You understand? Yes. But now I'm on it. Like I'm live. It's not that some of you think I'm not live. Let me show you. Yeah. Yes, you see, I'm live, ne? So let me know if you have a challenge. Yeah, let me know that so that I'm able to answer those questions, or so that you don't repeat the same thing when it comes to the final exam. You understand? That's why I do these memos and then we discuss about them before even they release the final memo. Why? Because I want you, it's like it's me and you writing the same paper. How will you obtain those marks? You understand? So this, if I discover there is a small challenge, definitely that challenge you'll also have it. So we present what you try to make them to be, you're able to pass if that the same questions come back again. Because once they give you a memo, you just look for where you got it right and you won't get enough time to go through it. Anyway, let's continue, and then but I just want to show you that let me know if you have a question so that I'm able to help you out, and then we solve this problem, and then let's go. Hey. Out of Africa hypothesis. This one we talked about it, ne? But now this time, what they did is they stated it for you. They say that out of Africa hypothesis, modern human, you see, I told you the word modern is very important. Next time when they ask you to state the out of Africa hypothesis, you know how to state it. Modern humans originated from Africa and then migrate out of to other continents. So some of the fossil of genus Australopithecus has been discovered in South Africa. I told you don't go without knowing two fossils from at least Australopithecus africanus in South Africa. So they brought Australopithecus africanus, not only Australopithecus, but this one is Australopithecus africanus. Mrs. Place. So give the name of the scientist who discovered that. Who discovered Mrs. Place? It is Robert Broom. And then who discovered the Littlefoot is Ron Clark. So you guys, I had summarized this in your distinction material book. Ne? Yes, I summarized it here. You see, Littlefoot, Ron Clark, you see, Mrs. Place, Robert, you see, they just brought the way it is here from the distinction material book and then they put it. That's it. If they don't ask you this, these are the only things they're going to ask you. If they don't ask you this, ask you this. If they don't ask you this, they ask you this. You just need to memorize this small table. Done. However much other books are having a lot of issues. Anyway, let's continue. State this fossil site where Littlefoot was discovered. Fontaine. So you say Fontaine caves. Yes. So I don't know whether if you don't add the word caves, they will give it to you, but it's Fontaine. Fontaine is Fontaine. All right. Give the species name of fossil, Australopithecus sediba. Yeah, they are looking for Australopithecus sediba, which we call the transitional fossil. Yes. State the fossil site. Okay, we are done with this. Name the genetic evidence support out of. I told you that there are three evidences. I told you there are three evidences. Number one, fossil evidence. Number two, genetic evidence. Number three, cultural evidence. And I say that when they give you cultural evidence, you say examples are tools. When they give you genetic evidence, then you say the example is mitochondrial DNA. We don't use Y chromosome anymore. You understand? So if you wrote Y chromosome, 0. So genetic evidence, which is the mitochondrial DNA. When they ask you fossils, obviously you know the fossils. So you give the what? The example of the fossils. I think I'm clear, and it's clear in that regard. Simple. All right. So we go to that mark. We say the genetic evidence is mitochondrial DNA. Someone can write in the abbreviation. Others can write it here. Sir, please explain the last question. Now you are saying last question. Which question are you talking about? Sir, where can I find this book? Yes, from our link of, if you just check there, you say join, you'll be able to access the book. All right. Let's continue to the next question. That book was called Distinction Material Book with a reason, and every year I update. So don't use the last year's version. You have to use, if you are going to do next year, I'm just talking about next year, use the next year's version. You understand? Because every year is updated. Yes. They're saying the diagram below represents the DNA replication. We did this several times. So they're saying during which phase in the cell cycle does DNA replication takes place? So what they did is those questions I told you yesterday is what exactly they brought here. Yes. So during which phase cell cycle is during interphase. They're saying that natural shape of DNA. This one I emphasize, they say that we call it double helix, not double helical, not helical, not coiled. No, the only statement we accept is double helix, because when this guy who James Watson and Francis Crick, they described it as, check, they said that is DNA is double helix. That's the only with complementary base pairs. It's double helix. That's how James Watson described it, and that's how we're supposed to describe it. So I think I'm clear there. Let me check and see this person who said which question. Safiya Muhammad. Safiya Muhammad Okay, I think I answered you. Ntime. I don't know which question you are telling me to go through or to explain better, unless you point it out. They're saying that give the letter and the name of okay, sharp. State that we are done with this. Double helix. That's the answer. Name letter and the name of for each of the following monomers. We highlighted this several times. We said is a it's a nucleotide. Even I showed you and I said that guys, please make sure that I said that nucleotide, and I said sometimes they call it building blocks, sometimes they call it monomers. That's what I said yesterday. Yes. So I think you got it right. It is nucleotide. You see, they said building block and then monomer. Exactly how it is written here in the, you see, building blocks, and then they say monomer. Are you seeing? Yes. I think now you are getting it right. Please, you want a distinction material book by Saidi. All right, let's continue. Sugar in this one is obviously it's deoxyribose sugar, complementary base, nitrogenous base. Okay, in molecule D, when you look at molecule D, it is you have thymine. Definitely is gonna be what is they're saying the complementary base okay of D. So they are not saying that in RNA what they just want in the DNA, which is the adenine. So you have to write the word adenine. So someone is going to ask me, what about when I wrote capital A? I hope they will give you it right. Yes, capital A, they will give it to you. Then we go to the next question, 1.6, so that we wind up with the first section. Identify which of the diagram A, B, or both A and B belongs to Homo sapiens. Obviously, this is Homo sapien diagram A. So you write diagram A. Hominids. We are hominids. Chimpanzees are hominids. Gorillas are hominids. So the answer is both. I'm sorry if we didn't get it right, both A and B. Yes, so the answer is both A and B. Then describe 3 characteristics of organism skeleton that allow for bipedalism. That's exactly the same way we discussed it and highlighted it and I emphasized it. I don't know which language I'm supposed to use here, but this is it. They are here. Foramen magnum, the position of foramen magnum, forward position. Pelvic girdle, short and wider. And then they're saying that spine is S shape. So how many marks are there? They're saying describe. They're not saying explain. Foramen magnum in forward position because they're saying on the skeleton. Foramen magnum in forward position to allow the spine to connect vertically. Now you are explaining. Foramen magnum in forward position. But if you explained it, it's still correct. It's still fine. They will get your marks. Foramen magnum in forward position to allow the spine to connect vertically. Cool. Get your marks, né? Yes. They can't cross it because you added something which sense. You understand? Yes. So they're saying that foramen magnum forward position, okay, to allow the spine connect vertically. Then number 2 is the shape of the spine, which is S shape to allow flexibility. Yes, and then pelvic girdle short and wider to support the upper body weight. So exactly that's how you were supposed to write these 3 points bullets. And you see I put them in different colors, which shows that those are different marking points. Yes, these are different marking points. Oh yeah, man. You did not mention later. I did not mention 1.53. 1.5. 1.5. Oh, thank you. Thank you. Thank you. You see, that's how I 1.5. 1.53. You said I did not mention the 1.53. Oh, thank you so much. Monomer, thank you so much. Thank you so much. Nkanyisire. You see, that's the reason. That's why I told you that sometimes I'm rushing. And when I rush, I end up forgetting some things, you see. So, yes, Nkanyisire, thank you so much. They're saying that 1.53, it is letter. Monomer is D. So, it is D, which is a nucleotide. And then sugar of DNA, we said is the oxyribose sugar, which is C. And then the complementary base for nitrogenous base found in D. Found in D, which is adenine, and then in this case it is A. They just said it. So you guys have to say adenine because A is adenine. And then this, because this one was obtained from this side, you understand? It was obtained from this. You see, the arrow is showing it was obtained from this side, therefore adenine from this side. So that is it. Thank you so much. Thank you so much. Raswell, you will get it. Don't stress about it. Multiplication, we are tricky. Okay, thank you so much, guys. I think section A, state 3 characteristics of the scar that allow binocular. Here, the characteristics of the scar that allows binocular, the eye sockets, they are in front, eyes in front. You understand? It has two eye sockets. Basically, if you have one eye socket, because they're talking about the characteristic of this organism to allow what? Binocular. So, two eye socket. So, if you say eye socket, two eye sockets, they're in front, you see, so that they're able to see. Binocular means you're able to see with the two eyes. So, two eye sockets meaning that they will have two eyes. You understand? Yes. Think that is it. If you got another answer which is correct, then they'll also consider it based on the discussion of the memorandum. Who got 50 out of 50? Who got 40 out of 50? And if you got at least 40 out of 50, yeah, you a star. And then if you got 45 out of 50, you a moon. And then if you got 50 out of 50, you are a sun. Yes, all right. Question 2. Let's go to question number 2. Hey. The table 1 shows the partial DNA sequence of human, and table 2 shows the amino acid coded for by different messenger RNA codons. Best triplet number 1, 2, 3, and 4 human DNA molecule. Right. Name and describe the process in protein synthesis that is responsible. I told you this. Formation of messenger, that is what that is the transcription. But I told you that if you mention the process, they give you a mark for that. So it is here, transcription, formation of messenger. So when you are describing it, you describe it. You understand? You describe all of it. Transcription means formation of messenger. So they say the name. So you mentioning transcription is a mark. So mentioning. So in description, you have to get at least you have to get 4 marks. I released a simple video which was playing nice, and just to make sure that you remember this, you don't go to the paper without knowing it. I hope you guys, you listen to it and then you're able to get a distinction, or you get able to get good marks in this regard. So that is done. I'm not going to go through, but double helix DNA unwinds, weak hydrogen bonds break, double helix DNA unwind. You understand? Weak hydrogen bonds break. You understand? Then one strand is used as a template. So here I used a different version. So here it forms to form two separate strands. One is acting as a template. So you have to describe it. They just want only 4 points from there, and then you get it right. All right, describe, give the messenger RNA codon that is formed from the base triplet number 4. So base triplet number 4, I come here, I look for base triplet number 4. So they're saying because this is DNA, so a messenger is gonna be U, G, U. So here is gonna be G, U, U. So for that answer, they are saying name the amino acid coded for triplet number 2. When I come here, I need to find out what is here. It is messenger RNA. I explained this, and then so it means that I just have to convert it one time. So if this is gonna be UGC, UGC, UG, where is it? UGC. It's here, which is cysteine. Yes, so the answer there is going to be cysteine. So this is the answer for that. Then they are saying that mutation. When I was explaining this, I called it a devil, that because many students, they don't understand it. Yeah, sir. Someone say that sir bonge okay ntando say 1 point. If I said let's bro ridge, will they cross it? Let's bro ridge 1.6, 1.63. I don't know. Let me check. 1.63. Does it make it to be binocular? Because the question was about binocular. Does it make it to be binocular? I don't know really, but they might. But you are looking for something which make that organism to be what? To be binocular. You understand? Yes. Then we go to the next question. Explain. This one I explained it several times, and I say that you want it or not, that question is gonna come because I knew that it's gonna come. How did I know that it's gonna come? Don't ask me, because I'm a teacher who has taught for many years, and I know that. You know what happens when we see that you fail something? We keep bringing it until you understand it. If you don't understand it, we will bring it, we bring it, we bring it until you see that you understand it. That's why we bring things which you see that you have failed them. We bring them again. Is it fair? I don't know. Do they want you to pass? So you want all of you to pass? Who's gonna give you jobs? I'm just asking myself. Explain the effect. They want to know the effect this mutation has on the protein. The effect this mutation have on the protein. So what happened and how do you write to get these 4 marks? Saying that mutation caused triplet 1. So let's look at triplet 1. Triplet 1 is TG, TGT. Me, I use the TTT. Okay. Changed to TGT to triplet number 3, which is TGC. TGC, isn't it? So now I need to know what changed. This and this. Meaning that this was changed. So you write in exam that TGT, the third, you can say that the third base T was changed to C. That is the first mark. As I told you that TGT is changed to this. So you need to tell us what changed. The third base T was changed to C. That's 1 mark. I'm not saying that that's how they're gonna give you a mark, but that's how it's supposed to be. Yes, so it depends on that, but that's 1 mark, obviously. The second bullet will be you change this and you tell us which amino acid do they code for. The second bullet, this TGT is gonna be ACA, and then this one is gonna be, when you change it to messenger, it's gonna be AC G, isn't it? Yeah. Then now SCA code for what? So this is another marker, guys. And then the first one when you are writing it. This is one mark. This one mark. Hop, yes. All of them. Then you tell us what do these ones code for. How did this one code for? ACA, ACA, AC, SCA is here, which is threonine. And then SCG, SCG, SCG is the same thing, threonine. So meaning that now you tell us that these two code for the same amino acid, which is threonine. You understand? And then because the same amino acid, so it's another mark. Because the same amino acid, therefore there will be no effect on the protein. No, that word no effect on a protein is also another mark. So another one mark, change this one. The third base changed from this to this, one mark. Then you tell us that this code for this, so you, another mark. And then you tell us that this code for threonine and this one also code for threonine, another mark. And then there is no change in the protein, that's another mark, which are 4 marks. That's exactly what I said in a paper or in our preparation, and then I hope you got them right. Thank you, my boys. Thank you, my girls. Right. Let's continue to the next question. Let's continue to the next question. 2.4. 2.2 diagram below shows the cellular phase of meiosis, a phenomena that can occur during this phase of meiosis, which may lead to chromosomal mutation. All right. At the end of meiosis, gametes are produced. Cool. Explain one other biological importance of meiosis. So here you talk about variation. They're talking about biological. You talk about variation. You talk about maintaining the chromosome number from generation to generation, because production of haploid gametes, they talked about it. Tabulate the difference between meiosis 1 and meiosis 2. I told you go phase 1, go to phase meiosis. Prophase, crossing over takes place. There's no crossing over in prophase 2. You go to metaphase, homologous chromosome align along the equator in pairs. Then other side, chromosomes align along the equator singly. Then the next part, you talk about chromosome, homologous chromosome move to opposite poles. The other side, chromatids move to opposite poles. You form 4 cells. So here you talk about all the differences, the way I showed you in the book. So I don't need to go through them. About them like many times. You understand? Yes. So that is it. Then we go to the next question. Yes, these are the differences we said you must write about. There are the differences. Then we go to the next question. What are they saying? What are they saying? What are they saying? The next question, what are they saying? So the table must be a table. You draw a proper table, like, then you put here meiosis 1, meiosis 2. Bullet number 1, you write. Bullet number, you write. Separate the line. Bullet number 2, you write. You separate the line. Bullet number 3, you write. Yeah, it must be a table. It must look neat. Don't just draw a T. Then they're saying that name and describe the phenomena that occurred in this. The name, you see, they said name. So in this case, the phenomena is non-disjunction. So that is one mark. And then how do you describe it? They're not saying explain. They're saying failure of the chromosome to separate. Yes, or these chromosomes failed to separate. This chromosome failed to separate. And then the whole pair is pulled to opposite. It is pulled to one pole, you see. So it's being pulled to one pole, and then this was mitosis was anaphase 1, you see. It is anaphase 1. I don't know whether they mentioned it. If they didn't mention it, then it could be another mark. So at the end, yes, at the end of meiosis in the cell, if the gamete containing 3 chromosomes fertilize the gamete containing 2 chromosomes, would this lead to Down syndrome? I bet that many people, guys, please don't tell me that you said yes, because I emphasized it. I emphasized this, and I say that I'm emphasizing it so that you don't make the second mistake again. I'd even left, and then I said, oh guys, I remember this. So remember, at the end of meiosis, let's read again. If one gamete containing 3, if one gamete containing 3 chromosomes, I see, fertilizes a gamete containing 2 chromosomes, would this lead to Down syndrome? That's the answer. I will say no. You know why? This is not a human cell. Did I mention here the human cell? Never. And when I told you, what do you see here? What do you see here? How many chromosomes do you see? Do you see 46? Do you see 23 pairs? No. You see how many? I see 1, 2, 3, 4. So if one cell has this 1, 2, 3, you understand? So does it lead to Down syndrome? The answer is no. Why? Because this is not a human cell. It's not a human cell. Yes. And they didn't mention that this is chromosome number 21. It only applies to chromosome number 21. Do you understand? Even when we are talking about it here, when we are talking about it here, check. They say that 3 number chromosome 21. And here, even here, they don't say 3. They say 23 plus 22 gives you 45. Or 23 plus 24 gives you 47. They don't say 3, they don't say 2. Unless they say 3 for chromosome number 21. You see that? With the 3 number 21. You see? Number 21. You see? With the 3, it must be specific to chromosome 21 for someone to get what you call Down syndrome. Please don't repeat this in the final exam. You understand? And I emphasize this, that if you don't see human cell, please don't talk about that. Yes. So I think I'm done with that. Then they're saying that, what is happening with me? Safia. Thank you so much. I don't know what's happening with me. Maybe I'm so excited and stuff like this. Yes, he's saying that this I didn't. Thank you so much, Safia. Draw a diagram of a cell in a phase before. That's exactly what I say. That, hey, as if these people were reading my mind. Anyway, draw the phase of this cell before one that's shown above. One shown above is anaphase 1, because these are still full chromosomes, you see. So the cell before you draw for metaphase. But now they said labeled. Make sure that you label everything. And then number 2, make sure that you give a caption. Title. It must have a title, 1 mark. So these 5 marks is for Mahala. Honestly speaking, however much it had some challenges, but they brought a lot of open questions for you to answer and get marks. Yes. Let's go to question 2.3. This one, I didn't go through it. That's what I said. And I say that everyone knows about it. All right. Although a rare occurrence, cases of babies using switched birth in the hospitals have made new. Okay, you guys, you read this. Since the individual sequence, okay, unique. Mhm. So they used restriction fragment length polymorphism. Yes, this is the first one. It's the method. Or they're using polymerase chain reaction, which we call the PCR. They call it is a PCR machine. It's a very small thing, but it runs, spins very fast, very fast. The PCR machine, yes, and very expensive. Yes. Below are the results. Okay, the DNA. They are saying now question number one. From the text, state two methods they used to analyze. So these are free marks. So they say they used this and then they used this. They are saying beside on the DNA profile prov, mm, provided which baby or babies was uh, currently allocated by the hospital. So in this case, correctly allocated by hospital. So what you need to do is you need to go this, to go this, to go this. Like you have to match them using a ruler. Yeah, so you have to use a ruler and then you see that these things are matching. So after I did my matching on the screen here, so I saw that I saw that baby B, baby number two. Baby number two was correct with the couple number two. So it means that, so if you, this is a father, no, okay. Baby number two, they said baby number two. Baby number one was given couple A. Baby number two to couple C, meaning that, yeah, this one, okay, let's find out. This matches with that from mother. All of them must be matching. Yes. And this one matches with the mother. This one matches with the father. If the mother, swapping them. This one matches with the mother. This one matches with the father. You see that all of them are finished. There must be no bar which is left, which is not matching with either father or mother. So 2 becomes the most correct answer. Yeah, I hope you matched correctly. Yeah, if you find another one, try to match. Yeah, but based on what I've matched here with my eyes, but you're supposed to use a ruler. You understand? So that you bring it correctly. You understand? But since 1 mark, maybe it's that is. Explain your answer. The answer is explaining that you see all these bars. Yes, they match with the father and mother. You understand? All of them. Or the bars which match with the mother, the remaining bars must match with the father. You see? Yes. Of the remaining bars of the kid must match with the father. Or those which match with the mother, the remaining bars of the kid must match with the father. So that is it. Provide one other use of DNA profiling. I told you, go read about it. You have a lot about this. Talking about donor, you're talking about what happened to my system now. Yeah, talking about donor, talking about the forensist. Yes, how do they use it? We can use it to identify criminals. You understand? So this one is 1 point. If you say criminal identification by, you understand? Yes, diagnosis of inherited disorder. You understand? Yes, so you give those answers. Then we go to the next question. Someone saying 2.23. 2.2. What's happening to me? Wait, 2.2. 2.23. Oh, yeah. I answered it, guys. I hope I answered it. Yes, I answered it. The one which was talking about the diagram. Yes, I answered it. Hey, guys, I hope I'm not. Yes, I don't know. I don't know what to say. All right, let's continue. So I think I'm done. I've not skipped any question. Eh? If I've skipped, guys, let me know. Eh? 2.4. 4? Yes, yeah. They're saying that in this species of fly, Drosophila gaster, yes, which we call the fruit flies, yes, turn light brown, body color is basically dominant and dominant over black. So this one color is dominant over this, and then they're saying that long, long, this long, meaning that this one is capital T and long is dominant over this is small l, and then this is small t and l. Okay, sharp. The pinot diagram below shows the cross between the female and the male. Okay, Drosophila melanogaster fly as shown. State why the type of cross involved in this scenario is referred to as a dihybrid cross because two characteristics were involved. So you can talk about them, the color of the body, and then you can talk about the length of the wing. Yes. Then they're saying that state, okay, the formation of gamete with different allele combination can be explained by Mendel's laws. Name two laws that apply. So the formation of gamete with different allele combination can be explained by Mendel's laws. Name two laws. Mendel's law of independent assortment or Mendel's law of segregation. And then number two, you also have Mendel's law of dominance because one is dominant over one is dominant over the other. So Mendel's law of dominancy and this Mendel's law of independent assortment or Mendel's law of segregation. Yes. They're saying explain your answer. Oh, so basically, where there is a capital letter or a recessive allele, the dominant allele will only express itself. You understand? And then when during assortment, during segregation, these alleles will separate independently. The capital T will not influence small t, and the small t will not influence the capital T. They will segregate independently. You see, they will segregate independently. You see, so that's how Mendel also that there is no influence of one allele by another. You understand? Allele in another locus. You see? Yes, because these alleles are going to be going to different loci. So they're saying that identify the genotype of gamete of male A, the genotype of gamete. So this one, they're looking for that. How did I form this? Look at what is formed. So it means that if the capital T came from here, small t came from there. So it's going to be small t. And if capital L came from there, small l came from here. So it's going to be small t, small l. So that is it. It must not have space. Then the genotype of offspring B. Genotype of this. So now since I know this, capital small t, small l. So I'm going to say capital T, small t, small t. So it's going to be small t, small t. And then it's going to be small l, capital L. So that is Mendel's law of dominancy. So meaning that if the dominant allele is there, a recessive allele can't express. And then you start with the capital letter. So that is the genotype for individual B. They want the phenotype, not the genotype. Ne? Since they want the phenotype, then it means that it's gonna be small letter is what? Small letter is gonna be black and long because capital letter is there, the dominance is there. Long wings, black and long wings. Then the genotype of a female parent, genotype of the female parent, genotype of the female parent. Where is the female parent? So we see that yes. So we see that this one, they say that, what did they say here? Did they say, uh, Based on what we see here, we see that these were heterozygous. This is heterozygous. You understand? This is heterozygous. No. Where it came from, né? Because these two they combined, so it was like this: capital letter L, capital letter small t, capital letter L, and then capital letter L. Remember, this was they say that white and long. So that is capital letter T, small letter t, capital letter T, small letter t. So that is it. And then capital letter L, small capital letter L, and then it gives that. But because it is a genotype, we don't circle them and we don't leave space. So it's capital T, small t, capital L, like that. Yes. That's it. I hear. Yes, so I think clear there. Yes. Someone was asking a question that, yes, sir. Hi, sir. I'm seeing your memo and I'm super stressed. My hair might follow. Don't please, don't please, don't please, don't do that, don't please, ne? This is just a guide so that your hair doesn't fall off, because now you still have a chance to modify your life, you understand? Yes, you still have a big chance, and I know that when you experience that and they bring the same thing, you will never fail that again. I'm telling you, you will never fail that again. So we have explained the Mendel's law of dominance and the Mendel's law of independent assortment, or Mendel's law of segregation. Yes. So I think I'm done with those ones. Let's go to question 2.5. The diagram below shows two models of evolution based on the rate at which it occurs. Yes. I talked about this punctuated equilibrium and gradualism, guys. Ish. And I emphasize and say that please don't make a mistake. And I know that there are some people who made mistakes and they went without having it. You understand? And I emphasize that please, okay, just help me go to the paper. Please don't go to the paper without knowing this, without explaining that. Therefore, there we go. And I told you that if you see something with the ladders, with the ladders, that is punctuated equilibrium. That's what I said. And you see that something has no intermediate. There are no intermediates here. That's punctuated equilibrium. You see something is continuing gradually. That is gradualism. And even he used an example of people who go to ShopRite and buy Gentle Magic. They start to change their skin slowly. It doesn't today here, tomorrow there. No, it takes weeks so that the skin keeps on changing. You become bright and bright and bright. So I think it's clear. Next time you won't fail it. If you fail it, if you got it by chance, then it's cool, still fine, cool. And then tomorrow make sure that you're able to explain it. Give the letter of A or B. That would be best present gradualism, obviously. So gradualism is gonna be B. And then this guy explained evolution. Name the letter A or B. Yeah, this is punctuated equilibrium. Yes, punctuated equilibrium, you see. That's the guy. Punctuated equilibrium. They brought him here. Exactly. Yes, so it is a punctuated equilibrium, so the answer becomes A. Name and describe the model of evolution in A. Name and describe the model of evolution in A. Where is the model of evolution? They are looking at this. They're saying to describe. So when you say describe, name, meaning that punctuated equilibrium, you have you have explained. That's why they used this his name. You understand his name, so that when they ask you here, they ask you what is it. You understand? So it is the punctuated equilibrium. So your answer was supposed to be punctuated equilibrium, the way how it is here. And then now you come back and say, describe the mode of evolution. So here it has long periods of no change with the rapid periods of what change. Did I say the rapid? Yeah, long periods of no change with the rapid period of with the short period of rapid change. Long period of no change with short periods of rapid change. Long periods of no change with the short periods of rapid change. Longer periods of no change with the rapid periods of with short periods of rapid change. So that's what they are trying to ask you to explain. It is made up of long periods of no change with the short periods of rapid checking add, because you have already identified, so 1 mark is already gone. Yeah, name and describe name the type of fossil that support the model of evolution in B. So in B, that is the gradualism. Basically here we use fossil records, the what, the skulls and stuff. So we have different, basically we can talk about the body fossils, because the types of fossils, I know they are body fossils, molecular fossils, we have trace fossils, we have carbon fossils, we have pseudo, we call them pseudo fossils. They are not fossils, but they look like fossils. But in this case, we use those remains which are we can able to see, like the skulls. When you talk about how do they evolve, how do they form these transitional fossils, né? Yes, that's how you explain it. So the type of fossils we see, you see that it has these transitional fossils, né? Yes. So basically it explains everything about the body fossils involving transitional fossils. Why? Because here we don't have those transitional. Here in punctuated equilibrium, we don't have those transitional fossils. So yeah, I hope they will give all them, or they would, it will depend on what they have discussed, or based on your level. Question 3. I hope you guys, you got 50 out of 50, or at least 40. Last time you got 40 out 45, and then now 40. So that is the 80, 85. 85 out of 150. So now let's go hunt for some other 40 marks or 35 marks, and then we see if we can get it. Maize. Domesticated crop have been transformed over thousands of years and now look vastly different in comparison to their relatives in the wild. So they're saying that the maize belong to genus Zeme, which includes wild Theos Int. This is scientific. They know them as Zime. Yes, Zimes. Okay. Okay. So they're saying that this produce cob only 6 to 12 in rows, protected by the hard outer covering. Modern maize produced by farmers have cobs consist of 500 or more exposed to. Yeah, I didn't know. I'm just eating meal. Yum, 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 yum. Even I did. Oh, who knew how many lines do you have on the maize? We are just eating, enjoying, putting salt, and yeah, we throw the cob. My God. Diagram below shows the wild. Yes, name the process through which human produced. This is how do you call this process? We call it what? Artificial selection or selective breeding. This is it. This is the maize they are talking about. Here we go. These people, they will kill me. Ne? Yeah. So this is the maize they brought. This maize. and they brought this maize. That's it. That's it. So it is artificial selection. Sometimes you call it selective breeding. All right. State one negative effect that process mentioned above may have on the species. So first of all, the one which are not desirable might get extinct because they're not given a chance to reproduce. That's number one. Number two, it might change the taste of these individuals. You understand? Yes. Number three, you can talk how many they want. They want only one. Describe how this. Yes. Number three. Okay, you can also mention any of what you think. So, but we humans start to influence. The nature cannot determine what is the product. You understand? So, meaning that there is no natural selection there. And if there is no natural selection, when you're just going with what you want, some genes of this species might disappear completely. You understand? Yes, because it's not about the best suit. It's about the desire for the human to produce that. Describe the process mentioned above. It was carried out. So basically, you just explain this. Just explain this. So what did we want? What do we want in this regard? Yeah, yes, you just explain this. Yeah, that choose or select the maize in the field. You understand? That produce more maize, more this. You understand? So what happens? You see, you modify them, and then now let only those maize reproduce because they have more of the rows of this meal. Then select the offspring that produce more on this cob. You understand? And let only those offspring reproduce, meaning that those maize which have more maize on the cob are the one which are, you understand, allowed to reproduce. How do you do cross pollination, or you allow those ones to pollinate each other? You understand? And then you keep repeating this until this big maize is obtained. This is not just one day, it's about many generations. Yes. So describe how this product. Okay, we are done with that. Explain one economic benefit of producing modern maize cob has to the farmer. Yes, the modern maize cob, it brings about more food availability, and then they can sell, they get money. You understand? Yes. So basically that's it. Number two, you can also talk about increase the resistance, but is that economic? Yes. If it can resist the drought, then automatically it means that the person will get more money. If it can resist the diseases, then automatically it means that the farmer will not use more pesticide, insecticide. You see? Yes. So more cob, like more on the cob, meaning that the farmer will be able to sell and then get a lot of what? A lot of profits. So any of one, then which you can explain, then you were supposed to get a mark in that regard. Let's finish up. Someone saying, why me? Okay, they're saying that I've been getting distinction because of you. Thank you. I think I might go to the national course. My average is 95%. Yeah. Let's clap for you. Yeah. Yes, man. So I got 150 in this paper because of you. Oh my God. Thank you so much, guys. Thank you so much, guys. Hey, you make me proud and make me not sleep so that I prepare you more so that you maintain this. I feel. Oh, last time, I feel so good. I feel so good to hear that. And guys, those who are still low, you can still make it. I'm telling you, you can still make it and you can still improve your marks in this remaining time. Even if it's there, still you can make a change. I'm telling you. Yes, let's continue. Okay. Let's go to the next one. They are saying that okay, connected to two large mess. Okay, this two large mess. You guys, did you read this? It says, and is separated by two water bodies. Saying that this island of Panama is narrow strip, okay, strip of land that joins also. So is it? I think it is this. Oh, it's this, ne? Okay. Let's say North South America. Some believe that this strip of land formed 3.5 million years ago. The Caribbean Sea is warm. Where's the Caribbean? Oh, this is warm. Is warm, while the Pacific Sea is cooler. This one, okay. Then they're saying that pork fish. Muslims, do you eat this one? Pork fish, maybe it looks like a pork. I don't know. Or tastes like a pork. I don't know. Okay, can be found either side of the narrow strip. Okay. However, when scientists placed the male pork fish from the Caribbean Sea, okay, ocean into the same fish tank, they would not cooperate. They are now considered to be all right. So I have pork fish from Caribbean, and then pork fish Panamic. Panamic, we have the Panamic and the Panamic pork fish, and then we have the pork fish. Okay. Define species. Species, that one you know. Ne? Group of organisms that can interbreed to produce fertile offspring. I said yesterday that fertile offspring not viable. Name two isolation mechanism. These are the one. I said that I'm not going to go through them, but you have to know them. Yes, this one. See? Say don't go without knowing them. Name two reproductive isolation mechanisms that have been prevented from pork fish from copulating. What do you think? What made them to not to copulate? First of all, it could be the distance. You understand? Yes, it could be the distance. Basically, is temporal what? Or the distance between the two. Then number two, because this one is warmer and this one is cooler. You understand? So maybe they are breeding at different seasons of the what? Seasons of the year. So it also could be a problem. Yes. So there is a barrier. Barrier prevents them also from having this gene flow. So based on the scenario above, describe speciation of pork fish through geographical isolation. Now, how are I supposed to explain this? Today they put it 7 marks. They usually put 6 marks. As I said it, make a mistake to go to the paper without knowing this. And if you did that and you know that what is the results. Okay, let's finalize it. Let's finalize it. Let's finalize it. Let's finalize it. They're saying that there was. Let's find out who was the ancestor. Okay, large two masses separated to Panama. Okay, Pop. So basically, there was one group of this pork fish, né? Isn't it? Yes. And then it was separated what? By a geographical barrier, and then you have to tell us which geographical barrier was that. This canal. This canal, you understand, separated into two different populations. You understand? There was no gene flow between this and this because they are separated by different geographical barrier, so they experience different environmental conditions. You understand? Different environmental conditions. So natural selection occurs independently. So it was one group of pork fish Then what happened? It was separated by a geographical barrier, which is this. You have to mention it. Yes, form two different groups of porkfish, and then now they experienced, there was no gene flow between these two porkfish. You understand, because of this barrier. And then natural selection occurred independently because of the different now environment. One is cooler, one is warmer, you see. And then what happened after that? Since natural selection occurs independently, then due to the different environmental conditions, then they became genotypically. You start with the word genotypically, and then phenotypically different. You understand? And then when you bring together these porkfish, they say that they cannot, if you remove the barrier and you make them mix again, they cannot interbreed to produce fertile offspring. They said it here. They cannot. They cannot interbreed to produce fertile offspring. They said it here. They are now considered to be two different species of this pork fish. Then you can maybe mention it, i.e., this name, and this name. You understand? Yes. Yeah. Sometimes those two names could be a compulsory mark that if you don't have it, you have not written it, out of 7, you're gonna get 6, because they wanted to see these new species which were formed. You understand? Yes. Lastly, but not the least, is the pedigree. Okay, you're saying skin disorder caused by chromosome. You see, I told you that it's sex chromosome. Ne? Is it sex linked or non-sex linked? So we know that it's sex linked. So now you need to also know whether it is dominant allele or recessive allele. They showed you here it is a recessive allele. Now that's cool. Why do I know that's recessive allele? Because they said individual B. Individual B here, individual B, they said affected male, meaning that is caused by a recessive allele. Name the type of diagram indicated by pedigree diagram. Provide a possible genotype of individual A. Provide a possible genotype of individual A. Individual A, if you look at individual A, produced a baby who is normal. So it means that the individual A, since this one is normal, so, and this is sick, then automatically individual A could be XX, capital T, yes, even X is X small t Y. So if this combine with this is gonna form X capital T X small t. So this individual is still female, but is still normal. You see, it's cool. When it's like this, there is a chance that this combine with still this to form still this. So I think this, they will give you a mark, and they give a mark. How many marks we are given? 2 marks. Yeah, cool. That's why they say genotype or genotypes, so that you write these two together. Then they're saying that explain the relationship between inherited genotype and phenotype of individual C and the Mendel's law of dominance. They wanted the Mendel's law of dominance. Yeah. Mendel's law. Today they brought all the Mendel's laws. Explain the relationship between the inherited genotype, yes, genotype, and the Mendel's law of individual C. Let's find out who is individual C. Individual C, hey, yeah man. Individual C is here. So if it is individual C, explain the relationship between inherited genotype and the phenotype of individual C and the Mendel's law of dominance. Individual C, where are you? Okay, individual C, we have seen that. Oh, individual C, first of all, it is a female who is unaffected. So that is the phenotype. But the genotype, the genotype one, this individual C is like this, capital letter T and then small letter t. Why? Because this father gave that individual a small letter. So now how are you going to explain? So basically this is a genotype. That's mark 1. Then you tell us the phenotype, it is normal, it's unaffected. You understand? That is another mark. Then the other 2 marks, you tell us that because you need to talk about the inheritance. Yes, obtain this dominant allele from the mother and obtain the recessive allele from the father. Yes. So basically that's what they wanted in that regard. So explain why males are more likely to be affected by disorder than the females. So we explain this also, that males have one X chromosome. If a recessive allele of T appears on it, the individual is having the disorder, is affected with this disorder. Yes, while female has 2X. If a recessive allele appears on one. Don't know why they give it 2 marks, because it's supposed to be around 3 or 4, whatever. Use the genetic cross, show how individual D inherited. Individual D, again, they're asking the same thing. Inherited the genotype which caused this individual to be affected by the disorder. To be affected by genotype in the correct position on the right. Circle. Okay, they're saying circle the genotype in the correct position on this cross. Use the genetic cross, show the individual how individual D, okay, that one is cool, inherited the genotype. Okay, individual D obviously is a male, whereby is having X, small t, and a Y. So since this one is heterozygous, then we can. But they say using the genetic cross. You know what is the first point? As I told you, that the first point in this case, you're gonna write this individual D. Ne? So this individual D was on the second generation. So you're gonna say P2 F2, because P1 is this. Ne? Then this is P2. You understand? Second generation. So P2 F2. Then you say phenotype. Yes. And then here, okay, let me just do it on the blank page. Hey, someone saying that, yo, hi guys, wanna hire from Cape Town. How many marks does evolution weigh? Oh, all right. Yeah, they're saying, let me just use this blank page so that you're able to understand what I'm saying. So we are answering this question. Ne? They're saying use the genetic cross to show how the genotype D. So genotype D is this one. So how did this individual inherited? Caused by individual to affect by the disorder. Okay, so basically what happened is I'm gonna have P2, okay, F2. Yes, phenotype, phenotype. Then I'm gonna have genotype, genotype. Okay. Then here it's, I think this one, remember now these are the parents. So it's gonna be normal. So it's gonna be normal female crossed with normal female crossed with normal male. Okay, sharp. Then the genotype, when we see it here, we say that this is a heterozygous and then this is normal. So it's gonna be X small t. It's gonna be X capital T and X small t crossed with normal male, which is X capital T Y. Then you say meiosis. Then when meiosis occur, so you're gonna have capital T. Let me increase the thickness. Capital T. Then here is gonna be then it's gonna be capital X small t. Then this one is going to be X, capital T, Y. You see? Then these are gametes. So let me just make this one far. F2. So now I have to cross this, whereby this is gonna go with that. So I'm gonna have X, capital T, X, capital T. I'm gonna cross now this with that, whereby it's gonna be X, capital T, Y. And I'm gonna cross that with that, which is gonna be X small t, X. Okay, let me start with X capital T, X capital T, X small t. Then now this with that, which is X capital T, Y. So now this one no, this one no, this one no. Oh, did I cross it properly? Okay, that's why I prefer using a Punnett square. Okay, this with this. Capital T, capital T. This one is correct. This with this. You see? Capital T, Y. Then now, this with this. Okay? Yes. It's capital T, small t. Now, this with this is small t. This is a small t. So let me just say this one. So they said circle. This is the one which caused this situation in this boy. Yes, this is the one. They're saying that circle the genotype in the correct position of the cross. You know what they are saying there? Yes, this is the one. So it's basically like this. That's the one which caused that individual to be like that. How many marks are there? Then you put you can put phenotype, genotypes, everything, so that if any mark is there, they can still give you. I told you that when it comes to this genetic, you will write everything Kaufera. No one is gonna cross you because you overwrote. Yes. The last question is Nala and Luke are farmers of the same species of fish, small fish. Nala breeds homozygous yellow fish, while Luke breeds homozygous blue, and then this is yellow. Okay. Ooh, the question is going. In the investigation, determine the dominant color in this species. They allowed the investigation determined the dominant color in the species of fish. They allowed for yellow fish to breed only with the blue fish over 5 years. Okay, over 5. Oh, this is a key point. And collect 100 species each year. This is another key point. They say they allowed yellow and blue only. So it's another key point. Okay. Name the type of dominance which is indicated there. Okay, let me check and see. It's this. Yellow and blue. Why is this? So this one becomes an intermediate. Therefore, it is incomplete dominance, whereby you have an intermediate. It's not yellow, it's not blue. It's just a medium color, which is an intermediate. Therefore, it is incomplete dominancy. Incomplete dominancy. Provide a reason for your answer because you produced a new character or an intermediate color of the offspring. Okay. Name 2 ways in which the reliability of this investigation was ensured. Hey guys, here we mark the English. As I told you that here we mark the English. You need to know which English do you use. Name 2 ways in which reliability is done by improving by repeating the experiment many times. So how did they do the reliability here? They repeated the experiment over 5 years. So that is the key point. They repeat the experiment over uh, five 5 years. You see, that's number one. Number two, they used a large sample size. They collected 100 offsprings each year. You see, 100 offsprings each year. You understand? 100 offsprings each year. So this is the first point. This is the first point. This is the first point, and then this is the second point for the reliability. Give one way in which the results of this were made more valid. How did they make this species? How did they make this experiment to be more valid? Here we use what did they use? They use the same. They only allowed the species. You see, they only allowed these species. You see, to the one yellow and the blue to mate. You see, yes. So they, meaning that they kept the same type of species. Yes, they only used the same. The blue were supposed to be heterozygous, homozygous, and also this one was supposed to be. So that is, they use this. What did they use? Same way. They didn't change it over the experiment. That's what they are. So if you wrote any which was used, so they will credit you hopefully. So give to planning stage. So planning stage, you see that they had to identify the homozygous blue species and also homozygous, you see. Then number two, where did they fish them? They had to prepare where these fish are going to be bred from, like maybe the pond and stuff, you see. So how did they obtain them? Maybe also the workers who were supposed to be there, they had to train them. So how do they do the planning? Maybe is it allowed to just do the fishing without permission? So what did they do before they did the experiment? What did they put in place before the experiment was done? So bar graph represents this one. Obviously, you know, this is supposed to be on the x-axis, this is supposed to be on y-axis. And then remember, when you're marking the bar graph, it's supposed to be spaces in between. Yes, spaces in between. This space must be the same as this. This bar size must be the same as this. If you don't do that, we give you 0. Caption must be there without units. Here it must be there with units. Here it must be there with units. And then this is what the figures go here. But if you start with 0, 2, 4, 6, the interval must be the same. Then now here you write what was it? The colors, the green, the blue, the yellow. Yes. So after that we come here, title, the type of graph, the caption, the axis. We give you those marks accordingly. But I don't expect anyone to fail or to fail to get those marks. According to Darwin's theory of evolution, there is a great deal of variation. Yes, according to Darwin's theory of evolution, there is a great deal of variation in offspring. Use the natural selection. Describe how variation of offspring increase the number of individual with the favorable characteristic in the next generation. So what are they looking at here? According to Darwin's theory, okay, there is a great deal of variation, you see. Using the natural selection, describe how variation of offsprings increase the number with the favorable characteristics. So basically, it's a generic question. There is no example they use. So they will tell us, you describe natural selection. Yes, it could be. This is what they are looking for you to explain. This is what I wanted. This yes, organisms produce large number of offsprings. There is a great deal of variation among these offsprings. Then they'll tell us that some have favorable characteristics, yes, for survival, while others do not. They don't have these favorable characteristics. So when there is environmental changes, those with favorable characteristics will survive, and those with unfavorable characteristics will die. You understand? And then the organisms that survive reproduce and pass on allele to their favorable allele to the next generation or to the offsprings. So that's what they are trying to. They wanted exactly without change in that. Yes. So I think, guys, yeah, if you wrote Mendel's law of assortment for God independent, I don't think it's wrong. So hopefully it's correct. Yeah, guys. Yeah, I think that this read the question. It says show how C inherited the disorder. It is supposed to start with the P1. Okay, Bong Tando, you're saying read how C inherited disorder. Okay, if you have a question, let me know. You are saying that Tando, you are saying that they said here, describe how C. Where is C? Oh, serious, Tando? Let me check. Tando, thank you. Let me check. We're saying that. Oh, explain the relationship between the genotype of C. They're not saying that. They're saying use the genetic cross. Show how individual D inherited. Tando. They're not saying, sir, read the question. It says that show how individual C inherited disorder. It is not supposed to be P1. Stando, they say that explain using genetic cross, show how individual D. They're not saying about individual C. Individual C was mentioned up here. Ne? Individual D inherited the genotype which caused this individual to be affected by the disorder. You see, they want to know how this individual D, this is the first generation. Individual D is the parents are in the second generation. Do you understand? Yes, the parents are in the second generation. But sometimes this P1, P2, sometimes they say those who wrote P1, they will give you, those who wrote P2, they will give you, depending on what they have discussed. But if we are using what I told you, that the KO, the method where when the examiner sees the answer, gets it right, P2, this is P2. Why this individual is on P2? Yes, because this is P1, F1, first filial generation. The first filial generation, if you use this individual, therefore they become parent number 2. The parent number 1, offspring number 1. So the offspring number 1, they become parent number 2 for offspring number 2. So now this one becomes parent 2, then this one becomes offspring number 2. That's why we say P2, F2, because they are looking for D, not C. If it was C, then it would have been correct. But they are saying individual D. I think I'm clear there. Yeah, if there is any other question, sir, okay. Thank you so much, Karabo. I think, guys, everyone is satisfied. I don't want to go away when.
Welcome back to our session, which is guiding you for paper 2. We'll be looking at questions, most likely examinable questions, which are going to come in your paper 2, both in prelims and final exams. Don't forget that in most cases, we don't set new questions, but we use the questions which are prevailing, and then we modify them. So I'll be showing you how to answer these questions, and I'll show you different questions. Some of them I'll be just speaking about them, and then how do you answer those questions. So keep on so that you don't miss out anything, and don't forget to subscribe, like, and share to others so that you can also benefit. Saidi, as usual. Let's start with the question 3. The diagram represents the portion of nucleic acids. Before I start this, don't forget that we have specific topics which we are supposed to look at in paper 2. The first topic is DNA, code of life. The second topic is meiosis. The third topic is genetics. The fourth topic is evolution, but both evolution in natural selection and then human evolution. So it means that these are the topics which are being set in paper 2. So when you are reading, you must focus on these 4 topics or 5 topics. So we're going to look at all questions, and then the most likely examinable questions or questions you most likely to find in your paper. If they bring a diagram like this, you have to find out which diagram is representing. So this diagram represents DNA. Why DNA? Because of this, the weak hydrogen bonds. And then it's two-stranded. What are some of the questions which are most likely to be asked about the structure of DNA? Apart from labeling the structure of DNA, they can ask you to describe the structure. Apart from labeling it, describe the structure of DNA, and they put there 6 marks, whereby you have to tell us DNA is made up of nucleotides. Each nucleotide is made up of a phosphate, a nitrogenous base, a phosphate, and a nitrogenous base, a sugar, and a phosphate. It forms 2 strands. Each strand forms 2 strands. Each strand is joined together. So you describe the characteristics of DNA. Basically, what DNA has, it forms 2 strands which twist around to form a double helix. So you can get the 6 marks from there. So in this case, let's label it and then find out what is a V. What is V? V is the nucleotide, because a nucleotide is a nitrogenous base, a sugar, and a phosphate. So this is a sugar, and then nitrogenous base, and then a phosphate. So this circle or box is showing you a nucleotide. Then Z is weak hydrogen bond. Use the word weak. Weak. We want to see the word weak. Then hydrogen bonds. Weak hydrogen bond. And then this is a nitrogenous base. Nitrogenous base. And then also this is a phosphate. Sorry, a nitrogenous a sugar molecule, which is, in this case, it is a ribose, deoxyribose sugar. And then Y is a phosphate group, as we saw it here. So now, since you know that this is a structure and you know how to label it, now let's talk about name the nucleic acid which is being represented. So we have seen it that this is DNA. Then they're saying name 2 places in animal cell where you can find this DNA. Where can you find this DNA in animal? You can find in animal in 2 places. That is in the nucleus, and then in mitochondrion. If it's 1 nuclei, if it's 1 it's mitochondrial if there are many mitochondria. If there are many nuclei, if it's one, it's called nucleus. So it should not confuse you. They are saying name portion V. They are looking for portion V. What is portion V? We say that these are the building blocks. This is the building block of DNA. So it is the nucleus. Molecule Y. Where is Y? Molecule Y is a phosphate. We have seen it that it is a phosphate group. Then they are saying that bond Z. What is bond Z? Bond Z is weak hydrogen bonds. So I think we have seen the answer here. Weak hydrogen bond, you have seen here, phosphate group, nitrogenous base, we have seen it, and then portion W, which is the nucleic acid, which is the nucleotide. What is the natural shape? When they say natural shape, many students, they say helical, double helical. No, the only answer we accept in exam is double helix. Nothing else. You write something different from that, we won't give you a tick. Name the process during which this molecule makes exact copy of itself. How does DNA make exact copy of itself? How does it replicate? So the answer is DNA replication. So that is the process by which DNA makes exact copy of itself. Why does it occur? Some questions can come here. Why does DNA replication occurs? It occurs to increase or to double the chromosome number, Where does it occur? When you talk about where does it occur, then you talk about the nucleus. And then when does it occur? It occurs during interphase. Yes. And then why does it occur? Why does it occur? We say that to double the chromosome number. To double the chromosome number. And then lastly, how does it occur? That is the process of DNA replication. If they bring a question like this, how do you answer? This is a DNA molecule. So if you look at it here, you see it is a DNA molecule. And why is it DNA molecule? Because of the thymine, which is indicated in the nitrogenous base. And then also the weak hydrogen bonds, 2 strands, so it identifies it to be a DNA molecule. And it cannot be RNA, because even here they say that this is what? This is a DNA replication. Yeah, so question them. Molecule W, where is molecule W? We have seen it, molecule W is here. It is a nucleotide, correct. Molecule U, where is molecule U? The whole molecule is DNA, correct. Then they are saying that Z is weak hydrogen bonds, correct. And then what is V? How do we know the nitrogenous base V? Because nitrogenous base is compatible to this or complementary to this, therefore, and this is given it here, which is thymine, therefore V is equal to A, which is adenine. And then they are saying that name the part of molecule W, W, X, and Y. X is the phosphate group, correct, and then Y is the oxyribose sugar. Why don't I say sugar? You have to be specific because this is DNA, so it is the oxyribose sugar. Where in the cell does this process take place? Where do we find? We have answered this question that this process occurs in the nucleus. And then they're saying that name the phase of cell cycle where replication takes place. We have already answered it, and then we say that interphase. So at least when they bring such questions, you must be able to answer accordingly. So first try to answer what is being labeled. The moment you know what the picture depicts or what a picture represents, at least you are able to bring your mind and then you concentrate in that area. I'm not going to spend too much time here, only maybe if I see a very difficult question there, then that's when I can try to answer related to this, because I wanted to go to transcription and the translation. But molecule C is phosphate, and then this is a nitrogenous base, and then this is phosphate, sugar, deoxyribosyl sugar, and then nitrogenous base. It forms what you call a nucleotide. And then weak hydrogen bonds, and then this is a sugar, this is a complementary base. So this is a complementary base. And then here, yes, they say this weak hydrogen bond. And then we have this. How do you call that? That is not a strand. We call it a backbone. And then the whole strand must include with the nitrogenous base and this. So the difference between a whole strand and a backbone is the backbone does not involve the nitrogenous bases. If you are asked what is this bond, this is a phosphate sugar bond, but it is a covalent bond. If you refer it to physical science. Okay, two scientists who discovered, James Watson and Francis Crick, name one organelle where DNA is found in the nucleus and the mitochondria. Describe how mutation of DNA may change the structure of the protein. The moment DNA molecule changes, is going to change the sequence of the bases on messenger, and the messenger RNA is going to change the sequence of the amino acids in which they are being brought to form a protein. So if a different protein is formed, if the amino acid leads to a different sequence, then a different protein is going to be changed and or is going to be formed. Then amino acid leads to the same sequence as previous one. Then the protein is not going to, a new protein is not going to be formed. Tablet 2 different structure difference. Now, if the major point here is structure, the word is structure between the monomer, and they are saying about the monomer of DNA and RNA. So here they are talking about the monomer, and if you look at the monomer, they are looking at the nucleotide of DNA and the nucleotide of RNA. So you only look at the structure, which is the nucleotide, of which the nucleotide is made up of a phosphate, a sugar, and then the nitrogenous base. So there are only two differences here you can look at. You can look at the sugar. DNA nucleotide has deoxyribose, deoxyribose sugar, while RNA has ribose sugar. And then DNA has thymine, while RNA has uracil. So after that, you draw a table. It must be a complete table. Yes, it must be a complete table, and then you put DNA, and then you put RNA at least. Then you get a tick here, a tick here, a tick here, a tick here, and a tick for the table, which makes it to be 5 marks. And if you find a question like this and tablet, and there is only 10 minutes or 10 seconds remaining, don't waste time. At least you can grab that 1 mark. Just draw a table like this, and then put DNA, and then put RNA. Yeah, even if you don't have anything here, then we shall come and give you a tick. At least you score that 1 tick. So yeah, please try to use different ways in which you can get marks. Yes, yeah. So this is the same thing we have been trying to discuss. Phosphate, you have seen them, and then M2 process, which require the strand of the nucleotides in a single strand of DNA. Sometimes people think that DNA can be a single strand. It can be a single strand. What makes this one to be DNA is because of the thymine, which is being indicated there. So thymine is in this form, while adenine is in that form. Then they're saying that how many nucleotides are represented in the diagram? Yes, let's try to answer this. How many nucleotides? Remember, nucleotide is a phosphate, nitrogenous base, and a sugar. So this is one nucleotide, it's the second nucleotide, it's the third nucleotide, and then this is the fourth nucleotide. So the answer is going to be 4 nucleotides. And then they're saying phosphate molecule, then phosphate molecules, we just count them 1, 2, 3, 4. So the answer is also 4. And then they're saying that how many nucleotides, okay, I done with that. Sugar molecule, there are 4, but sugar, this is the phosphate, and then this is the sugar. Write down the nitrogenous base from top to bottom as indicated by the arrow as complementary base strand and this molecule. Here is just T, they're saying on DNA. Complementary. So they are not talking about convert it into RNA. Don't forget that A goes with the T, then C goes with G. So whenever there is A, T, we put A. Whenever there is C, we put G. Whenever there is A, you put T. Whenever there is C, you put G. So basically, that's how it's supposed to be answered. Name 2 processes that require the DNA strands of DNA molecule to separate into single strand as shown in the diagram. Which processes? 2 processes. We only find this in when the DNA replication replication is going to take place, and also we find this when there is a transcription. Remember here you form 2 strands, 2 separate strands. Each strand is acting as a template, while here you form 2 separate strands. Only one strand will act as a template. And while here 2 strands will act as a template. So these are the 2 processes which make DNA molecule to open. DNA profiling is also not a big deal. Technique, you have to know how to identify this. Read the question very well and then find out. Don't just answer the question. Detectives were investigating crime scene, found the blood on the broken window. Then the suspect that the blood was that of the criminal. To identify the criminal, they analyzed the DNA sample from different DNA sample from the blood and compare it to the 4 suspects. Now look, this is DNA sample, and then these are the suspects. It means that these are the DNA they found on the, but you have to see where it is corresponding. This one goes with that. This one goes with that. Still, yeah. This one is that. Use a ruler to identify this. That this with this. This with this. This with this. This with this. So it means that Jenny is a suspect for that case, whose DNA profile is matching with the DNA sample obtained from the crime scene. And the technique which was used, it's DNA profiling technique is DNA profiling, and what is being shown here is a DNA profile. So you must know the difference. DNA profile is not a technique. DNA profile is what you obtain when you analyze the DNA sample. So who was the possible criminal? We have seen it is Jenny, because the DNA profile, or the DNA profile of bars, they are of sample. DNA sample profile is matching with the profile of Jenny, or the band of DNA profile sample, they are matching with that of Jenny. So set 1 other use of DNA technique, or state other use of the technique identified in the in the question above, meaning that the DNA profiling, what is it used? Can be used to identify the relatives. They have talked about the criminalism, so you don't talk about that anymore. So talk about the identification of the perfect organ donor, You can talk about lost relatives. You can talk about diagnosis of the inherited diseases. So there are so many uses you can use to when you are dealing with the DNA profiling. So it's the same thing. You see that is the same thing. I don't usually take too much time here because these questions are really open. Who is a suspect? You know how we have identified it. What diagram is DNA profile? What does the diagrams above represent? They're not saying the technique, they're asking for the diagram. Therefore, it is DNA profile, not DNA profiling, because here they said which technique, name the technique. So it's DNA profiling, but here it is DNA profile. Which suspect? Look at the bands and then you match them, give a reason. So the suspect, if you look at them, you find out that Q is most likely to be the suspect because the bands of broken glass, they are matching with that of Q. So they're saying that name 2 possible disadvantages of using this evidence in the court. It might not be accurate 100%, because number one, they can human error, number one, and also it may not be unique if a small sample is used. Also, they can a human being can manipulate them, so it may not be 100% accurate. So we have seen many questions concerning about DNA profiling. Download this and then try to do these questions. Where you get stuck, come back to me. Ask me questions. I'll be able to help you out. My number is always on the website 084-833-1738. Yes, and then speak to M. Saidi, who is always ready to help you out. Then another question is still the same tablet. Whenever you talk about tablet, make sure that you draw a nice table, and then so that you get that 1 mark. That's why there are 5, because they're asking for 2. They give you 2 marks there, and then 1 mark for tabulating. Describe the process that is being represented or responsible for formation of molecule B. So here, molecule B is messenger RNA. Why messenger RNA? It's because of this. So this is not, you see, this is messenger RNA, which is uracil. Therefore, and also 1 strand is being used. So that's evidence to show that this is transcription, because they're saying this is non-coding strand. So it means that only this is acting as a template. Yes. So they're saying that describe, so what are they looking for here? Describe the process that is responsible for formation of B. If you look at B, is messenger. So you describe. How do you describe here? Let me just talk about it. As I told you, talk about DNA double helix, unwind, weak hydrogen bond, break, and unzip. So when the weak hydrogen bond break, it's the same thing saying unzip. So you get 1 mark there. To form 2 separate, the word separate must be there, 2 separate strands. Only 1, you see it here, that only 1 is acting as a what? Is acting as a template. Only 1 is acting as a template. So it's being shown there that this one is acting as a template, and then using free floating messenger RNA. So you have to write messenger RNA nucleotide, yes, to form a complementary strand where adenine goes with uracil, and then guanine goes with cytosine. Adenine goes with uracil, and then guanine goes with cytosine. The complementary strand move out of the nucleus via the nucleopore to the cytoplasm on the ribosome. So basically, that's what you're supposed to explain. Please don't try to bridge it, because it's a process. 1 step leads to the other. If you start with what is on at the bottom and then you start with it, then you lose marks, because the sequence is not the same. So you cannot start with the last step and then you end with the first step. You have to start with the first step and then you end with the yes. So molecule B, we said it is messenger RNA. Name the process is transcription. Explain how the protein molecule would be affected if codon GUU is changed to CGC. So if here it depends on two things. If there is a table, yes, now you compare the table and see if it's gonna lead to a different amino acid. But because now this is open, then we assume that it's gonna be a different amino acid. For example, if you say that you started with GUU, yes, and then GUC, from this to that. If this, let me explain this. If this is tryptophan, yes, codes for tryptophan, and this one also codes for tryptophan. However much this has been changed to this, is going to code for the same amino acid. Therefore, the functioning of the protein is not going to change. But if this tryptophan and then now this one codes for example, leucine, then it means that there will be a change in the functioning of the protein because this is tryptophan and then this is leucine. Therefore, a different protein will be changed, will be formed, and then still the functioning of the protein will also change. So explain how the protein molecule would be affected if this is changed to this. So therefore, you tell us that if GUC is changed to GUC, GUU is changed to GUC because they say that codon this changed to, because this, when they say codon, it means that we have started from the messenger RNA. Then this will lead to different sequence of amino acid. Different amino acid will be brought to the molecule or to the messenger RNA, not messenger RNA, will be brought to different amino acids will be brought to ribosome, and then a different protein will be formed. So that's basically you have to understand. And then when you come here, this is translation because it is occurring on the ribosome. So what is A? A is messenger RNA. What is B is the ribosome. And then what is C? C is the transfer RNA. What is Z? Z is the amino acid. So this is the monomers, amino acids, monomers of the protein. And then this is the transfer RNA. You have seen it. And then this is the ribosome, and then this is the messenger RNA. So basically, you need to do that. And then what is this? These are 3 bases. So therefore, these are codons. Yeah, remember, the 3 bases on the DNA, we call them codes. And then 3 bases on the messenger, we call them codons. And then 3 bases on the transfer, we call them anticodons. Organelle X, what is organelle X? We have seen it. We have seen all the questions. You see now, when you start with trying to label them, you'll be able to answer all the questions. Molecule W formed, you see, organelle Z, X, we have seen it, ribosome, monomer W, the monomer. We are saying the monomer of molecule W. Remember the building blocks, that's what we call the monomer. Therefore, it's going to be the nucleotide. Be the nucleotide. Yes. And then molecule W and Y, W and Y, we have seen it. W is messenger RNA, and then Y is the transfer RNA, and then molecule organelle X, we have seen it is the ribosome. So still the same thing. You can see some people, they call this a protein. This is not a protein. It is only a protein if it is 50 and above amino acids. So what is this then? This is a polypeptide chain. Yes, so it is a polypeptide chain. So what is X? This is the amino acid. And then what is this? This is transfer RNA. And then what is Z? We have seen them. These are anticodons or anticodon. And then this is messenger RNA, and the 3 bases on the messenger RNA, we call them codons. You see, you can't fail such questions. When you come to the question, you see that they're asking you the exactly the same thing, and then asking you describe the process of transcription, which we have described. So basically, at least you have some light on DNA, and then maybe we will talk about the process of translation, and then after that we wind up with this coding and then the mutation. These are some of the short question which you need to know before you go for the exam. Number 1, DNA replication. You have to know DNA replication, the process by which DNA makes the exact copy of itself. Where does it occur? We have seen it in the nucleus. When does it occur? We say that it occurs during interphase. And then we said, why does it occur? We say that to double the chromosome number. And then how does it occur? That's the process of DNA replication. And don't forget that here you have to explain that here are the tools. You form 2 separate strands, and each strand is acting as a template, and that is the major difference between DNA replication and transcription. So in most cases, people confuse transcription and the DNA replication. Then number 2, describe the location and the structure of DNA. Here I explained it at the beginning, the structure of DNA. In most cases, give it 6 marks. And then location, we have 3 locations, that is nuclear DNA, mitochondrial DNA, and chloroplastic DNA. So if they talk about in plants, then you talk about all of the 3. But if they talk about in animals, you only talk about the 2, that is the because in we don't have a chloroplast in animals. So describe the structure of RNA. Structure of RNA, it's a little bit the same as DNA, but only few things which are not there. That is the thymine, the double strand, and then also the weak hydrogen bonds. Describe the involvement of different types of messenger RNA process in protein synthesis. Here they are talking about the functions of each. For example, you talk about transcription and then translation. So it is like you are describing the whole process of DNA of protein synthesis. Another thing you need to know here is also describe transcription in detail, and then also translation also in, yes. And then also what they can ask you is why is the mutations which occur. And I say that if you change the code, for example, from GUA to GCA, then if these two they code for the same amino acid, therefore the protein will not change. The structure of the protein will not change. But if the different amino acids are being coded for, then the structure of the protein will be changed. So let's go to meiosis. Let's go to meiosis. There are so many questions which can talk about DNA. And also the base calculation, whereby we're supposed to talk about if adenine is equal to 10%, what is the percentage of guanine, or the ratio of guanine, whereby you have to tell us that adenine goes with the thymine, and then cytosine goes with the guanine, whereby if this is 10, or definitely this is 10, and then 10 plus 10 is equal to 20. Since this is percentage, therefore I'm gonna say 100 minus 20, which means 80%. And then 80%, so what happens? So it's gonna give us 80% for these two, so divide by 2, and then from 40%, therefore this is 40, then this is 40. So this is just a simple recall. I hope you remember. Please don't go without knowing it, because at least those 2 marks you'll be able to answer them. And then if they are looking for the ratio, you can put them maybe adenine now. Adenine, we said it is 10, and then maybe with the guanine we said is 40. So divide by 10, and then we're gonna get 1, 2, 4. So sometimes they swap it and they say 4 to 1, and then because when now you have seen 1, and then you have seen 4, you have seen 4, you have seen 1, you just circle that. It is what? It is wrong. So when you go to meiosis, so many point where crossing over takes place. We call it chiasma. You see a chiasma. Splitting of the cytoplasm, we call it cytokinesis. Okay, we can answer them. Failure of the chromosome to separate during meiosis, the non-disjunction, the structure in the cell that forms spindle fiber, centrioles, and then the phase of meiosis where homologous chromosomes are aligned along the equator, that is Mera phase 1, because they're saying homologous chromosomes. And the point at which the 2 chromatids of the chromosome join, we call it centromere, and this division of the nucleus. Division of the nucleus is karyokinesis. And then they're saying that the site of meiosis in a female, because in female we find meiosis in ovary. So you write ovary there, ovary, and then the exchange of genetic material between chromatids in the homologous chromosome, it is crossing over. And then they are saying the genetic disorder caused by extra chromosome on chromosome number 21, that is Down's syndrome. Yes. And then they are saying that the structure formed between the centrioles by the centrioles during the cell division with spindle fibers. Yeah, spindle fibers. And then they're saying that non-sex chromosomes in human, we call them non-sex autosomes. And then they're saying the condition in the cell where there is only one set of chromosome. How do you call that condition? We call that condition haploid. And then the structure that is responsible for formation of the spindle fiber during spindle fiber, we say that is called the centrioles. Yes, and then the A chromosome aligned along the equator, none, because here it must be Mera phase. Random arrangement of chromosomes, it must be Mera phase. So it's A, B only. The site of meiosis in plants, anthers, and ovary, both in, this is male, this is female, and then the process of meiosis produces meiosis result in 4 haploid daughter cells, 4 diploid. So this one becomes wrong. So this is the correct one. The mechanism to introduce genetic variation, crossing over, is correct. Random arrangement of chromosome is also correct. So it is both A and B. So basically, you see, yeah. So now we are starting the other part of meiosis. All right. Please don't forget to subscribe. Stay in touch. If you have any problem, we need to smash these questions, and then we get all the marks at the end of this period. Yeah, when you come to meiosis, there are some questions we can talk about. Let's see what we can go through so that they can also benefit you in answering questions related to meiosis. I know it is a simple topic, but is a little bit challenging to some other. But let's see if we can smash these questions, and then we get a better understanding. If you don't understand the topic, let me know. I'll help you out so that at least we can have more distinctions in this year. All right. They are saying that here you are seeing diagrams. What comes into your mind is the kind of phases which this diagram is indicating. So what is C? C is the spindle fibers. What is B? This because there are two chromosomes of the same kind or the same size and structure, therefore is homologous chromosome. Then the joint between two, or the point, the joint between two chromatids of the same chromosome, it is a centromere. And then diagram A, what does diagram A represent? Because of this one is starting to form, so it can represent prophase, prophase 2. Why prophase 2? Why not prophase 1? It's because now this, they are individual, they are not in pairs. And then number 2, you see that crossing over took place a long time ago. And then this one is metaphase. The chromosomes are aligned. If you turn this, and then comes this side, and then this goes that side, so that this thing is in between your chest. So you see that if this is one side of the chest, and then this is on the other side of the chest, I think now you see what I'm trying to say. So you see that they're in the middle. So why? These are the spindle fibers. Yes, if this is the spindle fiber, if this is the spindle fiber and this is the spindle, sorry, centrio. So these ones are in the middle. That's why you say that it is under metaphase. But metaphase, which metaphase? It is metaphase 1, because these chromosomes are in the middle. Then they say identify A. We have seen it. A is centromere. B, what is B? B, we have seen it homologous chromosomes. Then C, you see, we have seen it as a spindle fibers. Identify the phases in diagram 3. Diagram 3, because they are separating. Yes. So if they are separating, then they are moving to opposite poles. You see them? They are moving to opposite poles. Therefore, it is anaphase. But which is it? Anaphase 1 or it is anaphase 2? Is it anaphase 1 or anaphase 2? Because now there are chromatids which are moving to opposite poles. Chromatids moving to opposite poles. Therefore, it's going to be anaphase 2. Anaphase 2. And then they're saying that if it was anaphase 1, then this would have been individual chromosome. This would have been a chromosome moving to the other side. Also, this one would have been a chromosome, and the centromere there, this would have been a whole chromosome, and also this would have been a chromosome. So it would have been anaphase 1. But because their chromatids moving to opposite poles, therefore it is anaphase 2. Then they're saying that write down the number of the diagram shown in the sequence which the phase occurs. The number of the sequence. They're saying write down the numbers of the diagram to show the sequence in which. So basically, we have to look for meiosis 1. The chromosomes are supposed to be in homologous pair. So this one becomes, this is homologous pair. So it is 1. Therefore, it is what? It is metaphase 1. And then this one is prophase 2. And then after prophase 2, they separate, which is, so it's going to be 1, it's going to be 2, and then it's going to be 3. So when you are writing the numbers here, it's going to be diagram 2, diagram 1, and then diagram 3. So it's going to be diagram 2, diagram 1, and then diagram 3. Then one difference between metaphase 1 and metaphase 2. In metaphase 1, the chromosomes are aligned along the equator in homologous pairs, while in metaphase 2, chromosomes are aligned along the equator singly. They are individual chromosomes are the one which align along the equator. So that is the major difference. And then if we see another question, so label it. Very simple. You see, we usually bring the same kind of questions. So this is the spindle fibers. This is the centromere. This is chromosome. Non-homologous chromosome is one chromosome, and then this is the central, centrio, which is being produced by centrosome. Okay, centrio. And then what causes these differences in the chromosomes or chromatids is because of crossing over. When does crossing over takes place? It takes place during prophase 1. How does it take place? Homologous chromosomes, they align together. Chromatids of the homologous chromosome, they cross over at a point called chiasma, and then they exchange the genetic material, and then they separate to form different chromosomes which are genetically different. Basically, that is the process of crossing over. You can't go to the exam without knowing the process of crossing over. Identify the phase of meiosis in diagram 1. Diagram 1, because now, yes, the chromatids are separating, is the same thing like this one. It's the same thing. The same thing like this is exactly the same. Yes. So it's gonna be anaphase 2. And then this one is going to be metaphase 2 because the chromosomes, they aligned along the equator singly. Yes, they align along the equator singly. Then they're saying that identify the phase. You are done with that. State what happens to structure D in the next phase. Where is structure D? Structure D, these are the chromosomes. So the chromatids, they move to opposite pole due to contraction of the spindle fiber or due to the shortening of the spindle fiber. Name the process during which genetic material were exchanged or was exchanged. We say that the process is called crossing over, and then we said it crossing over, and then we said it occurs during prophase 1. State the consequence if the mentioned does not occur. If this process does not occur, it means that organisms it will reduce what we call the genetic variation. It means that more organisms will be more similar to each other, so it will reduce variation among the organisms because crossing over leads to genetic variation. Then they're saying that give the number of the chromosome present in the original parent cell in this diagram. They didn't say this is a human cell. I don't see anywhere there saying this is a human cell. So you have to know, you have to use the number which is being given. So because this is meiosis 2, and meiosis 2 leads to the diploid state being halved. So for example, you start with 1N, and then you form 2 cells, which is 1N, and then another 2 cells you divide to form 1N, 1N. So if you start with the 4 here, therefore it's gonna be 2, it's gonna be 2, it's gonna be 2, it's gonna be 2, it's gonna be 2. So it means that this is meiosis. Therefore, this is 1, this is 2. Therefore, the total number is 2. Please don't talk about 46 if they didn't say that this is the human cell. And then now they are saying original cell. So if it is original cell, therefore it's gonna be 4. And then you see now they are saying, specifying it, human cell in the sample diagram 2. Because diagram 2 is haploid, therefore it's gonna be 20. It's gonna be 23 chromosomes. So basically, that's how you're supposed to answer such questions. So you can answer these questions, at least in meiosis. What is A? A, in most cases, students confuse it with a nuclear membrane. This is not a nuclear membrane. You can't have a nuclear membrane when the cell is under a metaphase. Therefore, this is the cell membrane. This is the chromatid. This is the chromatid. This is the spindle fiber. And then this is the centriole. So this is metaphase 1, while this one is anaphase 1. Why? Because chromosomes are separating. What about this? This is the telophase 1. Why? Because they are chromosomes. They are chromosomes which are still double-stranded. Yes. Or at the beginning of prophase 2. Yes. So you see that now once you name that, you are able to answer this question. What is phase 1? We have seen it is metaphase 1. What is 3? We have seen it is telophase 1. Then you're saying the letter only of the part contains DNA, contains DNA. Give the letter of only part that contains DNA. It is B, because in this case they didn't mention any chromosome here. It's only B, which is chromatid, and then attaches the centromere of the chromosome, attaches the centromere chromosome, which is C, attaches is the centromere. Yes, and then they're saying that forms spindle fiber, which is the centriole. Where is the centriole mentioned? Is D. You see, we are able to answer these questions. Name the organs in humans, male, where meiosis occur. In humans, male, it occurs in the testes. While in female, it occurs in ovary. In plants, it occurs in anthers, that is male, and then stigma, ovary in female. So here they produce sperms, they produce ovum or ova. Here they produce pollen grain, while here they produce ovules. Yes. So basically that's a simple illustration of meiosis occurring in plants and also in animals. Describe how meiosis leads to genetic variation. So now here you are talking about only processes which talk about genetic variation. Here you talk about crossing over. So you describe crossing over. Then you talk about the random arrangement of chromosomes during metaphase. Metaphase. Yes, so basically that's what you need to explain. It can lead to genetic variation. You can also mutation can also be part of meiosis. Why? Because if you talk about Down syndrome, it occurs during the process of what? Occurs in process of meiosis. But these two, they can help you to get those marks you are looking for. Here is an example of a karyotype. You have two individuals. Identify which karyotype. Here, when you given a karyotype, look at, is there any abnormality? Which one is the male and which one is the female? So if you look at this, no abnormality, all of them, they are in pairs, but if you look at that, one is long and then one is short. Therefore, this is X, this is X, and then this is Y. So therefore, this is a male. And then both of them are the same size. Therefore, this is a female. So now from there, you can now go and then try to answer. What term is given the chromosome number 1 to 22? So we call them, because they can ask you to describe the structure of a karyotype, you call them orosome, the orosomes. And then 23, these are gonosomes. Why? These are sex chromosomes. Yes. State the gender of P. We have seen it. P. Where is P? Male, because of this. Give one observable reason, because the chromosome number, the chromosome number, the chromosome pair number 23. One chromosome is bigger than the other. Therefore, one is X, one is Y. Hence, the person is a male. Then they're saying give one observable. We have seen it. Each of the pairs shown is a homologous pair of chromosome. State the origin. State the origin of each chromosome in the homologous pair of the chromosome. So here, where do they are two. Where are they coming from? These pairs, one is coming from the father, from the father, and then one is coming from the mother. Yeah, that's why there are two. That's why we say that when the sperm comes, is haploid, and then fuses the ovum, which is haploid. So one chromosome, another chromosome, when they combine, you form the pair. So it's the same thing. Sperm, ovum, sperm, ovum, sperm, ovum, sperm, ovum, like that, like that, like that. So one comes from the father, one comes from the mother.